You know that Everybody, and welcome to NC State is a land-grant institution for the people of North Carolina, and we respectfully acknowledge the lands within and surrounding present-day Raleigh that are the traditional homelands and gathering places of many indigenous peoples, including 8 federally and state-recognized tribes, the Coahary, Eastern Band of Cherokee, Haliwa-Saponi, Lumbee, Meherrin, Occaneechi Band of the Saponi Nation, the Saponi, and Waccamaw-Siouan. We share an ongoing responsibility to safeguard these lands, to respect the sovereignty of the tribes and indigenous nations residing in North Carolina. NC State honors all indigenous peoples who have been and continue to be an integral part of our university's history and culture. So I'm Jennifer Kuzma. I'm a professor in the School of Public and International Affairs and co-director of the Genetic Engineering and Society Center here at NC State. I am also Associate Director of our NSF-funded Engineering Research Center, Precision Microbiome Engineering, or PREMIER. And I co-lead PREMIER's Societal and Ethical Implications Core with Professor Joseph L. Graves, Jr., who is here today and will be giving the closing remarks. First, I want to thank you all for being here in person or online. We have over 100 people registered between these two venues, about 60 online and about 40 in person. Your time is valuable, and we greatly appreciate your participation in the workshop. I also want to thank my workshop co-organizers, Dr. Kristen Landreville, Sharon Stauffer, and Patty Mulligan, who did the bulk of the work to make this workshop possible. In fact, I was thinking on the drive-in this morning that if I were to get sick today, they could probably do without me, but if one of them were to be sick, this workshop would probably fall apart. So I really want to thank them. So microbiome engineering of the built environment to improve health is a relatively new area of pursuit. As many of you know, societal and ethical implications are often given little attention as new technological areas develop. But this hasn't been the case for PREMIER, which is striving to center SCI work to guide its effort. So I want to thank Dr. Claudia Gunsch, Director of PREMIER, who will be speaking shortly, for recognizing the importance of SCI in the design of the center. I also want to thank Vice Chancellor of Research and Innovation at NC State, Dr. Mladen Volk, for supporting the work of the SCI Core, our GS Center, and the rest of PREMIER at NC State. The nexus of the built environment, microbiome, and SCI is a very young area of inquiry, and we have some of the world's and nation's best experts in that area today. But through this workshop, we will be paving new ground as we bring together these diverse expert stakeholders to apply their knowledge from more established areas to this nexus. We will cover societal aspects such as social equity, informed consent, ownership, privacy, bio and research ethics, risk governance, regulation, public participation and engagement, and other areas over the course of today and tomorrow. So, the goals of the workshop on this slide are threefold: to grow a community of people investigating SCI and microbiome engineering of the built environment, to bring SCI experts together in conversation with the scientists and engineers of PREMIER in order to instill SCI thinking and practice into PREMIER technologies before and as they develop. And finally, to co-develop with you all an SCI research integration and action agenda for microbiome engineering of the built environment. And because we have a lot of speakers and a really tight schedule, I'm going to stop my opening remarks here and thank you again for your presence today. So the next slide has an overview of the agenda, and today we'll have 5 different sessions, and tomorrow will be more of an in-person deliberative workshop where we'll co-develop the research integration and action agenda. In our first session, we have 2 speakers, and as a reminder, their full bios are available online, or there's a QR code in your packet where you can scan it and a link to their bios. And after their presentations, we'll have some time for Q&A and discussion. So first up is Rob Dunn, Professor Rob Dunn, Senior Vice Provost for University Interdisciplinary Programs at NC State, who will be giving opening remarks. Rob has done extensive, novel, and high-profile work in public science and microbiomes. Then Professor Claudia Gunsch, Director of PREMIER and Professor of Civil and Environmental Engineering from Duke University, the leader of our ERC, will present on the state of science and engineering associated with built microbiomes. So thank you both for being here. Thank you all for being here. And Rob, Are you ready to start us off?
Let's go. Thank you, Jennifer. So I've got 10 minutes, and I'm going to try to go quickly. And I was trying to think about how can I be maximally disruptive in the first talk, disruptive in a good way. And so I thought it would be interesting to bring a comparative approach to thinking about microbiomes in houses. And so I'm going to start with the comparative approach in general and sort of beg the question for the day, what is normal? What are our baselines as we think about doing interventions in houses? And I'm going to be thinking about evolutionary baselines. And so I'll walk quickly through some examples of work we've done. The first is on stomachs. The classic story of what our stomachs do, especially in humans, is they help us to break down proteins. And so they're part of digestion. This is clearly only a partial story, as Deanna Beasley was able to show a number of years ago. Deanna was really intrigued because if you looked in the clinical literature, there was a bunch of work in medical context in which people with reduced stomach pH were more likely to get sick from pathogens. Deanna started to think, what if the bigger story is actually that part of what the stomach evolved to do is to kill pathogens from your food? If that were true, you would expect species that eat food with more pathogens in it to have more acidic stomachs. And I'll share these slides. She found this to be true. Long story short, that the more likely your food is to have pathogens, the more acidic your stomach is. It's a bigger study. But one of the wild take-homes is where humans fit in this story, is that we have stomachs that are closest in pH to the stomachs of vultures. This is an interesting evolutionary reality. It's very different from other primates. Only one chimp ever has been studied with regard to stomach pH. We're about to change that. Its stomach was neutral. And so first of all, something interesting has happened in human stomachs evolutionarily. What is it? We don't yet know. But also with regard to application, and this is where I'm going to try to pull these examples around, any procedure or medicine that raises the pH of the human stomach makes it more neutral, increases the risks of pathogens surviving to reach the intestines. And so this is medically important because we alter pH in medical contexts all the time. It's also important because it's probably the case that it's much more difficult for humans to manipulate their microbiome as adults than it is for many other species with less acidic stomachs. I'd love to talk about the large intestine. Ask me about it later. It's one of my favorite parts of the GI. I'll just say that Amanda Hale and Aaron McKinney, who Claudia mentioned earlier, have a great paper in which they show that there's much more variation in human large intestine morphology than anyone noticed, and that most of our comparisons of humans to other species were based on 4 people. They studied 36 cadavers at Duke Medical Center and found more variation than was known in the entire literature that changes how we think about large intestines and shifts our baseline. Skin is super interesting as a biological part of our world. Amazing Ron Weck sculpture. Biologically, skin is super rich. The standard story of our skin, human skin, is it's covered with Staphylococcus bacteria. This is a real image, false colored by Martin Ogerle. But those are Staphylococcus, the blue there. And so that's medically normal. That's our baseline. That's what our interventions are relative to. But we started studying this a number of years ago in many contexts, including armpits. Armpits are super interesting microbiologically. We have glands in our armpits that evolved to do nothing other than to feed microbes. Longer story. They actually weep. They weep food for microbes. The microbes metabolize that food and produce aromas. Armpit hair is different because it's there to wick those aromas. What does the microbiology of those armpits as part of the skin look like? We started to study that with Julie Horvath Roth a number of years ago. One of the things we quickly saw is that the armpits and skin of chimps, gorillas, monkeys, and humans are very different. That most monkeys that have really reduced apocrine glands in the armpits have really biologically diverse skin microbiomes that are not dominated by any particular thing. But if you start to look at gorillas and chimps, you see a lot of Corynebacteria, which is what armpit glands most feed. It's a slow growing, it's like the beech tree of your armpit forest. But then if we look at humans, we see way more Staphylococcus and we see huge variation person to person. What's going on there? It looks like a big part of that variation person to person is antiperspirant and deodorant use. Antiperspirants and deodorant kill off that Corynebacterium, favor Staphylococcus. This was a huge intervention in our skin microbiome, and it actually affects the skin in general. Nobody approved it, didn't have a regulatory process, radical change. It probably makes you more attractive to mosquitoes. Vaginal microbiomes, Elizabeth Miller and Beth Archie at Notre Dame were starting work on baboons. And they had the observation that in baboon vaginal microbiomes were roughly neutral. And so we started this conversation about what does this look like across mammals in general. Turns out the paper had not been written, and so we dug into to look at the comparison. And it turns out almost all mammals have neutral vaginal microbiomes, which is super interesting because the human understanding of vaginal microbiomes is that neutral is a disease state. Something interesting is happening here. Chimps have slightly less neutral vaginal microbiomes. Humans have very acidic vaginal microbiomes. This is largely because of lactic acid bacteria that are producing that acid. Some big transition happens here. It looks like part of what's happening is that human vaginal communities, there's lots of glycogen compared to other species. That glycogen feeds the lactic acid bacteria. Why is it there? It has never been studied. We have no idea. I have hypotheses. This is where the literature sits. And so whatever normal is, we don't fully understand it yet, and no one's ever looked at vaginal microbiomes in populations that are not historically agricultural. No studies exist that I've ever seen. Clinical importance, normal pH and ecology of human vaginal microbiomes may really be normal for agricultural peoples, maybe a contingent on diet. There's part of this story that we're clearly missing as we think about interventions. Fermented foods, I've worked a lot on fermented foods. I'm going to summarize like 5 years of work in 2 slides. Fermented foods are a way that we have been intervening in household microbiomes for at least 14,000 years and probably much, much longer, recent evidence suggests. And so we've been interested in what does this intervention look like, how controlled is it, and I will summarize quickly by saying that traditional fermenters are extraordinarily talented at using microbes to achieve states that they would like to achieve in foods, and by virtue of the spillover of foods into houses, the biology around houses. Extraordinarily talented, and we paid almost no attention. And one of the things that we wanted to do when we were doing this work, to circle this back, is to figure out how much interplay is there between the house, the body, the food, the flavor. And so we did this study with Pajau Center for Bread Flavor in Belgium, where we brought together bakers from around the world. They all made the same sourdough starter, and what was different was basically their hands. And we were looking at, is there an effect of hand flavor? This came from a Korean tradition, solma, which is the hand flavor, which is what the chef gives to food. Is this, is there hand flavor in baking? And so we did this big experiment. Bakers are impossible to work with because they want to bake. They don't like pauses. But we did it. And so the baker on the bottom right, he was supposed to make the same bread as everybody else, and he turns around and that's what he'd make. I'm like, oh, come on. And so what we find is that yes, that there's an effect of the baker's hands on their sourdough starters, and you can detect that in the flavor. But what we hadn't planned to study was whether the baker's hands themselves were normal. And what we found was that their hands were covered in yeast, and most hands in studies, which is to say the hands of most college-age students in the US, have no yeast or very little Saccharomyces. These bakers were covered in it, and they had up to 80% lactic acid bacteria. And so their bodies had basically become starters. There's anecdotal evidence that it's their whole body. And we later asked them, like, if you put your hands in water, does it start to smell acidic? And they're like, yeah, everybody's does, right? I mean, this is, but the question here, is what's the baseline? So historically in agricultural traditions, most of us would have been engaged with fermented foods. Most of us would have been engaged with these microbes. Is the baseline for the agricultural tradition wrong? We have no idea at this point. And then circling back to the house, and I think I can land on time, we spent a lot of time studying the life in houses around the world. We have a new study with Jane Oja in Estonia of, I think now, almost 1,000 houses from countries across environments globally. Love to talk more about that at some point. We did lots of just going into houses. We did lots of sort of DNA-based studies of houses. Found thousands of species we didn't know about, of fungi, of bacteria, not surprised to this room. As far as I know, no one's ever studied protists in houses. And so just as a measure of our blind spots, unless somebody in this room has, we know nothing. They're there. Nematodes, not consistently. What we found is that these environments where we try to kill things, that they're often the richest and unusual microbes. So hot water heaters, fridges, freezers, they're amazing, wonderlands of life. And then we see that there are effects of control in these environments. And I wrote down what I wanted to say here. like this, what I think that if I were to sum up what we found about houses, is that what we've done in the last years is the lack of precision microbiome engineering. We've engineered the microbiome but with a perfect lack of any awareness of what we're doing. And so what do we see has an effect on microbiomes in houses? You know, lots of things, but male to female ratio, armpit versus vaginal microbiomes. You know, if you have a room with lots of men in it, it's just like a giant armpit, and we can detect that forensically. Almost certainly that's modulated by product use and diet. Fermentation affects what's in the house, at least in the kitchen. Dogs have by far and away a bigger effect than anything else we've ever studied, and it's consistent. Cats, a little bit on fungus. It's not huge. It's not very predictable, but dogs, they're a big deal. Windows, AC, you know, we and other people have found this. You know, how do you let air into your house? It has an effect. These are all relatively guileless interventions. Like these changes in who lives in your house, how do you open it up, but they have big effects. I could talk more about that, but to me the most extreme example of this is that we had Jonathan Eisen and team at UC Davis did a study of the International Space Station using our same methods, and so we sent them the swabs, and so it was perfectly comparable with what we did. And so what is the space station, which is our engineered built environment, look like? And the short answer is it looks a lot like a toilet seat. And so this is our precision engineering of the microbiome right now, is that we're able to make a room like this be a lot like a toilet seat. You know, who's to say what we want, but that's probably not it. And so what is the norm? What are we shifting the microbiome relative to? And I'm just 1 minute over and I'll close here. Megan Temis did a study of chimp nests a number of years ago to figure out what the microbiome looks like in chimp nests. My guess when she started this work is it was going to be chimp pathogens, chimp feces, chimp ectoparasites. It was nearly exclusively leaf and soil microbes. And so from an evolutionary perspective, until very recently, certainly for 8 million years, that's our baseline, leaf and soil microbes. And so whatever we're talking about doing now, it pales in comparison to the transition between this and the toilet seat of the International Space Station. And I'll close with one final slide here, which is to say, I think the other thing comparison offers us is other examples. And so there are many species that live in societies that manipulate their microbial environment. Some are really loose and manage a diversity of microbes. Leafcutter ants manage a central fungus in their nest, which they eat, walk on, live on. They manage bacteria on their bodies that they use as pesticide to control that. They're monocultures. And so here is an alternate example from nature. It's not where we want to go, but I think it's useful to think about what are our possible things, where's our baseline, and I'll close there. And then here's where Jane's work is coming in. Those are her samples for the next generation of what she's doing with microbes in houses. Thank you.
No, that's fine, that's fine. So just as a reminder, we'll take questions from both the online and the in-person after all the speakers have gone. So next up is Dr. Claudia Gunsch.
Perfect. Well, it's a pleasure to be here today, and I want to thank Jennifer and her team for organizing this meeting today. So as Jennifer mentioned during the introduction, I'm Claudia Gunsch. I'm a faculty member at Duke, and I am a professor of civil and environmental engineering. So I come to these questions really with my environmental engineer hat. As I'll talk about today, some of my background is really in bioremediation, so thinking about how we can engineer microbiomes with an eye on detoxification. And it was through that work that I had a few experiences that led me to start this work. So I thought I'd back up. Rob showed a number of examples and vignettes of different microbiomes that are important. But taking a step back, for those of you that maybe aren't as familiar, when we think of microbiomes, we're really thinking about all of the microbes in a particular system. And so here we're talking about bacteria, we're talking about fungi, we're talking about viruses, and all of these different components play a very important role ecologically within that system. And so we know microbes are everywhere, as the examples that were shown in the opening presentation. And here I'm showing a couple of examples in agriculture where they're important in terms of nutrient cycling, crop yield. And so we know that this is the case, but we don't always understand what are the driving factors. And that's really what we're trying to understand. So hygiene hypothesis, so you are probably very familiar with this. We know that some microbes are bad, right? We know that there are pathogens that exist, and so we've gone to great lengths to remove those microbes, but we do it in a non-discriminate way. And by default, we kill everything, and this is true. We bleach things. We put all of the hand sanitizer on our hands, and that kills everything. In health, we utilize antibiotics, and that kills all of the microbes. Of course, it kills the ones that we want to kill, but it also kills a lot of other things. And so there's been a lot of work in the medical realm looking at what the impacts of this is. And there's a general understanding and acceptance that the decrease in the number and the composition and the diversity of the microbes in our body is associated with negative health impacts. So this could be allergies, increase in inflammatory modulated disease, for instance, and there's a whole realm of these negative health impacts. And so we've started understanding how microbes and humans interact. And so some of those microbes will have will be pathogenic, and so they will have a detrimental impact. Some of them are commensals, meaning that they're beneficial, and so those will maybe produce something that a microbe in a community needs, and will have that positive impact, and mutualism, where both of those organisms need to be present. And so we think about symbiosis and how those microbes work together, and also how they interact with the human to lead to positive or negative health impacts. And so probably the best studied system, I would say, is in the gut microbiome. So we know a lot about how microbes interact in the gut. And for many years, we thought it was just the gut, but it's very clear that the microbes that are in your gut also interact with other parts of your body, with probably the best example being the gut-brain axis, where we know that the microbes in your gut produce particular metabolites that then interact with receptors in your brain. And so there's been a number of studies that have shown that your gut microbiome may have impacts on mental health or Alzheimer's, just as a few examples. And so we've utilized this information, and it's been in the media, it's been in lots of scientific papers, thinking about how the microbiome plays a role, and what we can do with this information. But the microbiome is important, but the other component that ties with, again, what Rob was saying in the introduction, is that we are not living in isolation. And so we are in the built environment, especially in the US, more than 92% of our time is spent indoors, and we are interacting with many components, all of the microbes that are on the surfaces and the air, the pets that we have in our homes, and there's a number of other factors as well. And all of this is what we call the exposome, which includes not only the microbial components, but also the chemical and physical components as shown here. So this could be metals, trace metals that are present, for instance, in the water that you drink, or volatile organic compounds that may be emitted from some of the products in your home. And we know that all of these factors are at play, and we often just study a small subset of them without thinking about the entire ecosystem as a whole. And so here, and I don't think I need to convince you since you're here today, but thinking about the microbiome of the built environment and how that plays a role within that broader exposome is an important and understudied and, well, not understood part of this equation. And so there was an interesting paper that was just published in the Proceedings of the National Academy of Science just I think it was last week or maybe 2 weeks ago, that was talking about this concept of healthy porosity. And I thought this was a really interesting analogy, and thinking about the human body that really has adapted over time. We have our immune system that really is there to help protect from invaders that come in and to try to maintain a balance within our body. And in many ways, you can think about the buildings that we spend our time in a very analogous way. And so there's microbes that come in and out of those particular spaces, and the humans that construct those spaces and operate those spaces have control of how we operate those systems. And what we've done over time is we've closed in these spaces for energy efficiency. We may put products in there that we don't understand how they may be emitting particular volatile organic compounds, for instance. And so we've, in essence, removed the outdoor from the indoor, and we've gone to great lengths to make sure those spaces may be more sterile than they should be. And the result of that is that this diversity, the microbiome diversity that I was talking about initially, has decreased. And so there's some efforts in going into thinking about interesting strategies about how do we bring the outside inside. And so this is biophilia, and so I'm showing here a couple of examples from this company, Grow Up, that has essentially green walls, and I'm sure you've seen this. And there are not only mental health and well-being benefits to this, but also, again, bringing some of these microbes in the indoor environment that may be beneficial. But the bottom line is we really don't understand what are the driving factors. If you don't operate these properly, you may, in fact, make things worse, right? You could bring in mold if you water them too much. And so we really need to understand how all of those pieces. It's clear that it's important, but we don't really understand all of those components. And so with COVID, I would say our environmental surveillance of microbes, in particular pathogens, has improved drastically. When our world was turned upside down when COVID hit back in 2019, 2020, we figured out how to monitor for the presence. And so this diagram shows this. We're really good at doing it within a hospital. We can do it at the community level. But what we're not as good at doing is, one, thinking about the microbes that are non-pathogenic, and thinking about doing it at a smaller scale. And the reason for that is that we just don't understand how all these microbes are crosstalking. And so there's been a tremendous amount of work in developing predictive models that are better at associating the different features that are present in a microbiome, but we still don't understand all of the pieces that are needed, all of the parameters that are needed in terms of having an inoculum, so having a microbe actually stick in a particular community. We really don't understand that. And so this is a great paper if you're interested in really capturing the depth and the breadth of all of the parameters that we need. I mentioned in the human context, we are way further along than in the environmental setting. And so there have been tremendous advances that have gone all the way to the translation and development of therapeutic interventions, as shown up here, with some actually approved interventions in the humans. However, within the microbiome of the built environment, we're still I would say, pretty far away from being able to do that at an aggregate level. And so when we think about engineering in the built environment for the microbiome, there's really three main parameters that are at play. The first one is physical, so understanding the materials and the coatings that we put on our different products and how those lead to colonization and propagation of particular microbes. The operational, so the plumbing systems, the air conditioning systems, how do those environmental factors that are associated with that lead to either the growth, again, colonization of those organisms. And then we have this opportunity to utilize biological interventions. And so I'm listing a few of them here. We'll be talking about these throughout this workshop. And I'll give some examples, some of these that I've used in my own research in bioremediation. So shifting to bioremediation and drawing the parallels for built environment, one of the strategies that we've used in our group is what is known as genetic bioaugmentation. And so one of the challenges that is a common challenge for environmental applications is you may have an organism in your lab that is capable of carrying out a particular function of interest. And when you insert that microbe into a complex microbial community, that microbe isn't able to survive. And so this is a very common problem in bioremediation. I have a library of microbes that can break down contaminants. When I go out to the field and I put it in that soil, it doesn't survive. And so one of the strategies that we've utilized is actually taking advantage of what are known as plasmids. These are extra chromosomal fragments of DNA that are present in many bacteria. And often the genes that are needed to break down these hazardous compounds are found on these plasmids. And so what we can do is take these donor cells that contain these extra chromosomal fragments of DNA, put them in the soil, and induce the transfer of that plasmid into the native microbes that have already evolved to be able to survive and compete in those environments. And so these are transconjugates. And so for bioremediation, the addition of these catabolic genes capable of breaking down these compounds is a beneficial feature. The flip side of that, if you want to think about this in a built environment context, and where we can learn from this, many of the antibiotic resistance genes are also found on these plasmids. And so these same natural processes of horizontal gene transfer are happening. And so we need to understand what is actually driving those transfer mechanisms so that we can interrupt that particular process. Another example is just utilizing all of these high throughput sequencing technologies to be able to get a better snapshot of what and who is present, what are the functions that those organisms are carrying out, so that we can then develop models and target particular recipients for genetic bioaugmentation, or strategies for understanding how we can stimulate the growth and propagation of beneficial organisms or reduce colonization of those organisms that we don't want in the system. And so a lot of the work that we're doing within the center focuses on thinking about the plumbing system. And plumbing is a really interesting system because it contains lots of biofilms, which are very difficult to engineer. They're associated with a huge number of infections, and we are trying to figure out some of these strategies and applications to that particular systems. So some of the strategies that we're utilizing, I mentioned the plasmids, so thinking about how we can work to prevent the transfer through horizontal gene transfer of these plasmids. We're testing bacteriophage, so utilizing viruses in a very targeted way to be able to kill specific pathogens or specific organisms. This is also a way for us to be able to deliver particular features of interest. And then finally, encapsulations. And so encapsulation is the idea that we can add particular microbes and they're protected. And so we don't have to worry as much about them not competing, at least not initially. And so you can imagine a future, and this is very much where we hope to be at the end of this center. You could imagine a time where we could actually sense for the presence of particular organisms that we know are associated with particular health outcomes, and engineer or implement one of these strategies that I talked about to protect human health in general. And so hopefully that's where we'll be. But you can imagine there's lots of societal and ethical implication questions that we need to address before we can think about that. So with that, I will stop. Not too much time. So, thank you.
Rob, do you want to? Yeah. So, if Rob and Claudia, you want to take a seat at the table, or so that we can field questions. So we do want to field questions both from the in-person audience as well as the online, but we'll start with in-person while our speakers are getting settled. So, yeah. Hang on, Chris, just wait for them. Yeah, thank you. We have a mic to pass around so that the online audience can hear this. Great,
here. It's more of a comment to something Rob said about impacting the microbiome in the house. I am actually one of them weirdos that looks for small things in my house like ciliates and tardigrades, etc.
Oh, amazing.
Wow.
Well, as soon as I retired, the first thing I did was buy a decent scope. But my point is simply this: one of the things that a lot of people don't think about, and I do now, whenever I do anything with the aquarium, with the fish tank, it is absolutely loaded with critters of all kinds, shapes, and sizes. And so I scrub well after I handle anything to do with that. And I have no doubt that that impacts other parts of the house, just because you got water, air bubbling through. Occasionally the cat might try to get up there. And I have no doubt that it changes other parts of the house that I haven't sampled yet.
I think mostly an observation, but maybe one quick response.
Rob, could
to
you
that
get maybe,
yeah, sorry.
unless, is that okay? Yeah, a little closer to the mic. There we go, thanks.
Yeah. So one observation from our work is that we've sampled I think 1,000 houses at this point, but there are many things people do in their houses that are rare relative to a sample of 1,000, but really common globally. And so I think we had 5 houses where people had indoor goats. So I don't know how we get, weird sample somehow, but it's not enough for us to say what's going on there, other than I suspect it has a huge impact, and that the diversity of things, the ways that people live in their homes is much greater than I think we appreciate from a university perspective. And so how do we build those into what we're doing and how we're thinking about? And I think the aquarium is a good example. You know, if you've got an aquarium in your house, it's obvious to think about it. But if you don't, it's not even, I don't think we even asked the question in our work. Yeah, so it's a good point.
Great. Yeah, David.
Hello, I'm David Resnik from the NIH. I'm sorry I'm arriving a little late. I did read the background materials, though. And so let me understand, some of the methodologies would involve release of genetically engineered bacteria into the environment to try to alter the microbiome?
So we're exploring a wide range of different features, and so that's really what we're going to be talking about today. But some of the features that we are considering would include that, but also just introducing a probiotic-like approach where we're introducing microbes or viruses. So some of it would not be genetically modified, but other approaches would involve, utilizing or applying CRISPR-type technologies as well. So it's a wide range of things that we're considering.
Okay,
Yeah,
thank you.
I like the way, thanks for the question, David. I like the way you broke it down between the physical, was it the coding, and then the biological. So all three kind of approaches, like HVAC plus the coatings yeah. So, yeah. But that's one of the things we'll be considering in our case study tomorrow for the in-person deliberative portion, as well as genetic engineering, yeah. Rob, go ahead.
Oh, no, I was just going to quickly introduce I would say, like, our understanding of the history of movement of microbes in houses suggests we've already done versions of this. And so, for example, we don't know yet, but I don't think Saccharomyces cerevisiae brewer's yeast is native to North America. And data-wise, it's debatable still, but my guess is that's what we're going to find. It's now one of the most common yeasts we find in nature outside of houses. And how was it introduced? It was introduced through bread, through beer, through wine. And so we've already done a lot of interventions that are worth thinking about and that we haven't studied very well. And so I think a complement to what you're doing is knowing what we've already done, which I think we've not taken sufficient care with. But per Claudia's point, the methods for taking care with those stories have fundamentally transformed. And so now ancient DNA and protein techniques allow us to ask the question, was Saccharomyces cerevisiae here pre-colonially? We couldn't have done it 5 years ago. And so I think we're in a fun time, yeah.
Yeah, and I'll just add to that a lot of the work that I do in bioremediation is exactly that. I mean, horizontal gene transfer has been happening forever, right? But we don't understand how to model it, to do it in an intentional way. So that, I think, adds to what you just said, Rob.
Great.
So, Rob, you mentioned.
Yeah.
So, Rob, you mentioned. Okay, there we go. So, Rob, you mentioned that you were looking at a question of unicellular eukaryotes, what people call protists, in households. So one low-hanging fruit in that regard has got to be Toxoplasma gondii, right, which is we should find all over houses of people who have cats. And it would be interesting to know how common is that in those spaces.
Yeah, I think it's a great question, and the data and samples are out there, and it's just one. For me, it's more of an example of how early we are in some of this understanding. That we have the approaches, we have the ability, and we have organisms that have a huge impact on our biology. Toxoplasma gondii clearly has the potential to influence human behavior in all sorts of ways. And so, what does it mean when it's in some places and not others? What do the big patterns look like? And so I think there are lots of opportunities. But I would also just flag that it is hard to overemphasize the extent of our ignorance at this moment, just in general, but also about the biology of where we live. Jessica Parzignat and the College of Veterinary Medicine recently did a study where she studied backyard chickens in North Carolina and looked at antibiotic resistance and found that the backyard chickens where antibiotics are not being used are full of resistant bacteria. And theory suggests they shouldn't be there because you shouldn't have a selective pressure favoring those cassettes and other hunks of genes that are producing antibiotics. And we don't know why they're there. And so I think as we think about this landscape, we have to have the give space to ignorance, give space to the reality we might not understand something fundamental.
Oh, yeah, go ahead.
Sorry. Erika Szymanski, I'm at Colorado State. Claudia, I'm so glad you flagged a metaphor that's all over the Premier website, which is great, of analogizing human bodies and built environments, analogizing essentially bodies and buildings. And you mentioned it again in this presentation, so I wanted to pick up on it and wonder whether, particularly with the two of you sitting next to each other, that's an analogy you're deliberately using, and how that plays out in terms of the approaches you're thinking with respect to healthy buildings, what a healthy building might mean. Is that something you're exploring in terms of defining health in this context?
So I think one of the big questions, right, that we've gotten is how do you define the health of a microbiome of the built environment? And we have this question in the human body: what is a healthy microbiome? And our healthy microbiome might be completely different. And so I think the analogy is one that we're thinking about, but taking it that much further is difficult. We don't know if the systems to model that will be analogous, but we know so much more about the immune system of the human that I think there are important lessons that we can draw from. So that's as far as I'm willing to go with that analogy.
Yeah, it's a super fun question to start the morning with. So I certainly use that metaphor, and if I'm being more careful with it, I think we also use the language extended whole genome, extended microbiome, extended holobiont to think about. So if there are many solitary bees that make a chamber into which they put their egg, and that chamber is typically filled with pollen or nectar. We now know that several of those species ferment the pollen and nectar, and so when the baby is born, it lands in a vat of beer, in essence. And so that to me is an extension of the body of the organism. And so if that's then the metaphor, then the dwelling is not a body, it is an extension of the body. And so I think it's useful to interrogate that over the next couple of days. Like, what do we want that metaphor to be? How should we think about health? Is health really a house and a body that together are healthy, in which case the details of the story of the inhabitants are more important than they would be in the abstract if the building is here and the body is here. But boy, that's a good question. Thank you.
We have time for one more question, I think, only. Was there any online, Patty?
Not right now.
Okay. Amber? Wait for the mic, please. I'm sorry. Sorry about that, everyone.
Amber Benezra. I'm going to speak next, so you'll hear a lot more from me. But I just think it's so fascinating and interesting to flag that the gaps in the scientific knowledge are exactly places where transdisciplinary collaboration can fill in, because how people live, if they have aquariums or goats in their houses, anthropologists have been living with those communities for decades, right? Like, there's a lot of social science knowledge about communities and how people live in their houses and all those sorts of things that can fill in those gaps, right? If we just figure out how to talk to each other, but that's a different story.
Great point. And so I want to thank the speakers again. You guys both did a wonderful job, and all the questions. So thank you for a great first session, you guys. And next up, Dr. Kristen Landreville will introduce the next panel.
Claudia broke it, sorry.
Hello, everyone. I'm excited to present our first, or I guess our second panel. It's on social equity and microbiome engineering of the built environment. And our first speaker here is Amber Benezra, and she's an assistant professor of STS at the School of Humanities, Arts, and Social Sciences at Stevens Institute of Technology. And Amber, would you like to come on here? And your slides should be up in just a second here. There we go.
All right, thank you. Thank you, and I wanted to thank Jennifer and Kristen and Patty for inviting me. I'm a social scientist, I'm an anthropologist. I've been working on this stuff for about 15 years, but it's really exciting to be in a room, a bunch of people who are talking about it now. So I'm really glad to be here. So I've been studying guts and human microbiomes, and the scientists who study microbiomes for about 15 years. As everyone in this room knows, human microbiota is the community of microbes in and on us. And especially in our guts, we have more microbial cells and genes than humans. This is an old figure from about maybe 10 years ago, the original estimation of how many microbial genes we had to human ones. Recent studies in the last years have shown that it's much, 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 much more than that. And so for an anthropologist, what's really interesting about thinking of humans as holobionts or microbial ecosystems is that all of those microbial populations come into being through our social interactions, right? By mode of birth, by what diets we have, who we live with and love. If we have dogs, as Rob pointed out, it greatly affects our microbial populations. Who you kiss every day, right? All of those things that are largely social, all of those intimacies affect what microbial populations we have. So there's an inextricable entangling of biological processes with social practices, right? How we live affects what microbes we have. And also that we're not freestanding units and not bounded cells, talking, as Claudia talked about, the porosity of our bodies, also is a really interesting question to anthropologists, right? What makes us human? What are humans? And how much our environments affect all of these things. So microbes circulate and confound the outside of our bodies, the outside of the world, and back again. So all of these questions that we're talking about in terms of environments, buildings, bodies, are also really interesting to anthropologists, right? So in my work, I've seen a lot of guts. I've seen guts when the in becomes the out, mice sacrificed and dissected, blood and epithelium exposed to oxygen, tiny stomachs split and scraped. I've seen human guts in pain, a confluence of urban poverty and diarrheal disease so severe that bodies can't stay nourished. And I've seen what guts produced, bacterial genomes sequenced from human feces for clues about the intestinal landscape. A postdoc at the lab I collaborated with said once to me, with human samples, feces are all you have. You have to make a story from that. So when you deal in guts, shit is inescapable. But shit also tells a story about what microbes are in the gut and what they're doing there, right? So I worked in, I know I have a very brief amount of time, so I'm gonna sort of skip ahead. I'll skip past all this stuff. Okay. So I worked collaboratively for several years with the Gordon Lab at Washington University School of Medicine. Jeff Gordon is the PI there. The scientists there were attempting to develop a translational pipeline between microbiota and malnutrition. And I conducted ethnographic fieldwork at the lab where the scientists were doing this work, and also at the lab's field site in Dhaka, Bangladesh, where mothers and children were enrolled in a microbiome and malnutrition study. So obviously for an anthropologist, these two very disparate places are really interesting. These were the two sort of ends of my research network. A very, very well-funded, American research laboratory, and essentially what the UN defines as an urban slum in one of the most populated localities in the most populated place on Earth. So I worked with scientists and researchers at the Center for Genome Sciences and Systems Biology, but also at the I lost my place. The International Center for Diarrheal Disease Research was the research laboratory in Dhaka, where microbes were both conceptually non-existent and practically a matter of life and death. So ICDDRB is an awesome free public hospital in Dhaka, but that's where the samples were collected and research took place in Dhaka. So enteric disease is a daily and epidemic problem in urban Bangladesh. In the Mirpur community of Dhaka, generations of families have been health and social science research subjects for over 50 years, and study participation has become a part of everyday life. In Dhaka, human microbial kinships are inherited and transmitted across time and environments. As lab members studied the relationships between gut microbes and malnutrition, I explored how an anthropologist could become a member of the scientific team and contribute ethnographic data while asking biological scientists to make space in research design, experimentation, and implementation for social science expertise and thinking. And I think that's what this group is doing as well, right? Not sort of tacking on an LC component at the end of a grant when you get called in as a social scientist to evaluate how the science is doing, but upstream in sort of designing the research, thinking about who you're sampling, right? What your experiments are doing from the beginning. So Dr. Gordon and I wrote a perspective piece for PNAS in 2012, which came out one day before my first daughter was born, but it was called An Anthropology of Microbes, and this has sort of been my research trajectory since then. We wrote, quote, "The development of new methods for understanding the microbial world provides an opportunity to reevaluate the way we view our human biological and cultural diversity," end quote. So we began to consider how the natural and social sciences might use the studies of the microbiome to jointly engage public health problems like malnutrition. So questions you probably have at this point: what do anthropologists do? I'm probably the only anthropologist in the room, right? Okay, fair enough. That's a good question, right? So sociocultural anthropologists study the complex cultural lives of people, their decisions, their social practices, their relationships. We use the methodology of ethnography to ask questions about people in the world. We do what we call field work. The ethno part is observing and talking to people. The graphy part is analyzing and writing about the information that we have. Our work is primarily qualitative. We don't produce clean, noise-free data. Our data is full of noise. It's basically all noise. Our sample sizes are usually tiny. For instance, I talked to 9 families for the time I was in Bangladesh. Very, very small N. And when I talk to scientists, which I often do, I'm often asked to quantify my data, and instead I use that as an opportunity to show the value of qualitative data instead of trying to quantify it, which is a hard sell sometimes. So what does anthropology have to do with microbiomes? As an anthropologist, what I love about the microbiome is that it brings together social intimacies with our biological selves in ways that show us that those two things are interconnected in a way you cannot take them apart, right? Again, how we're born, what food we eat, who we live with, who we love, and those microbes digesting our food, training our immune systems, interacting with our states of health and illness, are all tangled up together in these really important ways. So social scientists are beginning to have microbial concerns as we start to learn about how poverty, race, lack of access to healthcare, and food all shape our microbial populations. So sociocultural anthropologists do not like to make generalized conclusions about cultures, society, or groups of people. But ethnography provides insights into what life is actually like for people on the ground, giving important details that might not be visible in something like microbial genomics, right? So how can anthropologists and human microbial ecologists work together? So an example that I like to give about my research in Dhaka, Bangladesh, was that the lab had been receiving samples they had enrolled these mothers prenatally. They'd been receiving samples for years. No one from the lab had ever been there. Not only did I want to humanize the place in which the samples were coming from, but I also wanted to learn about these people's lives. So one thing I learned was, so these mothers were enrolled in this study. They got free medical care for being enrolled. That was their sort of payment. And they sampled feces from from baby's first poop, right, through 3 years of life. So they had a lot of samples. But what I found out once I started talking to the mothers, that they thought that the free healthcare they were getting was garbage. Because it was free, they thought it wasn't very good. Also, they were really, really used to taking antibiotics. They knew antibiotic brands by name because it was sort of part of their cultural practice, and there were pharmacies in which you could get antibiotics. So because the doctors in the study weren't over-prescribing antibiotics. It was a microbiome study. Clearly, they didn't want to give people antibiotics unnecessarily. The mothers felt like they were getting bad medical care because they knew antibiotics and they knew them as treatments. So they would sneak off and get antibiotics without telling the study doctors, which was obviously messing up their data. But none of the scientists knew that, right? Another example that I can give about the sort of value of ethnography was during the time I was there was a huge water boiling campaign, a public health campaign. So water in Dhaka is notoriously polluted. It is full of industrial effluent, sewage, arsenic, heavy metals. So everyone is told to boil their water before they drink it because it's just not safe. But when I was hanging out with these families every day, I noticed no one boiled their water. I was like, what's going on? I know you all got this message. And what I found out was gas service was really unreliable, and maybe 5 families would share a 2-burner gas stove. So gas service would go off at 9 a.m. for the rest of the day, and they wouldn't be able to cook anything. So they would cook all of their meals for the day in the morning, and they would mothers especially would have to make this decision. Do I spend an hour boiling water? No, that's a waste of time. I'm not going to do that. I need to cook food so my family can eat. So those disconnects between maybe the scientific information or the public health information and what people actually do on the ground is the value sort of social science and anthropology. Right? So these are just pictures from Bangladesh, obviously. So I've continued to develop this anthropology of microbes, drawing together the collaborative capacities of ethnography to actively contribute to scientific projects to try to partner with scientists. So the last question, these are just more scene pictures. This is so there are open drains in front of every house. There's no public infrastructure for garbage collection, and so that's what happens. And then people often left their water out to sit, which they thought made it cleaner, which obviously was a misconception. So the last question I have is isn't interdisciplinary collaboration hard? Yes, it's almost impossible. Even when people come to the table with the best intentions, it's very, very, very difficult, right? So we all know that each of our disciplines has a very particular language, a specific way of conducting research, and even a special way of writing papers. When Dr. Gordon and I wrote that PNAS paper, we had probably 45 versions of it before I stopped counting, because we don't even construct sentences the same way in different disciplines. And we're not taught how to go across disciplines, right? Everyone loves to talk about collaboration, everyone loves to talk about interdisciplinarity, but we're not trained in how to do that. Right? When you go to apply for an academic job, it's in a department. You can't be an interdisciplinary scholar, right? Journals aren't interdisciplinary. You have to be in a field when you apply to a grant. So structurally, our academic and research system isn't set up to really facilitate that kind of collaboration, right? And we just don't even know how to talk to each other. I will just also say very briefly, even as an anthropologist, this is a really problematic thing for me that I'm really grappling with in my research. I have lots and lots of pictures of Bangladeshi families. Part of that was really good for the scientists to see the humans on the other end of their research network, but this is the one picture I have of anyone in the lab. It's like an ad, like I don't even know who that is. I would never have asked any of the scientists to pose for pictures the same way that I asked Bangladeshis to, and that's kind of fucked up, right? The way that we create subjects out of certain bodies and not others. And I think that's really important for me to think about as a social scientist and for scientists to think about, right? Which bodies are we experimenting on, right? So I wrote a book. It came out last year. It's all about my experience in the Gordon Lab. And spoiler alert, our ultimate failure of a collaboration just because it became too difficult. But I think it's a really important thing to talk about because these collaborations are so hard, right? So when thinking about, what we think of as the microbiome, I started to realize that social and biological scientists may not even start with the same idea of what is important to a microbiome, what healthy is, right? What social determinants of health are. So for human microbial ecologists, things that are important to study, like you might think about diet, right? That's an important part of what goes into making the microbiome. You might think about geography, genes, mode of birth, and especially things like DNA, vaccinations, right? Those things all contribute to a picture of microbiome. As an anthropologist thinking about the microbiome, all of those things might be important, but also things like gender, class, and ethnicity, working in environmental conditions in a garment factory. Dhaka notoriously has a huge and very unsafe garment industry, right? The violence of a national colonial history, right? Bangladesh only became a free democratic nation in 1971. It was colonized by 3 different nations and also now has the largest number of NGOs of any nation. Those things are also important to people's biology, right? What ends up happening to them. a corrupt and non-existent governmental system of sanitation and water. So when we start to pile all these concepts on top of each other, this picture gets really messy, right? No offense to any scientist in the room, but this is not a picture you necessarily want to work with, right? It's very hard to draw conclusions or move forward with any kind of scientific research when your picture is this messy. But I guess what I would argue is that this picture is also more detailed and more complete and eventually more precise. So if we can figure out how to make the social science data interact with the scientific data, we would have better science, right? What is my time? I still have time, right? Okay, great. I really speeded through that. So I will obviously take questions about, that's the work I've been doing for 15 years. I'm going to sort of put that aside to move on to the second part. And that's what my future work is sort of heading towards. In recent years, I've been really thinking about how racial categorization works in the microbiome. I've written a couple of papers about race in the microbiome, how ideas of race keep surfacing, even though we would assume that microbes don't have race, since race is a social construct. So I've written a couple of papers with some scientific collaborators, a lot of junior scientists, which is, Jeff Gordon is an amazing human and scientist. He's also known as the father of the microbiome. I was a lowly PhD student when I started working with him. The power imbalances were great, right? I'm starting to work with junior scientists who are thinking about how to do the science differently. And I think the Premier Project is a perfect example of sort of the future of science and what things how we need to center social justice in how we even devise our scientific questions. So this idea that, race is not based in biological reality, right? We can all agree on that, right? You can't really look at someone's genes and know what their race is because race is a made-up thing. So the old concept is that genetic variation between races is large and uniform and can be used to sort and define racial identity. Newer genetic concepts are that genetic variation among humans blurs the lines between populations. So race is a social reality that structures societies and how we experience the world. So if we start with the idea that race is a social construct, great. I think we all agree on that, right? That's universally pretty much agreed between social scientists, biological scientists. But what does that actually mean? How does that actually play out in scientific work? When I wrote this paper, Chasing Ghosts, about a future anti-racist microbiome science, we did a sort of survey of all the microbiome papers written since 2009. A lot of microbiome work in the literature mentions race and racially categorizes subjects. Papers that included talk about or discussion of racism were like 2 out of thousands, right? So that's obviously a problem. By categorizing study subjects or populations by race, you're instantly implicating and entangling yourself in a complicated and discriminatory history. So my experience has been scientists are like, yes, race is a social construct. We're totally on board. Yes. Let me show you what happened to my black subjects and my white subjects and how their microbiomes are different, right? There's a disconnect there between how we think race actually works and then using race to categorize people. I know this is also compoundedly complicated by the fact that funding agencies want you to demographically identify your subjects because of histories of inequality in who got to be test subjects. So that also makes it really tricky and complicated. And the same goes for sex, right, and gender. So if race has no coherent basis in biology, it is also true that the harmful effects of historical and contemporary racism have real impacts on people's lives, bodies, and environments. So new trends we see are looking at people and saying, well, I mean, the vaginal microbiome literature is very notorious, right? One of the foundational studies says black women, Asian women, and white women have different microbes in their vaginas, right? I hate that study, and I hate that it has sort of become a building block for how we understand the vaginal microbiome, right? Because it has nothing to do with people's race, what microbes. It has much more to do with their lifestyles, their sexual activities, their diets, their environments, right? But those things are really hard to study. It's easy to just slot people into racial categories. So sort of So why does this all matter? Why am I going on about this to this group? Because if you start with racially categorized subjects, first of all, how? Are you asking people to self-identify what their race is? What do you even mean by race? Do you actually mean genetic ancestry? Do you mean what their social position is? In what place are you doing that, right? Because what we might mean as Black in America means something different in British Columbia, means something different in Venezuela, right? Means something different in Jamaica. So because those things are so socially relative, if you say this happened in Black subjects, what does that even mean, right? So you're starting with a very sort of messy, imprecise category. When you look for biological differences between racial groups, so if you start your study with categorizing people into different racial groups and then look for differences between them, that's also not the good beginning of a study, right? That's not a good way to sort people from the start. Okay. And then when you attribute health or microbial differences to different groups and explain those things through race instead of causal variables, you're not doing good science. Sorry. Sorry to tell you that. So the reason that this is important is because it's inaccurate, it's imprecise, it's discriminatory, and it's harmful. I'm just going to speed sort of through these things. Especially in the microbiome, when people use race, things that they're using it as a proxy because it's easy. But what they actually might mean, these are things that actually affect people's microbes, right? What diets they have, what their environmental exposures are. If they live in in public housing where there's lead paint, if they don't have clean drinking water, what is their access to health care? That's going to affect what microbes you have, right? Do you have access to clean water and sanitation? And also stress from systemic racism. Like, this is one of the hardest things to wrap our heads around, is that there are health disparities based on race in this country, 1,000%. It's not because people of different races are biologically different. It's because racism has an effect on your biology, right? So that makes it hard to figure out how to solve the problem without continuing to concretize these differences between people, right? So in this paper that I wrote, and continuing on my work now, really where I see changes need to happen are institutionally. So who teaches, does, and evaluates the science? I think that's a huge part of this premier project, right? Like, who are the scientists? How are we educating new and underrepresented populations of scientists? But also, who gets to be reviewers on papers? Who are editors for journals? Who's making the grants? Transdisciplinarity, that is also really important. I would like a voice like mine to be on every scientific team, right? Thinking about how to inform the science differently. And also ethics and empowerment. How do the communities we are studying, the people that we're sampling, what are they getting out of the science? Why should they care or give you samples? Why should they let you go into their houses or give you feces? What is besides contributing to the scientific good or knowledge, which I'm sorry, they don't give a shit about, it doesn't matter in their daily lives. What is the benefit sharing for the communities, right? Because we continually sort of sample the same vulnerable populations. What are they getting out of it, right? Okay. Those are some articles I've written, and these are my thanks, and that's it.
Benezra, thank you so much. I'll move this a little bit closer here. Okay. So up next, we have Andrew Hardwick, and they are a doctoral candidate at NC State School of Public and International Affairs. And their presentation title is Ghosts in the Built-in. Sorry. And so
All right. Hi, everybody. My name is Andrew Hardwick for pronouns they, them. My work relates to ghost variables I'm doing for my dissertation. Its relationship with policymaking and narratives. And this work is supported by NSF and Premier. So what I want to convey to you today throughout my presentation is basically there's an overuse of stories about providing biological reasons or lens, right, to explain variations between different social groups, like talking about race or gender, when we should be considering more social explanations like Amber was talking about in her work, right? And my research is looking across different areas where you might see stories like journal articles and then the news articles that cite those journal articles, the people that tweet about them on science Twitter, and eventually ends up in the policymakers' decisions around funding or guidelines and rules and regulations, right, about how we should manage our built environment. So first half, I'm going to talk about just ghost variables in the built environment. So we all pretty much know what the built microbiome is. We're here, right? We're talking about managing spaces like hospitals or people's houses and such. We're trying to figure out what their microbiomes look like in these spaces, how that's affecting people's health, either directly or indirectly, like their skin microbiomes or their gut microbiomes, right? And then today we want to precisely engineer them, right, like Claudia was talking about, in ways that improve people's health. But the issue I'm going to be presenting you here today, and Ember was talking about, if you start from the basis of what are called ghost variables, right, where you're talking about like someone's race to begin with, for example, you could miss the mark and actually worsen people's health outcomes or just reinforce a lot of narratives around like bad stereotypes around like race or sex and gender, right? So what's a ghost variable? Basically, it's some usually socially defined variable that you're using as a proxy for a number of other variables underneath it. And commonly, when people are talking about ghost variables, they might use these as the end-all explanations for variations between groups. So you might start at someone's race and not looking deeper, and that's your basic explanation for why you see a variation between different groups of people. So there's a lot of different examples you might find of ghost variables. I mentioned race is a pretty common one, or ethnicity, things like that, right? You might also consider sex or gender. Rob talked about, for example, you might notice using sex to determine you could look at microbiomes and use that to determine the sex of people in a room, but you should also consider how the way people live is gendered. For example, in the US, how often people clean is gendered, for example, and that might affect their built microbiomes, right, in their spaces they live in. And lastly, another area you might consider is geography. So like if someone comes from a developing or developed world, right, that's a very broad generalization of a number of different countries and places that people live in, right, when there's a lot of variation between those two groups, developed and developed world. Building up from ghost variables, there are other aspects that people talk about and consider, such as associative factors. These are associated with the structural reasons about why you might see variations between social groups, such as exposures to pollution or quality of housing, but you're not quite there at the mark yet about what's the root cause of what's going on. Then you get to structural drivers where you explain, okay, this is the end-all be-all. This is explaining really what you're seeing. These are things like variations of racism, redlining, structural racism, colonialism, things like that. Or you might consider aspects of sexism too, misogyny, like in medical practices or transphobia. And I like to use this iceberg metaphor when I'm talking about this a lot. So you might, if you're just looking at the surface, see ethnicity, and that's all you're looking for, and you think, oh, that explains everything I've seen. But as you start delve below the surface and think about this more, you notice how like variations in pollution explain people's different effects based on ethnicity, right? You might go even deeper and find that environmental racism at the end of the day is explaining the variations you're seeing among different racial groups, right? And I would say this is an ongoing problem right now for research in the built environment. For example, you can reinforce myths about socially defined groups in relationship with places. For example, Amber mentioned her work with DeWolf, where they talk about chasing ghosts in microbiome research. One of the things they mentioned, for example, was the association between indigenous groups and the idea of the places they lived in being pristine or undisturbed environments. Common association, these myths around indigenous group is like a time back machine or something to figure out what the long past was like. Well, that's not really the case. These people are continuing living their lives in the modern day, right? I've also seen other examples where people associate, for example, black neighborhoods being more like violent, chaotic versus white neighborhoods being more peaceful or calmful, right? Things like that. The other aspect is that what I'm seeing right now is you're ending up funding research that perpetuates biological lens. Amber mentions this seminal work about the vaginal microbiome, and I also noticed as well, like NIH continuing to fund research that uses this biological lens, like the hypersexuality myth around women of color to explain variations in their vaginal microbiomes, when really it's a lot of other social factors that are a lot harder to say, right? They're a lot more messy, but that provides a much better explanation of the variation you're seeing among different women. Building on from that, in my work on policy making, there are past examples of people using these biological explanations to develop policies right around like drugs and such, and that's where, if we're not careful, I think microbiome research could end up going as well. For example, when you look at race-based policy making, you might have heard this black-only heart medication called Bioethical. You can get it like the US or UK, for example. If you're a black patient, you can't get it if you're a white patient. So my spouse could get it, but I couldn't, right? And Sahin, her work Superior, which talks about the building up of race-based science in the modern day, she interviewed a number of people about this medication, and one such scientist described it as it's as effective as flipping a coin. Whether you gave the black patient the Bioethical medication or you gave them medication based on like their age, heart condition, things like that, right? So at the end of the day, you're not really helping people out, I would say. At the same time, you're reinforcing harmful myths. If I can believe this myth about my black patients versus my white patients, then what are myths might be reinforced? For example, that Black people are more pain tolerant, that Black people are more likely to have sickle cell anemia, when really it's about, did you grow up in an area that had a high prevalence of malaria, for example, right? Or that Black people have denser bones, things like this, right? If I can believe one myth, why can't I believe them all? And then when you look at sex and gender, which is an area I've been looking more into, one such example is FDA made this rule around a drug called zolpidem, where on your sex you were given a different dosage. What they found was originally women more often than men were suffering from dangerous levels of drowsiness the next day, like couldn't drive a car safely right after you took the drug. And they decided to go with the study that showed sex was explaining the variation. That's how they turned their dosage, right? But seeing your other book, Inferior, which talks about relationships between women and science and science-based policymaking, talks about how BMI provided a better explanation for their dosage, right? But that's a lot more messy and harder to understand and prescribe right as a physician instead of just using your patient's sex or gender, right? And I will say for sex and gender-based research, it does get a bit more complicated because, for example, thalidomide, which is a nausea drug you get in the 50s and 60s, it was associated with really bad birth defects for pregnant women, right? They originally, when they did their clinical trials, they only considered men at the time. They didn't consider the impacts on women or pregnant women, right? And so that's, other than the spectrum, right, a bad issue, right? But if we go to the other side, we always say there's a difference between men and women, then you can reinforce basically myths around that, when really it's more of a spectrum. Sometimes it varies, sometimes it doesn't. So we need to consider, like that iceberg metaphor, you need to consider all different aspects, right? And at the same time, I notice this a lot as a non-binary person. When we use this kind of thinking, we reinforce this idea that there are only like two sexes or genders, right? When both sex and gender are on a spectrum. And normally I use these terms sex/genders because I see people conflate the term a lot still in my research. So then moving on to my second half, I'll be talking about some of my research that I'm doing and how I'm using stories to understand these ghost variables and how we might think about like metaphors, as I heard earlier, to understand these concepts, right? How we might talk about them. So I'm using a framework called the Narrative Policy Framework. Basic idea is that we all tell stories, and that's how we communicate information. And it's also how we're processing information as well. I'm telling you a story right now about my thoughts about federal policymaking around drugs and such, right? And you're going to relate that to stories you've heard before and decide, is this a good story? Do I believe it or not, right? And the other aspect is you might look at the different elements or content narrative to see how they influence decisions, including policymaking. So there's different bits and pieces we can use to compare across different stories, right, to see how they might vary or differ. And then lastly, there are patterns in both the stories that people use. So for example, I've done some preliminary work looking at people's stories around like ghost variables or structural drivers, and notice that there are certain strategies they use again and again when I'm talking about like ghost variables, for example. And I'll show a little bit of that later in my slides. Another aspect is that based on like our beliefs that we share, like our political, moral, cultural beliefs, we might have similar responses to the same story, like if I have the same moral framework as someone else. But if we differ on our morals, we'll respond differently to the same story. The other major part of this framework is the idea of the micro, meso, macro, all interconnected and interacting with each other. So on the micro level, it's all about different individuals, how they tell stories like I'm telling you right now, and how you process the information, right? And so when we're looking at the built environment, we might ask for simple is, how do I tell a good story about these structural drivers if I want to convince people that they should care about like structural racism and such in the research? Okay, how do I tell a convincing story that matters to them, right? And then you might also think as well as what people process in their head, like what do others think about ghost variable stories? What is it about a person that makes them think like, oh, I should use this biological lens in my research when I hear it from someone else, right? And then you can look at the meso level, which is all about groups or how coalitions, collections of groups either are basically fighting for their case in some kind of policy decision space. So for example, in built microbiome research, I noticed a lot, and this might focus on like an argument for like a biological lens, a biological focus versus like a social or social biological lens to explain what's going on. And so you might ask, for example, like me, what are the lenses people are using when they're addressing the same research question and how that might affect the work they're doing, right? And then other aspect that's common in this area when you look at the meso is who's winning the fight over their stories. And really what I've noticed so far is that there's a heavy use of like these biological lenses right now. That's the main stories I'm seeing a lot. As Amber mentioned, not a lot of people talking about the effects of like racism in microbiome research, right? That's kind of the losing stories right now, right? And we want to try to change that. Then you can look at also the macro level, which is all about how different institutions or cultures influence how we think about things. So commonly, we might think about like our belief systems and how that might influence our study of the built environment. If we have certain beliefs about certain socially defined groups like races, sex, and such, how's that going to influence our take on the built environment, right? And so when you look at story, there's two different aspects to it. First, there's the elements, which are the basic pieces you stitch together to build your story. You might talk about characters. Normally, this is a person, or commonly I see microbes as well because people give them agency. Like I'll see stories about people talking about we're losing the war against microbes when it comes to like antibiotic resistance, right? And we're personifying them, right? One such example might be you describe the race of your research subjects. Like for example, in a journal article, I know someone described their research subjects as ethnic Chinese that were not the offspring of interracial relationships. That's a very strong biological lens, right? That they're reinforcing to other researchers that this is how you should be thinking about when you're studying the built environment, right, in your research subjects. Then you can talk about the plot, how things have changed over time. For example, we might talk about how pollution has changed over time. You're kind of getting closer to like the root cause about variation and such, right? About what's going on in someone's built environment. And then setting is very common, basically the place that something's happening in, either physical or metaphorical. Commonly it's physical, so you might, example, if you use structural drivers, talk about a red line neighborhood and how that relationship with like green space, for example, is impacting people's health in their built environment. And then lastly, there's a moral that's basically your argument about what should be done, right? So commonly for journal articles, where I'm starting from, you'll see an argument about what we should be researching next, right, and what I should be getting funded for, things like that, right? And so if you're using a very biological lens, ghost variables, right, you might say we should, I've seen this, study different races to understand their biological differences, just saying, I use this biological lens, let's do more of it, right? Let's keep doing that again and again. That's what we're communicating to other researchers, and that's what maybe like science Twitter is gonna pick up, or the news article is gonna pick up, right, when they publish about your article, right? But at the very least, these stories normally need some reference to a character and some connection to the policy that's going on, right? The other aspect is content. You might talk about, for example, the beliefs that you might find in a narrative. So you might use like cultural theory, Jonathan Haidt's moral framework, things like that, or people's political beliefs like left-right spectrum in the US, for example, to explain the narratives you're seeing and the way people are talking, right? And the other aspect that I've been focusing on is what are called narrative strategies. They're used normally at the coalition level to either improve your case or try to like win your story or preserve your win. All right, got it. You got like, for example, the angel devil shift. Angel, you sanctify yourself, say how great you are because you're winning. Devil shift, you talk about how terrible people are because of you're losing, and you try to convince people that like we shouldn't be doing things the way things are going, right? There's also causal mechanisms, which is very interesting, looking at ghost variables and such. For example, mechanical is all about the system, so you might talk about systematic racism. Intentional, you might talk about like red line, how that's intentional, how people did. But what I know is this, for example, with accidental, people will talk about how ghost variables associated with this kind of accidental cause is just by happen chance by the race, for example, that people are experiencing differences in like their vaginal microbiome, for example, and they're not thinking deeper about it. And when you communicate that to other people, that's how they're gonna think too, right? The other strategy I've noticed a lot is called scope of conflict, where I try to contract it or keep it the same. With the biological ones kind of being the winning story, I would say right now, that's kind of where the focus has been, about we should just talk about this biological focus and just that, versus expansion, where you talk about all the different like today's social issues, like structural drivers that we should be using, right, to explain what's going on. And to end my talk, I'm going to be telling you two different stories from journal articles using asthma. One talks about it in terms of ghost variables more, and one uses more of a focus on associative factors. So one article writes, since World War II, the prevalence of many chronic allergic and inflammatory diseases such as asthma and rhinitis has increased dramatically. So we're talking about this terrible problem, right? Asthma and such is increasing in plot over time, right? And they described it as the prevalence of asthma and rhinitis may reach a plateau in developed countries in recent years, but fast increasing trend is still observed in developing countries. And so throughout this article, they use kind of like a rural-urban divide. They kind of keep it vague and such about why people are experiencing variations in their exposure to like asthma and such, right? They're not getting as deep and more specific about what's going on. And at the end of the day, I don't think you're in it with as precise research you can use for modifying the built environment. Versus this story that uses associative factors, where they talk about asthma is the most chronic disease of childhood and is responsible for substantial morbidity and healthcare costs. So again, terrible problem, right? Asthma, we should address it. But this time they focus instead on, they say childhood asthma associated with sensitization to inhalant allergens, whereas the effect of early life exposures to these allergens or the sources of asthma risk is unclear, might differ among allergens. So in this case, they're getting much more specific. In this case, they looked at examples like people's exposure to smoke, cockroaches, pet dander, things like that, right? That were much more specific about their built environment, providing, I would argue, a much better approach to like modifying our built environment versus like a rural-urban divide or a developed rural divide, right? And so I'm gonna end here, and what I want you to encourage to think about is how can we tell maybe better stories around our built environment and how we might encourage other people to do the same as well, like Amber was giving in her talk. Thank you.
Okay, just a reminder, we'll take the Q&A after our last presenter here. Our last presenter is Davida Smyth, and Dr. Davida Smyth. She is an associate professor of molecular microbiology at Texas A&M University in San Antonio, and she'll be speaking with us about curing your teaching with microbiomes. Let's welcome, thank you.
Okay. Thank you very much for having me. I'm delighted to be here. And now for something maybe a little bit different. I'm going to talk about teaching. So I'm going to talk a little bit about three main organisations that I'm involved with. Well, two organisations and a project. The first one is the National Centre for Science and Civic Engagement, which I think Amber and Andrew will be very intrigued about. And then REMNet, which is Research Experiences in Microbiomes. But the first thing I'd like to introduce is Sensor. Sensor is a project that's been around for about 20 years. And it's a bunch of faculty, about 6,000 faculty across the country, some international faculty as well, me included, but I was in America when I joined. But we're very involved in civic engagement, trying to teach students science through civic engagement. And if you want to boop that QR code, it'll bring you to our website. But you have the slides as well. But Sensor has basically been built around four main ideals, right? And these will be probably a little bit different for you all to engage with. But what we try to do is have our students engage with science through teaching of issues of civic importance. These are capacious, wicked problems that don't have an easy solution, right? They're usually public. We also try to encourage our students to take responsibility for their own learning, right? So this is kind of, if you tell students, here's the problem, why don't you go and help us fix it? That will help them kind of engage with the issue more. Another thing that we try to do is, it's very important actually, is we talk a lot about the power and limits of science. Science is not all-knowing, it is very problematic. And so we try to teach our students this as well. And last thing is we try to make our students bring them on a journey from local to global, right? So we start with a problem that they can relate to, and we bring them up to the grand global challenges of our time. And so what I want you to do is I want you to think about civic engagement as the why of learning, right? STEM education can be civic education, and we often separate them in the US. But to get our students to engage with what they're learning about, you have to really get them involved with an issue. And it may not be what you care about necessarily. I polled the first years at my institution to see what kind of issues they care about, and they care about homelessness. I care about antibiotic resistance, they care about homelessness. We're not on the same page, right? So often if you ask the students what they care about, they'll tell you. But I moved to Texas 3 years ago, and if any of you know anything about Texas, we like our water there, we like our swimming holes and our rivers and everything else. The one thing we don't do very well is protect them, okay? So when you think about the issues in Texas, there are many, 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 many issues. I love giardia myself, where people go swimming and they get giardia. Drought, wildfires, flood, sewage. I mean, there's countless issues in Texas that you can deal with. But if you're talented, you can turn this into biology. See what I did there? It's magic. You can take all of these issues and you can convert it into a conceptual modality for your class, right? So it's really, really critical. So the second thing I want to talk about is what Andrew and Amber were alluding to, is who gets to do research in this country, right? Now, I work at a HSI, minority serving institution, right? There's about 8 faculty, and I'm going to use terms that we just said we wouldn't use, but we're Hispanic serving, but 78% of its students are Hispanic identifying. But I want you to know that very few students get to do research, okay? Very, very few. And this is national data, and I can tell you this is not typical. At my institution, about 3% of the graduates will have had a research experience, right? So how can we make this, how can we change the needle here? How can we move the needle? How can we get our students to get this undergrad research? Because it's really important. Students need to know how to do science, to be scientists, right? And the nice thing about a CURE course, which is a classroom undergrad research experience, is that every student who takes that course gets a research experience. Not just the ones who talk to the faculty member, not just the ones who know the system, right? The first gen students, the students who are from low socioeconomic groups, they can engage in these science processes as well. And look what's important here, iteration, making mistakes, repeating things, right? Dealing with things not working. That's what science is, right? And when they do that, they learn all these lovely outcomes, right? You're talking about, they get mentoring that is built into the course. It's not something they have to look for, okay? They can mentor each other. They get to do problem solving. They increase their critical thinking. So cures are a great way to get your students involved in science. So I want to just poll you all here, and I know you're not all faculty, but how many of you have ever done a cure with your students? 2, 3. I'll leave that out. I'll leave that hanging. All right, so I'm going to introduce research experiences for microbiomes. We are a RCN UB funded by the NSF, and our mission is microbiomes for all. We want to get all students to engage with the microbiome, not just those of us who have the resources, have the money, have the time, have the expertise, right? And I'd love you all to sign up. I'll be sending around a QR code later, and I'll make you all join my network, because you have to. That's not a choice now. But cures and microbiomes. The built environment is perfect, because if you are at an inner city school, if you are at a resource limited institution, you cannot go outside. It's expensive. So you can do the inside of your school. So we do a lot of elevator button stuff, right? We do a lot of water fountain stuff. But we also go out into the city, and we've had students like swab the streets, right? We do all sorts of things, swab statues. It's great fun. And depending on your resources, this is me at the new school where I used to work in New York. I don't know if you can see it there, but I have my students make the libraries. So they use the magnets to do the whole thing, right? So from start to finish. Depends where you're at. And here's Bernadette. Bernadette, this is a woman who's not at a fancy school. She's not at an R1, right? And she is publishing with her undergrads looking at sediment, right? This is one of her members. And here is Sean Coleman. He is working with pigs. So basically looking at pig microbiomes. And again, through the network, has learned how to do the analysis, is doing it with his students, and is publishing and presenting at conferences. So it's really important for us. So what can your students do? If you're creative, you can come up with some really nice learning outcomes, right, which will engage your students around issues. And here's a couple of mine. And if you look at the bottom, I love to do like antimicrobials in the built environment. So I study antimicrobials in building products, things like paint, right? You can think about air pollution in terms of microbial abundance, right, if you can sample the air. And then thinking about gentrification, things like this. So look at this. Here we have what these courses actually involve. So we are looking at water samples from local rivers. And I love looking at Superfund sites. You can think about soil for antimicrobial activity, and then also looking at gentrification of neighbourhoods, right? So as neighbourhoods are gentrified, the soil is being impacted, but where is this happening and what communities are being impacted by this, right? And this is where we combine the two, sensor and RemNet at the same time. So here's our microbiome pipeline. This will look very familiar to all of you, right? So we basically take the students on the journey from microbial communities, whatever it is, whatever we can do. We can't do human samples in HSIs and minority serving institutions, so we do these environmental samples. And we basically bring them up to CHIME. We get them at least to analyse their data with CHIME. And we teach the faculty how to do this, right? So these are faculty who do not have training like y'all. We try and get our students to do this whole pipeline from start to finish. And it's important to consider your budget. So for those of us who are at R1s, R2s, you have big budgets, right? Schools like my own, I'd be lucky if I have $1,000, right? Most schools, we can maybe send out 6 samples for sequencing, right? For an entire course, right? So if you have limited funds, what can you do? You can do other stuff, right? And you might be able to collaborate with the lab if you're lucky. So this is what I hear. We can't do this. I just can't. I don't have the money for this. I'd love to, but I can't. So what can you do? It starts simple. Readings, right? Get your students to do collaborative readings, and they don't have to be papers. They can be other types of readings. So be creative with your readings. The students want to be engaged. They don't really deal with the primary literature that much anymore. So you can start thinking about papers and stories that relate to an article and describe the article. Have your students do that. And this is one of your own. This is Carlos Scholar. I was hoping he'd be here today because I put his face on the slide just in case. But he does case studies with high throughput data, right? These case studies, if you don't know what a case study is, it's a narrative that has some questions that go along with it. Really useful for teaching your students to engage with issues. And then Remnant, we are crowdsourcing everything. So we put together a journal club and we use Hypothesis. If any of you have never heard of Hypothesis, it's free. It's a group annotation tool. You can have your students annotate papers, and we're trying to get the faculty in Remnant to do this as well so we can share resources. We all know how hard it is to teach PCA and everything else to students. We have tutorials, we have webinars, and lastly, we have guides on everything as well, freely available for anyone who wants to join. We also do YouTube webinars. We do this with the ASM, with the American Society. We offer these every year, and we train faculty over about 4 sessions on how to do microbiome analysis, right? From everything from heat maps to using Excel to dealing with R if you want to. This, again, is open to faculty, and we've recruited a bunch of faculty this way, and it's a great way to get your faculty to feel empowered. I can do this with my students. And the last thing I just want to mention is that the ASM has just recently updated their curriculum guidelines. Now, if any of you use the guidelines, you'll know that they were written about 10 years ago. They're a little bit old. And so the new guidelines have things like this. I'm trying to read now sideways. But microbes are ubiquitous, are found in diverse and dynamic ecosystems. These are kind of concepts that you can start thinking about in terms of microbiomes. We can talk about the growth of their survival, and their growth depends on where they are found. Cultivation independent techniques are needed to study microbiomes. We need to start thinking about them in diverse populations. All of this is very much so tied to the microbiome, and these are our new guidelines, which will hopefully serve us for the next 10 years. But if you're wondering what a course looks like, this is what it looks like, right? So very basic stuff. You start with your experimental design, you come up with some sort of a question. I have my students write questions, go out and make observations and write questions. Then they write better questions. And then we go into the lab and we do the whole thing. Now, parts in grey and everything else, these are all optional. You may not be able to do this stuff, right? You may have to go and get another data set from somebody else, but that's okay. And then at the end, you have your students do presentations and everything else on their data. And if you're exploring data, you can explore data that you've generated, or you can explore data that someone else has generated, right? You don't have to do your own stuff. You know what you can do too? You can use an old kit. They can be analysing water. They don't have to know, right? You can have the data made for them, okay? But this is important. Nefeli is from the NIH, right? It's a great pipeline that you can walk your students through. It's all plug and play. You just enter the data. They have tutorials on there and with data that's already been made for you. And you can have your faculty use this and your students use this. And then, as I mentioned before, we have these lovely webinars, all freely available. We charge you the first time you do it, and then everything is free after that, okay? But another thing I'd like to just ask you all for help with is we gather resources all the time. So we're working with very low funds here. So we pull services that offer sequencing for faculty, right? So unless you're at an R1, you don't have a core. We don't have cores. $60 a sample is an astronomical amount of money to us. We just don't have it. So we're always trying to find resources that we can use. And obviously then for equipment as well. And if you are a faculty member at a community college, at a minority serving institution, talk to your reps. Talk to the reps, they'll often cut you a deal. There are education discounts for a lot of these things, so you can get everything cheaper. Pipettes, but remember, Eppendorf pipettes, very expensive, very, very expensive. Okay, and you don't have to print the posters. So it's another thing. So I remember the day where I had to pay $90 for a student's poster. You don't have to anymore because you can do things like this. So if any of you have ever heard of Padlet, this is a free software. You can have your students do digital poster sessions, right? And if you look up there, you can see the stars. They rank each other. They do peer review. They make comments on each other's posters. It doesn't cost anything, right? And the nice thing about it is you have these forever. They live on your Padlet account. So this is it. Consider engagement the why of learning. Why are our students in the classroom? If you ask your students, well, maybe not your students, but if I ask my students, oftentimes they'll be like, it's on my course plan, I have to be here. And that's a terrible thing to hear a student say, right? But this will help you. If you put the why of learning as being, you're going to solve a problem that's local, that's important to you, we're going to do it with Sensor and with RemNet. Hopefully the students will engage more. And just to let you know, we are not alone. We have a huge network now, we're going international as well. We're always adding new members, and we welcome you all to join us. I'm trying to get into Ireland as fast as I can, so I can go there and visit. And that's about it. So I want to thank all my network colleagues. I have a bunch of folks that I owe everything to. I used to work with the New York City Department of Environmental Protection. I did a lot of work with wastewater surveillance and other things. And this is all my funders and everybody else. And thank you very much for listening.
Great. Great, thank you so much. Should we have them come up here to the front? So if you guys, the presenters from the first panel, could come and sit at the front, that would be wonderful, and pass the mic when needed. Thank you. And we have a question. Okay. Okay. Yeah, and I will be taking the mic and moving that around. Barry, I will grab it.
Okay.
Okay, you can down, you can stay up there.
You can stay up
Oh, okay, sure, sure. So Patty has a question from online.
Sorry, I have to read it off the screen. So our question is from Carlos Scholar. He says, Carlos Scholar is here, but online. How can we tie into the ASM guidelines and do sensorized courses? And what is one course that you have in future years to achieve both ASM guidelines and sensor goals?
Oh, well, so I'll give you an example of a course that I've developed, and I'm going to be teaching this in fall. It's called How the Toilet Changed the World. And it's a first-year seminar class, and I'll be having the students learn about sanitation and microbes and everything else. Maybe not microbiome work per se, but they'll learn about the microbiome, right? And one of the things I try to do in that class is to bring in this cultural competency, the cultural sensitivity, because a lot of students have this perception that the westernized way of doing everything is better, and it frankly isn't, and we know this from a lot of data. I bring my students on a tour of the world. We go on field trips. We look at public toilet design. We look at access to toilets in the United States. And because I'm in Texas, we'll be looking at the borderlands, in particular the colonias, regions along the border where folks do not have access to adequate sanitation, and trying to improve that, move that needle along, and trying to work with the communities themselves. So the wastewater work I do, if you are watching what's been going on with COVID surveillance, it's very septic, it's very sewer system oriented. It doesn't take into account latrines, open drains, folks that do not have access, the places where wastewater surveillance would actually benefit the most, frankly. But in addition to that, taking into account the ethical ramifications. The word surveillance in and of itself is terrifying for people, and it can be used for nefarious purposes as well. So how did toilet change the world? I'll fill you in fall. Thanks, Carlos. Nice to hear from you.
So I want to thank the panel for a illuminating discussion. This is more a comment than a question, but I'd like to get your response to this. And one of the things that I have always rejected in my career is the false separation of social factors from biological investigation. And anybody who's a biologist really should understand why that is such a false separation. There are only two groups of animals that have social behavior. There are over 30 phyla of animals, and only two of them have social behavior, the social insects and the chordates. And so clearly, social behavior was important in the evolution of our species. And therefore, okay, if we're going to study any biological problem, we should be also, I mean, in humans, we also should be studying the social structures in which those humans live.
Would you like to respond?
I mean, obviously that's an excellent point, and I think we need to think about why that division still exists, right? What power structures benefit from that continuing to be separated, right? And the biological sciences is very invested in objectivity, which somehow doesn't include the social, right? And that's kind of where the problem lies, as if those things can't also be done together. And in my work, honestly, asking scientists to be experts on social, cultural, ethical issues is not exactly fair, because you all aren't necessarily trained in those things. So that's why we have to work together across disciplines, because there are people who have done PhDs in thinking about these problems, right? But there has to be a valuing of those expertises as equal, which has not been my experience. Mostly I'm trying to convince scientists why they should care about this, right? Because you get funded in science and you don't necessarily have to. So trying to really make the point that it makes the science better seems to be my in that question.
If I may, I'll just add one thing on to Amber, what Amber just said. My students told me that their Mexican grandmothers are sending them antibiotics in the mail, and I never would have known if I hadn't asked them that.
So we have another question from online from Rachel Noble. And she says, I would love to tie our rural WW surveillance work into her network. Could someone there give her my email address? And so, sorry, I didn't read. I just read the first part of it when online. But yes, so Rachel wants to connect with you, and we will get you guys together. Great, thanks.
This is a comment for Andrew. I know that you are working on your dissertation, and so I just wanted to provide a bit of feedback that may be illuminating for you. The narrative policy framework that you are using, as you are on this quest to address systematic discrimination and racism, be aware as well that this narrative policy framework comes from a very Western perspective, and the types of elements of character, plot, narrative, content are all Western themes of storytelling. And that may make sense since you are basically developing this within a Western context to look at how Western policies are enacted. But do know that there are other types of narrative frameworks and theories that come out of Eastern traditions and others that do not have the same kind of flow or function or elements of narrativity. And so that may be something that you may want to add as another distinct element that will at least allow you to interrogate some of the assumptions that you're bringing to your own analysis as you go forward.
I would say I appreciate the feedback. It is a bit of a challenge to try to challenge Western ideas of thinking, right, in some of my research, while also, as a grad student, I'm limited, right, in what I'm taught and such and what I use, and that's the line of thinking that I'm, being taught in my degree program and such, right? Thank you.
Kieran O'Doherty from University of Guelph. Thanks to the panel. It's really great to kind of have finally these kind of forums where we have all of these disciplines come together. I've got two questions, one for Amber, one for Andrew, but they're connected, so I'll put them together. For Amber, I completely share your concerns about how race is used in not just microbiology, but in science in general. But I wonder if a strong kind of statement about you shouldn't use these categories at all is necessarily the way to go. And I'll just give a couple of thoughts here. The first is that, so for example, the vaginal microbiome story, I know exactly those papers and I've torn my hair out about them. But my real beef is with how causality is implied. So for example, women of this race are more likely to douche, right? You see that. But if you change the more likely to just simply more prevalent, it actually gives a very different kind of story. Because then you can start, to my mind at least, you can then actually open up okay, well, why is that going on? Because after all, there are connections between racialization and culture, and there are clusterings around that. And so this also connects to the whole social determinants of health literature. If we don't actually have research dividing, people identified as black have worse health outcomes. Indigenous people have worse health outcomes. If we don't have those identifiers, we need to throw them. So I'm just wondering about do we really want to have a strong you're not allowed to use those biological variables, you have to be all pure, or can we use this in a more mindful way? And then to Andrew, I was thinking about the issue of sex and gender, how clearly a lot of literature is very bad. And just something I've been thinking about is, in my home discipline is psychology, even though I'm very far distant from it now. But in psychological research, it seems to me that one of the problems is that when people report results from findings, they define their variables or whatever, and then the results are never ever categorical. The data is never ever categorical, but it's reported as if, so we find, women and men respond differently to whatever. And then there's some, statistical significance, but of course these are just averages, but it gets reported as, men respond like this when women respond like this. So I wonder if an underappreciated part of the problem in all of this is that there's of course all the other problems that you've mentioned, but one underappreciated problem is that thinking and reporting and talking in terms of absolutes when actually we're always just talking about averages.
Amber, do you want to go first?
So My answer is yes, we need to completely get rid of that categorization of race. It's not accurate scientifically, it's not precise. And the holding on to it is because if you start with categorizing people by race, you're already in a mess. You've already fucked it up, honestly. So you have to start from a different place. If you want to talk about cultural relatedness to douching or environment, then to talk about, it's just the same as sex. If you want to talk about people who menstruate or people with uteruses, you don't have to say women and men. Those, again, are social categorizations. A paper came out by NASEM last year about why we shouldn't use race as population descriptors in genetics and genomics anymore. It's a 200-page comprehensive paper that says if you want to know this is what you really should look at, instead of relying on race as a proxy because it's lazy, it's bad science, and it's racist. So believe me, with my book, I've given many talks to microbiology departments, and everyone is like, but what about my decades of data? Am I supposed to just throw it away? And my answer, it's easy for me to say, but yes, the answer is yes. Because your data isn't good data. So if you want to, what do you actually want to find out about these populations? Don't start with dividing people because it's easy into racial categorizations, right? Look at what you actually want to find out about these populations instead of starting from those places. Those categories aren't real biological categories. So when you slot people into them, I mean, what does it even mean to say someone is black again? Like, what, are you just asking them to self-identify? Because that's not scientifically accurate.
What if I want to find out if black people in the United States have worse health outcomes on average than the rest of the population?
You should ask yourself if people who are subjected to systemic racism have worse health outcomes, if people who don't have access to health care, if people who live in poor housing. Those are the more nuanced questions. If those people have worse outcomes. Yeah, and systemic racism is one of those things that you can apply, one of those factors you can apply. But to say, do black people have, it's just not a good question. Sorry.
All right, yep. And kind of echoing Amber here. On the sex gender side, I guess I think about like modeling, for example, and such, you might want to use the course averages there, right? And that's really easy, I think, in a scientific paper to communicate to other people. I can go to your results section, quickly scan and look for like that asterisk that says this was a significant result, right? I think like Amber was talking about her work, we need to be talking more about the messy aspect too, encouraging people to read about that, right? That there was some variation among our male and female participants, and also being clear about, did we assign them those categories like Amber's talking about, or are we self-identifying, are we reporting that to people, right? And offering more options for people to provide variation, for example, for people to list as like male trans, for example, or female trans, or like non-binary, right? Because I think you'll end up with a more accurate data set at the end of the day, right, to work with for either your modeling. And really, it's that qualitative side, that messy side, I think that we're missing out on, that people need to be doing more. Like, I'm trying to mix both of my research, and it's really hard, because it's very messy trying to look at individual stories and such and draw connections between them versus like I give them a quantitative like code or something, right? And I use that for my modeling, right? And basically, I would encourage people to do a mix of both, and I think that will lead to like a more nuanced story whenever you're talking about like aspects of like structural racism and such, or variations between genders or sexes, things like that, right?
Thanks. I have a question for Andrew and a question for Davida, and they're connected, so I hope that this makes sense. Andrew, the question I had for you is what happens when we treat DNA sequencing as a ghost variable? Insofar as we don't really care about a microbe's DNA sequence, we care about what it does in its environment. Davida, the question I have for you comes from research done by two fantastic undergraduate researchers who I asked to look at assumptions embedded in the various kinds of computational pipelines that are applied to binning sequence analyses. And I'm so glad to see that facial expression, because the thing I wanted to ask you is how you think about teaching students to think critically about how the choices they make in applying something like QIIME matter to what they're doing, matter to their results, rather than just turning the crank. Because what these two undergrads found was that we couldn't address our research question by looking at the published literature because the communication around why microbiome analysis choices matter is just so poorly, it's not transparent, it's so poorly communicated.
Go first. All right, I guess I'll go first. So if I'm thinking about like ghost variables and discussion of like DNA sequencing, which comes to mind is really like Ancestry.com and such, its use of like DNA sequencing to assign you to a specific racial category, and that strong claim about like you're the specific race because you have these certain specific markers, right? When it's a bit more complex than that is what I think you'll end up happening again and again when you're doing that. With sex and gender, again, you might start with like a binary focus, right? When there's a number of different intersex instances that can occur, or I've seen it, for example, kind of tangential, the idea of like people's using brain scans and such to assign like a gender to that, like that there's a male or female brain, right?
I actually meant DNA sequencing of microbes.
Oh, so, microbes, sorry. Normally I'd think about it in terms of humans.
I don't care about humans.
Fair enough. Okay, if I'm thinking about microbes specifically and we're using ghost variables, I think what you might see is a bit of over-personification is what comes to mind then. Is like, I mentioned like the war against antibiotic-resistant bacteria, for example, and such, when it's much more issue of like thinking about how we're creating an environment that encourages certain microbes to survive or not. Like Rob talked about, why is it suddenly these chickens that weren't getting antibiotics, right? Having antibiotic-resistant microbes in that area, right? If you're using like a ghost variables focus, which is more of a like a, I don't know, a better word to use than lack of nuances, for example, and such, you're not really gonna have a good way to like explain what's going on there, right? I hope I answered your question. I normally think about people in the relations to microbes.
Of course I care
Yeah.
about humans, but this one,
I'm gonna add something to a previous question. Just as an example to Kieran's question, I'm white, right? But I'm not a Caucasian, white, standard, typical American. I'm an immigrant, I'm Irish, I come from a completely different context, cultural context, right? So I use the same analogy that I was just asked about there about in terms of binning. When we bin microbes, we put them into categories, right? And those categories are human constructed categories or computationally constructed categories. And those categories are dependent on what's happening at that time, at that moment in time. So when I do Nepheli as an example with the students, they can choose which database data, whatever database they're going to use, and they will typically get different answers, right? The COVID pandemic showed us the exact same thing when we were trying to assign variants of concern and and looking at SNPs and polymorphisms and which variants over the course of the pandemic, depending on the day that you analyze your data, that changed, right? So I think the idea of how we categorize microbes and how we bin microbes and whether we're calling them OTUs or whatever, all of that it ties in very nicely to that thing I just mentioned there. Remember those ideals? The power and limits of science, right? It's incredibly limited in terms of what we know in the moment, right? And our knowledge of everything changes over time. And I think that is the benefit of teaching students the importance of writing everything down, right? Metadata. The metadata is where it starts, right? If you can get as much information by interrogating the story of what you're studying, the data will be better. So if you want to start with the limits of bacterial genome sequencing, how you sequenced it, the kit that you used, the pipette that you used, the filter tips that you used, everything impacts the genome sequence quality, right? And that's before you even get to the analysis, right? So I think the onus is on us to do a better job of recording everything that we do in as much as we can so that the data sets are better. Soil is not just soil, right? And that's really important. And I actually learned this I went to a soil, I've been studying microbiomes for years, right? I went to a soil conference in Ireland and I learned about all the different subcategories of soil when you sift it. And I was like, so you see what I'm saying? So we're limited, but it all boils down to talking to each other, working across the disciplines, trying to get as much out of the story that you're trying to tell and taking into account that we are not aware of all the limitations of what we're doing in that moment. And it'll probably be different tomorrow.
Yeah, I think David.
I'd like to get back to this notion of race and ethnicity as ghost variables. I've done research on this in my work, looking at things like who's most likely to get convicted of research misconduct and looking at that as variable. And what we found was that the variable, I guess a statistician described it as heterogeneous. There was something going on there, but our variable was not precise enough. So what we really needed, like we were using things like going on names and things like that, and Asian, and so what we really needed was we needed more precise information that was underneath what we were seeing, like things like culture and ancestry, because we knew just from the stories that there was something going on there, and this sort of crude variable was telling us something, but we needed to dig deeper into that variable to find what was going on. So I'm not sure I would call it a ghost variable. I'd call it maybe a crude variable or something like that. But there was something real going on there. The other variable we looked at was gender. And there's definitely something very real going on there in the research misconduct field. Like something like 70 to 80% of people that are found guilty of misconduct are male. And you know, we have very good data on this, and there's got to be something going on there. We don't know what's going on exactly. We need to figure out, we need to find out more, we need to probe that data more, what's going on. But it's definitely real, it's very robust. So I wondered if you'd comment on that.
I would, I'm going to get myself in trouble here, but in terms of the size of the lab, so the folks who are being accused, and again, I'm just pulling this out of my head right now, but the folks who are being accused of research misconduct are oftentimes in very large labs with very huge lab groups, and perhaps are not as into the data as possible as they could possibly be. And I would like to look at the distribution of frequency of folks at those types of institutions and what proportion of them are women. Right, so just think about that. The number of women who get to these kind of huge monster labs and are running these huge monster labs, it's not very many, not very many.
Can I ask why you're looking at gender when you're looking at research misconduct? Like, what is the outcome you hope to find? That men are worse than women, or what? I mean, just like, you know, like when you start by looking at genders.
Okay, so we did this.
Could you hold on 1 second while we pass the mic?
Yeah.
Thank you.
We wanted to find out whether the Office of Research Integrity is being discriminatory in its application of punishments, because we know there's discriminatory punishment, you know, in the criminal justice system, right? So we needed to have those variables in the analysis to find out whether that was going on. Fortunately, we found out that it looks like they're not being discriminatory. They are actually, it's mostly things that you would think matter, like how bad the misconduct is in terms of number of retractions or, you know, other things that happen. But we did find some interesting things, and it told us we had to really dig deeper underneath the variable to figure out what was going on.
I mean, like Davida saying, it might be more interesting to see what institutions, how many women are in, did you look at population, like how many women are in that field or in, you know, STEM overall, right, or in these labs? Who's funding, which funding organizations,
No,
right? Like
it was a small data set,
right.
like
So yes.
350 cases.
Yes.
So what it is, so
Yeah, I'm just piling on those variables.
yeah, I mean,
Like in my
what it
brain.
is, it's you know, it's food for for additional research,
Yeah.
right?
Institutional prestige, bias, all those sorts of things.
Just one last thing I want to add is something you're noticing is, I guess the question I have is for the data set, was it something that was given to you, or were you able to compile this data yourself?
Some of it was publicly, a lot of it was publicly available. The ORI publishes their cases.
Okay.
They also, in the cases, they use pronouns most of the time. So we were pretty sure about who the women were versus the men. The racial ethnic data was not given, and it was because we had to use artificial intelligence to infer it from names, which is a very, very, very crude way of doing it. But it still was, you know, we were able to get something from that.
Okay. What I just want to pick up from that is that something I noticed when I talk with people Premier about ghost variables and such is people ask me, well, I use like secondary data sets like you're talking about a lot, right? And all I'm given is like race, so what do I do instead? And so I encourage, like, if this is your limited data set, thinking about, like, talking about your limitations, right, that you have within such, for example, right? That's just what I want to add.
Yeah, we talked about the limitations a lot in the article, and I talked about it we had a discussion with the students. I did this with students that, you know, and we had a discussion very early on. I said, look, should we even look at the race ethnicity variable? and we had a lot of discussions about, race being a social construct and everything that's been, and they're like, well, we can't not look at it. I think most of the students agreed we can't not look at it. We have to look at it and see what might be going on.
Look at it, but think about what it means, right?
Yeah.
Because also, I mean, a thing to think about is that people from middle to low income countries and women publish less and get cited less. So if you're looking at, research misconduct, who has the most papers out there to begin with, right?
Okay, I mean,
Variables too, like,
Could we have
like
Dr.
student,
Graves speak?
postdoc, senior investigator, those kind of variables. But.
I mean, using socially defined race to address a social question is entirely fine. I mean, the reason that the category was created to begin with was to differentiate people within social hierarchies. And so, and again, the PNAS report, population descriptors in genomics and genetics research, makes that point really clearly. So when I use the word race, you will never hear me say race, because people always conflate social and biological aspects of the definition. So when I'm referring to a social question, I will say socially defined race. If I am referring to the biological concept of race, which has a very different intellectual history from the social concept of race, I will say the biological race concept. And in terms of, there's no genomic basis to committing misconduct. So that's not ever going to be part of that kind of study. It's really going to be where you come into the position of social power in the discipline in question. And that, I think, using race there as a social definition works fine. And by the way, in my closing remarks today, I am going to talk about that distinction, because while the panel did a great job discussing the question, I don't think they did a complete job. So I am going to, for biological science researchers, I am going to talk about the difference between those categories, their history, and how they operate in biological research.
Do you want to do one more question, or because we're at time?
Is this, okay, good. So I'll take this last question from Glenda Kelly, who is the Director of Evaluation and Assessment with Premier. And she says, what about using cluster analysis to cluster individuals by factors that predict microbiome health, like access to food, diversity of plants consumed, exercise, etc.? Are there references using this type of approach in microbiome health research?
I mean, that's great. Yes, do that. I mean, in the NASEM paper, they say using environment is really the big step, right? Like going to environment instead of race as a population descriptor is one of the biggest steps. And I think, and I just have to say that I'm working on a paper with Ariangela Kosick, Nicole Farmer, Catherine Maki, and Sue Ishak, interpreting the NASEM paper for microbiome science. So how should microbiome scientists also not use race as a population descriptor?
The only thing I'll add to that is if you're using environment, don't assume it first, because that's what I was noticing when looking at build microbiome research is people assume what places are like first. You should try as much as possible to come with an open mind, right? Tabula rasa, and fill it in based on your actual data.
All right, well, thank you so much. Let's thank them again for a wonderful panel. We'll take a break now before our next session and come back at 11:20, right? Oh, am I right about the break? I don't have the agenda in front of me, I apologize.
Yeah, 11:20 to 11:40 break.
So we come back at 11:40.
Oh, so did I end too early then? We had to talk. Okay, I don't have the agenda in front of me, I'm so sorry.
Good afternoon. Yeah.
Don't forget your password.
And there is a very different jargon and everything, you know, it can be very overwhelming, and so I didn't know if you had like. Yes, absolutely, so I
Next. Okay. Okay. I don't have you in my I'm moderating the back.
to the east. Feet to the east. To the west. Yep.
But then I also have like the funny ones. So I have multiple students over the years who knew that like the first and last name issues, which is my favorite. And then they knew that Christopher was supposed to be shortened somehow. So I would get emails that would say, "Dear Professor Christ." I will tell students to let me know what they want to call you. I have one group of students like international students from rural China. And they would come in, like the student would go, okay. And then I had another student who I said, I'm actually not going to call her that. Because she said, Professor, I see that you really love food. Please call me.
We'll get started again in just a couple minutes.
All good, Kurt? All right, y'all, let's get started. First off, we have Kieran Quinn. He's an assistant professor here at NC State University in the Department of Biological Sciences. He'll be presenting his talk today about the ethical considerations of microbiome research and historical African-American burial sites. Take it away, Kieran.
All right, thank you for that introduction, and good morning. So I'm going to present this in the scope of case studies, because really to go through the research, we have to tell a story, but I only have 15 minutes. So we'll start with my doctoral research with the New York African Burial Ground. For those of you who are not familiar, it is the oldest and largest burial site of free and enslaved Africans ever discovered in this country. Here we kind of have the background of the origin of the site. It was established by individuals who developed the Negro frontier, this free, burgeoning community right outside of the New Amsterdam colony or present-day New York. What you see in, let me see if I can get my pointer. What you see in this dark circle is the part of the burial ground that is still located in New York that has roughly 15,000 individuals beneath the streets. Everything in the white rectangle is everything that was discovered, excavated, and that research has been done on. The only other thing that I want to point out about this slide is this odd shape here. Underneath, the title says freshwater pond, or also known as the collect pond. That'll become important later on in the talk. For those of you who are familiar or just curious, this is right in Lower Manhattan. It's funny to think about one of the most famous cities in the world having a population that large that was just forgotten. Right? It is steps away from City Hall and the World Trade Center monument. So in the early 90s, if you can focus on that picture on the left, they went to erect this federal building. As they broke ground, all of these human remains started to become unearthed. They found that this was the New York African Burial Ground by going back to the historical maps. So at this time, Howard University steps in under the direction of Michael Blakey, which we can now define by the size of the project and the 419 individuals that they excavated as one of the most sophisticated archaeological projects ever conducted. They started this research from a different perspective than traditionally done, from a clientage perspective, developing this clientage model, reaching out to the descendants or the local African American community to figure out what their interests were and how we could fit the science to their interests instead of the other way around. They developed all of those reports on the right for the skeletal biology, the history, the archaeology, osteology, you name it, for the limitations of the time. And in the lower left-hand corner, you can see where they reburied the remains out of respect. So I come along to Howard University as a graduate student 24 years later. All of the human remains are reburied. As a biologist, wanting to learn more about this population that could be my not-so-distant ancestors, since I'm from New York, but all I have is the burial soil. But the thing that I do have to my advantage is this science. We now have next-generation sequencing, which is something that they didn't have in the 90s. So my first question was, can we get human DNA from these burial soil samples? But of course, at this time, first starting out, this had never been done. Some of these Neanderthal projects that we see today, cave sediments and such, had never been executed. They weren't even thought of at the time. And so we thought of a roundabout way to figure out answers to those same questions, but using the things that exist in soil, like microbes, right? So the question is, would we see any human-associated bacterial signatures in our New York African burial ground samples? Or would it just look like what reviewers said, dirt? And so ultimately, we found a mix of both, right? So maybe this is a way to, not just when you don't have access to human skeletal remains, but when you want to leave human remains at rest, but still use a historical resource to learn more about that population, we can use a pipeline such as this. Some of the things that we wanted to expand upon were the smoking gun, right? How do we know we're really looking at these historical signatures specific to the human who was buried there? So we try to figure out, okay, here's a microbe that is only existing in the scientific literature as defined by the skin microbiome or the gut microbiome, or even disease pathogen like cholera, okay? If anybody is familiar with the Akkermansia signature up there, it is highly, it is a topic of interest now, particularly in biopharmaceuticals for probiotics and such. And so now that in garnering this information, this also leads us to a conversation, about commodification, right? Now that we're learning what these things are, and we've had this conversation about Bioethical, how do we use them? So in our environmental signatures, we didn't only see things that exist outside of the human body and in the soil, but specifically wetland soil species. So just as we believe we're seeing the human that existed 400 years ago, we believe we're also seeing the evidence of that collect pond that existed 400 years ago. So I don't know how many of you have been to New York recently, but it's a concrete jungle. So what we're doing now is we're taking this a step further, right? We have this proof of concept using our microbiome analysis to establish human evidence, even without human remains. So can we expand upon this? Can we build a broader picture, and how do we do that? Can we determine something about the ancestral homeland of these individuals, right? Some population-specific information. We have a student now working on any animal-associated bacterial species because it's soil. So we're gonna have all of these additional things like crops, right? What we're finding is a lot of leafy root crops, rices, so now we're learning about their diets. Were they rich? Were they protein rich? Were they highly vegetative? But also because we're looking at bacterial sequences, we can also reconstruct outbreaks at the time. What were these individuals dealing with? What were they suffering from? And how can we compare those things to what we are experiencing today? So what we're doing in the Ashes Lab is we're doing this research, again, building on that foundation of the clientage model, but from a different perspective. I know the conversation usually, as it goes, even when I go to these conferences, is about community engaged research, but we really need to start thinking about community-led research. What are they interested in, and how can we build our research objectives around those interests? How can we now start to think about using non-destructive methods instead of exhuming human remains, having them placed or kept in museums and institutions and collections? Even thinking about how we can address these unethically collected remains, right, from these other institutions that now exist, or even with the Smithsonian. The only other thing I want to point out about this slide is the title of our lab, right? So when we think about this type of work, we also think about decolonizing science, right? The Clinton lab sounds very nice, but the Ashes Lab incorporates not just the researchers in the lab, the students, but also the community, right? So Ashes for ancestry, soil, health, and evolutionary studies, because that's what we do. So moving forward, we want to take all of these proof of concept ideas, everything that we built on this foundation with the New York African Burial Ground, and execute them at different burial sites around the country, where, as we find, a lot of these communities don't want to exhume the remains, unless they're at risk of having a highway built through the site or not, right? But we can go in, leave our loved ones at rest, and take these soil samples and still learn the same information. We want to encourage the community to teach us. We are coming from a place of privilege going into these communities and offering this service. Validate these community beliefs. I have a short video here. If you look at that center picture of the woman who has what we describe, or she calls, dowsing sticks.
To the west.
Yep. They start to move on their own while she's just holding them, right? So you see them move in one direction or another. So for the community, these oral histories, these practices, they're valid, right? You may have seen these dowsing sticks in paranormal shows or something like that. They also, you can sense some other structures under the ground if there's like an obstruction or water or something like that. But for this community, that is valid. For them to say, we see the headstone, we see the footstone, we see the depression, but also when we use these dowsing sticks, we know that there's an individual buried here, right? And that's something as researchers that we have to listen to, because that leads to empowerment through involvement, right? So we like to think about research in these two kind of environments, right? In the lab, what we do at the institution with all of these scientific, molecular tools and techniques, and we think about what happens out in the lab. But what could we create if we combine those two things or interchange them, right? Have our researchers out in the field engaging with communities, but also having these communities come into the lab. One thing that I mentioned at a conference that I was previously at was, I can only imagine for someone who has not been trained in biology to be able to go into a lab and pipette the DNA of their own ancestors, right? Or manipulate their own ancestors' microbiomes, or to literally see what a sequence looks like going across the screen. We realize that this is not always possible. People have schedules and jobs. So we also like to consider taking the lab to the community, right? So we've purchased a nanopore sequencer where we can go out and we can do exactly this. Take a laptop, we can not only show the community, but literally have them do the analysis themselves, which adds to that level of empowerment, which then in turn adds to how welcoming they are to, one, a trusted agent in the field, and how willing they'll be in the future to participate in research. And that also answers that question about underrepresentation, right? The final part about the data is, where does the data go? Who should have access to the data? Who owns the data? In our research, in our lab, and we would like to see this kind of incorporated just into research itself, is that the community owns the data. I don't know how many papers I read about the genetics of enslaved Africans or from an enslaved burial ground, and it's put into a genetic database that is specific to biomedical research. That means the community themselves cannot even access that data without going through the institution. This is their own ancestors' information, right? I don't know if anybody else sees a problem with that, but maybe that is something that needs to change. What we're doing, we're also partnering with a collaborator at University of Pennsylvania to build our own database, right? To make these things public for researchers, but also to have a space where the community governs their own research. They get these requests from researchers, and they get to decide whether they want to release that information, how much of that information they want to release, when, where, and how. I'm going to jump to my extra slide because I have a few minutes, which is nice. This point was mentioned earlier, right? So addressing this question of addressing this challenge, of representation for communities that are underrepresented is on a two-sided problem, right? The community understudy, but also who are the communities who are doing this research? Right. So on the left, we have all of the undergraduate researchers who worked with me on the New York African Burial Ground Project at Howard University. And on the right, we have the Ashes Lab, the researchers who are interested in doing this type of research, right? Who may come with a different perspective, who may have this experiential, common experience with the descendant communities, which means doing the science differently. So to kind of sum up, I want to think about ethical reevaluation of current practices, permissions, privacy, who has access, reframing research goals, right, around what the community may be interested in, encouraging community participation through trusted agents in the field, right? Data equity, right? Bioethics and bioequity should also go hand in hand, right? How does the community benefit from this type of research, right? Can we tell them something about their own microbiomes or health and disease, right? And finally, advancing restorative justice, right? Acknowledging these past injustices so that we can learn from them to do this science in a different way. So with that, I just want to acknowledge all of the moving parts that it took for this to happen from Howard, Penn State, and also NC State, and thank you for your time.
Great talk, Carter. All right, next up we have David Resnik. He's a bioethicist from the National Institute of Environmental Health Sciences. He'll be presenting his work thinking about the risk of engineering the microbiome in the built environment, a precautionary approach. Why don't you take it away?
Thank you. It's my pleasure to be here today. I'm called in at the last minute, a pinch hitter, so hopefully I won't strike out. But anyway, I'm going to talk about, just start off, I was thinking about one of the case studies that we're looking at tomorrow. This proposal to release these genetically modified genes into, microbes into a hospital environment to try to fight antibiotic resistance in hospital bacteria and elsewhere, I guess. So obviously it's something that, is potentially very beneficial, but there are risks that we need to think about, and I probably haven't even gotten most of them here, but just a few. Possibly encouraging development of new forms of antibiotic resistance among bacteria. It might have adverse effects on the good bacteria in the hospital microbiome. We all know about the microbiome. It's a balance of so-called good and bad bacteria. And it might have adverse environmental impacts downstream, getting into the waste water and all that, and who knows what would happen. So how should we think about benefits and risks? That's a part of bioethics, been that way for quite some time. You look at well-established principles like beneficence, non-maleficence, and justice. They all involve thinking about the risks, including the distribution of risks in society. And risk-benefit analysis is an important part of public health and regulatory oversight, which I'm sure we're going to be looking at later on today. Unfortunately, it seems to me that insights from bioethics do not get adequately incorporated into environmental and public health regulatory oversight. And I think that's one of the big points from my talk today. I hope you'll see that it comes through. So if you look at traditional risk-benefit assessment in the government, it uses what in decision theory might be called expected utility theory. What you do is you identify potential risks or hazards and benefits for different options related to a decision. Let's say we're talking about an approval of a new drug or a pesticide. You estimate the probabilities of these risks and benefits occurring based on the scientific evidence. You assign values or utilities to these risks and benefits, and then through some magic calculus, you can determine whether the option that best minimizes risks and maximizes benefits for society. The problem with this approach that a lot of people have argued is that it does not handle the situation of epistemological uncertainty very well. We may lack enough data or information to even identify the hazards that we're facing or estimate the probabilities of risks and benefits for different options. You can always get a subjective estimate of probability, but I'm not really a big fan of those because of bias. What we really need is enough data to make confident estimates of probabilities, and that's often not the situation. There's also moral uncertainty in these kinds of situations. We may lack agreement on how to prioritize competing values such as health, environment, economy, and so forth. And these things will be in conflict a lot of times when we're looking at pesticides and all kinds of other things that we're talking about approving. So in the 1970s, the precautionary principle was developed as an alternative to traditional risk-benefit assessment to try to handle the situations of uncertainty, which the proponents said traditional risk-benefit assessment does not handle very well. In a lot of these situations, traditional risk-benefit assessment would recommend that we do nothing until we have more research, right? That was what was said about climate change a lot of time in the, say, 1970s, 1980s, 1990s, even today. It's like, well, we don't have enough data, so we got to just do nothing, just sort of let things go. We don't have enough data. But we may need to take action to avert bad outcomes, even when we lack scientific certainty. So it is better to be safe than sorry. That's sort of the theme of the precautionary principle. Now, it can be a proact, can be like avoiding things, bad things that are catastrophes waiting to happen, like climate change, trying to do something even when we don't have all the data yet. Or it could be in a situation where we're trying to enact an intervention, like mask wearing at the beginning of the pandemic. There really was not a lot of good data in early 2020 about what type of mask to wear, what would be the best kind of, there was a lot of jostling back and forth. It took a while to really get that data in, but most people agreed that, look, it couldn't be a really bad idea to encourage people to wear masks at this point to try to prevent the spread of disease. So the precautionary principle, there's a lot of versions of it. One of them is something you find in the Rio Declaration on Environment and Development. In order to protect the environment, the precautionary approach shall be widely applied by states according to their capabilities. Where there are threats of serious or irreversible damage, lack of full scientific certainty, there's that concept again, shall not be used as a reason for postponing cost-effective measures to prevent environmental degradation. So there are a lot of versions, as I said, of this principle in international treaties and things like that, especially in EU countries. It has not played a prominent role in US policy or regulation. A lot of that is because of criticism from the business environment and economists about the problems with application of the precautionary principle. So just to quickly go over some of the objections to it. It's vague, poorly defined, people have argued. Of course, the reply to that is define it more clearly, which I have tried to do and others have tried to do in this field. It's incoherent. It may recommend conflicting options, like what is the bad outcome you're trying to avoid? You would avoid this bad outcome, but you end up with this bad outcome. You can't really tell you what to do. Well, this is not unique to any kind of general principle. Beneficence, non-maleficence, justice, all these things in bioethics could lead to contradictory recommendations. So the way to deal with this is to supplement things like the PP with additional policies and procedures that help you get to some kind of coherent answer that you're looking for. It's overly risk-averse and therefore anti-science, anti-technology, and anti-economic development. And I would say definitely there are versions of PP, the precautionary principle, that are almost like what you would say max-min in decision theory, like just avoid the worst outcome. Do anything you can to avoid the worst outcome. Even if it's like science fiction outcome, bad thing happening, like the whole world blowing up or something, just do whatever you can to avoid it. And these things obviously are not very reasonable. So you have to have a version that takes into account the costs of precaution. That is, what would you lose, what opportunities, what benefits would you lose if you're being really cautious? And also include alternative forms of precaution. So a lot of people talk about prevention. Well, but there's not just prevention. There's minimization. A lot of times in environmental risk assessment, you're talking about minimizing the risks or mitigation. Something bad happens, well, we're going to take steps to try to make it not the worst thing in the world if it does happen. So here's the definition that I favor. I think it's a pretty good definition, but I'm open to a lot of debates about it. In the absence of degree of scientific evidence required to establish accurate and precise probabilities for outcomes related to a decision, take reasonable precautionary measures to avoid, minimize, or mitigate plausible and serious harms. So the plausibility thing is important in there because we're not talking about crazy sci-fi scenarios. We're talking about bad things that we think could happen that are grounded in scientific fact. So what do we mean by reasonableness here? And this is not to be confused with rationality, which you see in economic theory and in certain kinds of moral theory and things like that, this sort of pursuit of certain goals. Reasonableness is like more of an all things considered best thing to do morally and taking everything else into account. So these are some criteria that I would recommend for reasonableness. There could be others, probably are. These are just some, I think, important ones. Proportionality, that is, measures must balance plausible risks and benefits proportionally. So if you have something really risky, you got to have a big benefit that you're hoping can pay off here. Fairness, reasonable measures are based on a fair balancing of risks and benefits. Fairness includes distributive and procedural fairness. Epistemic responsibility, reasonable measures comply with norms for the responsible acquisition and utilization of evidence, knowledge, and expertise. So you're making a decision based on the best available data, and you're continually looking for more data and revising your policies accordingly. And consistency. You have to, if you do one thing for one chemical, another chemical has basically the same risk, you should be treating that chemical the same way. So in terms of proportionality, some things to think about here. It's not quantitative, but qualitative. I often think of it as like, I think there's an important difference between quantitative and qualitative risk assessment. I think we're all familiar with sort of qualitative risk assessment when we take medicines and they talk about certain likely and most likely, and they're not really putting a number on it, but they're telling us sort of degrees of risk we're facing. It's sort of a qualitative assessment, but not numbers that we can put on it, sort of more of a degree of risk. Risks are justified or acceptable in terms of benefits. And there are lots of different risks and benefits we need to think about. In traditional risk-benefit analysis, they'll consider health and things like that. But there are other things to think about. We need to think about how something might affect culture, for example, which was just talked about. The broader environment, for example, we need to think about. There's a lot of different risks and benefits we need to think about when we're looking at proportionality. In terms of fairness, the distribution should be fair in terms of distribution of risks and benefits. So we need to think about who benefits most, who are placed most at risk. Are risks voluntarily assumed? There's a question of whether, this is really voluntary. There's people under certain kinds of coercion and whatnot. But I do think there is a difference between someone who decides to skydive and someone who's pushed out of an airplane or rides in an airplane that ends up they fall out of. I do think there are certain risks that we take voluntarily, and that must be thought about in the fairness here. The decision-making process, I think that was alluded to earlier with the community engagement piece that was talked about so nicely. How is risk-taking decided? Is the process democratic? Is it open? Is it inclusive? Is it deliberative? And so there's a lot of things we could look at here for a fair process of getting everybody, the community, the broader public, the stakeholders, all the people that are, affected by something involved in the conversation, just instead of the big wigs in the government and industry telling us what's going to happen, which unfortunately happens way too much. So, final thoughts about this. Although I believe the precautionary principle is a very useful principle, I'm not one that says it's the be-all and end-all to risk decision-making about risks. There are a lot of models out there developed by economists, philosophers, others, for making decisions about risks. And I think actually that what you have to do, and you have to decide what is the best strategy for making this decision. What is the best risk? Are we in a situation here where there's so much unknown that it's almost like max and min, just avoid the worst outcome no matter what? Is it a situation where we have a lot of evidence and we have a lot of agreement about values, so we actually can use traditional risk-benefit assessment or expected utility theory? I think generally when you have a new technology, something like the precautionary principle makes sense when there's a lot of uncertainty about the technology and you're starting out with it. For example, autonomous vehicles, putting autonomous vehicles on the road. I think a precautionary approach makes sense there. There's a lot of other areas where I would say that's what we should do first, but it's possible that when the uncertainty resolves, that then you sort of institutionalize the technology, and you can go to more of a standard risk-benefit analysis at that point, I think. So in my opinion, and I could be way, way off base on this, but just my initial thinking about this problem is that when we're talking about engineering the microbiome, the built microbiome or the microbiome inside your stomach or wherever, I think we should take a precautionary approach to this until uncertainty is reduced. Thanks.
All right, great talk. And last up, we have Nicolae Morar. He's Associate Professor of Environmental Studies and Philosophy at the University of Oregon. And his talk today will be about bioethical challenges regarding microbiome engineering.
Thank you very much. I definitely want to start by thanking the organizers for all the effort for having us here. Thank you very much. They really deserve a round of applause. It's really hard to follow Carter and David, and I'll do my best. I realize that I'm standing between you and lunch, which is a difficult task, and I have way too many slides, probably a little bit too much philosophy. So here's what I want to propose you. I want to tell you that there is a benefit in introducing a useful distinction between causation and constitution. I'll tell you a little bit more. I'll gloss over my motivation, which is sort of learning something very interesting that microbiome studies have produced relative to sort of biology, the ways in which we understand biology. And I'll tell you what I hope to do is map a little bit, use the distinction between causation and constitution to map at least some epistemic challenges, and then hopefully to give you a little bit of a map without probably getting too much into the details of some of the bioethical challenges at heart. So I want to open with Wendy Mitchell. Consider Wendy, she's a person who's struggling with dementia. And there is something very interesting. In order to fight with dementia, Wendy was putting all those notes throughout her house, was really helping Wendy to navigate her condition. And it was one of the ways in which she was able to actually really have a sort of a certain quality of life given her condition. So I want to invite you to consider something. What would it really mean for us to play with her notes, or even take all her notes away, or even probably destroy all her notes? Would we consider that we're actually really assaulting her, or would we be considering that to a certain degree we're doing nothing than destroying her property? And depending on your intuitions, philosophers of mind, and especially in a famous paper, Clark and Chalmers in 1998, they produced this idea. They had a question that was very interesting, and I'll bring this to microbiology, I promise. Where does the mind stop, and where does the rest of the world begin? And one of the positions that they took is what get to be labeled in philosophy, what is called active externalism. What is active externalism? The assumption in the background is that there are parts of the environment that play a very critical role in some of our processes, depending which are those processes. And what's interesting about Clark and Chalmers at that time, they kind of had this move where it said there's a trivial conception of this, which is to a certain degree it's causal. There are features of the environment that actually have an impact on our cognition. That's kind of obvious. Most of us will sort of assume it. But there is something interesting in Wendy's case, right? There seems to be that in Wendy's case, at least some of those notes are almost part of her memory structure, right? Those are those outside features of her body, which are not brain-based. And that's the direction that actually Clark and Chalmers wanted to make, that there's a robust sense where some of those cognitive processes are constituted by active features of our environment. I could go on and tell you how this paper has transformed the conversations, but I find this very interesting and kind of very important for us, precisely because they haven't Clark and Chalmers at that time, they didn't simply say, oh, there is causation and there is constitution, certain parts of the environment are more causal, others are more constitutive. They went on to say something which for them was really interesting at that time. They applied the parity principle. And they said it shouldn't matter where those parts are. It shouldn't matter if it's in your brain, or it shouldn't matter if it's outside. If they're integrated within a cognitive process, in this case, something like memory, and in this case, they are part of that cognitive process itself. So, too much philosophy to tell you why you shouldn't be Cartesian, don't worry about that, or even what they are proposing here in terms of a functional understanding. But what I really wanted, the point that I want to drive home is really that there is a sense in which, thanks to this causal constitutive distinction that they're introducing, and thanks to this parity principle, there's an assumption that we can expand the ways in which we're thinking about ourselves in a meaningful way, right? Not just simply say, hey, the world makes us us. And the first kind of direction, the first sort of probably objection that they thought was something like this, which is what got to be labeled as the cognitive bloat. What if the entire Library of Congress was in small notes and Wendy could reliably access those notes? Would the entire Library of Congress be part of her memory? That seems too much. Or what if the whole internet, I can easily access it with my iPhone? Does that become part of my memory to that degree that whole internet is my memory? So that's a very interesting distinction, and sort of Clark and Chalmers have a long response as to how do we stop expansion to a certain degree. One health to a certain degree is exactly would be the equivalent on the health side of this sort of blowing the extension to the size of the universe in a very interesting way, and probably not knowing exactly what's ultimately relevant. So if you are so far, you might want to tell me, Nicolae, we've known all of this. You've heard it on Rob's talk. Organisms have always extended themselves within their own environments, whether or not you think of beavers, whether or not you think of ants, whether or not you think of a variety of other organisms. We've already known that to a certain degree, that somehow organisms are not just simply stuck to their own internal biochemistry. But I think there is something very interesting that a number of folks have pointed out in all those sort of talks so far, that there is something new that happens with microbial biology in a significant way. And I think one of the ways in which part of what seems to happen with microbial biology is a shift in the ways in which we understand some of the most basic concepts of biology. And Carl Woese famously says in this manifesto that we're still probably prisoners to a human genome project that kind of drove our attention to just the internal biochemistry of the human organisms. And that sort of focused way too much. We painted a world that is just painted on us humans, right? The world and the universe sort of ends with us, or ends with the famous multicellular, or to the eukaryotes. And part of what Woese was trying to drive our attention, that if you were to look at life as such, you should be a little bit upset. Why? Because there's a clear bias against the smallest parts of life. Most of life here would seem to be those famous, fancy eukaryotes. If I were to show you a pie chart of the same tree of life, it will look most like this, right? Most of it's insects, hence the famous joke by G.B.S. Haldane. What does science tell us about nature? God loved beetles to a large degree, because most of life would seem to be beetles. But Woose's point is different, right? If you were to take microbiology seriously, and that's the motivation in a way of this direction, most of life will look more like this. Most of life would seem to be microbial in nature, and sort of shifting perspective. It's a very interesting sort of one of the points that I'm hoping to drive home, is that it really brings a very different understanding to the ways in which we understand fundamental concepts in biology, but also writ large, other kinds of concepts behind it. Woose's point, you all know it, is that most of life is microbial in life. And if I were to show you the last depiction of the tree of life, you will see that we lost our space on the tree of life. We're somewhere lost there, somewhere with the eukaryotes. But more importantly, would say Woese, there's something very interesting behind this. Not just most of life sort of is microbial in nature, but all life turns on microbial life. From the biosphere to the metabolism and to interaction between all those microorganisms. When he was writing this in 1998, it's a very interesting, the ways in which sort of wanted to bring more attention to the importance of microbial biology. There was a clear sense that the message was received, and to a certain degree, a number of microbial biologists and ecologists have worked in that direction to sort of push a change, right? Invite us to think differently about ourselves, invite us to think differently about nature. And among those people, obviously you all probably recognize this famous paper, sort of lead author Margaret Nightfall. And one of the clearest points is it's time for us to pay attention to the very significant interdependencies that all those multicellular organisms share with their associated microbiomes. And we know that those associations are significant because they play a key role from a variety of studies, from the immune system, from organismal development, to nitrogen fixation, to even to influences our reproductive system. So all those microbial biologists, I think they have drove home a very interesting point about the ways in which we should shift our understanding of biology to a certain degree. And they have all told us probably something like, whenever we look out there in nature, in virtually every organism that we have studied, symbiotic bacteria play a crucial role in host functioning. Philosophers have also sort of contributed a little bit to this conversation, and they probably said something that those relations, instead of being an exception, they seem to be the rule in nature. And if we take them seriously, again, we would need a certain kind of critical understanding of what are some of the core concepts that inform our research, and at the same time, what would be the challenges as we move forward. And one of the things that in that famous paper Mara Nightfall is pointing out is we need new concepts. We need new concepts of what a genome is. We need a new concept what a population is. We need a new concept what an environment is. We need a new concept what an organism is. And in large part, there was a clear response within the literature, especially on the microbial biology side, to try to find an answer. Like, what is the answer? And I think one of the answers that came forward was at least this idea that we have a second genome, and we should take this second genome very seriously, and as part of our whole genome. And you've heard about it, you've seen the pictures in Amber's talk. It brings tremendous diversity to our bodies. It adds tremendous genetic diversity to our bodies. It definitely plays a role in, as a modifier of disease, as an essential component of immunity, whether or not in our, definitely in our metabolic structure, probably even in our reproductive structures to a certain degree. So why did I start it with Clark and Chalmers? Because one of the questions that I hope you would take in mind is what's our microbiome? Is it more like Wendy's Notes, or is it more like the Library of Congress? Or maybe at the end of the day, you might resist this dichotomy, which I would be very happy if you wanted, if you definitely want to do that. So in a way, are they just those features that definitely have an impact, like sunlight on our cells? Or is it something deeper that constructs a different understanding of who we are as organisms, or writ large, whatever organisms are in general in biology? And I want to tell you that the causation constitution story, it's very interesting for us to a significant degree, because I think it will map different epistemic and bioethical challenges at the same time. In a series of publications with Josh Korber from Guelph, we had a proposal. We kind of said, we don't want to go all the way. We were way too clinical, focusing only on ourselves. We were either population health and writ large systems on the other side. Can we rethink the ways in which we're understanding those units of health? And we were trying very hard to kind of understand if we can if we can have at least a different direction with this conversation. So we proposed this idea of an extended health. And the assumption of the extending health was that hosted symbionts can be so tightly connected, they're a dynamic functional unit in a variety, and we know this in a variety of ways. And probably the assumption is that we should think of the bearers of disease, health, and disease as those dynamic functional units. And it's a very interesting question here as to whether or not this is true only for our microbes, or in Rob's question as to whether or not we should think of our home as being part of a certain kind of a unit of health. And I think part of the conversation for our panel is going in that direction. So what's the bioethical motivation of all this? Whenever we talk about genetic engineering, or whenever we talk about biological engineering for a very long time, we're kind of, to a large debate, we focus only on the human germline. I mean, if you look about it, whatever came out of CRISPR conversations is don't change the human germline. And there was a lot of worry for very, very good reasons. And part of it is why is it for a long time that we haven't yet considered what are some of the worries around, precisely engineering this aspect of our biochemistry that will sort of expand to our second genome. When we think about engineering, more often than not, I think we sort of tend to think about engineering in a variety of ways. I think the classical one, it's more like the Lego story, We take parts, you put those parts in a way, you do get an aggregate function, and once you get that aggregate function, you hope that it works for a while. And for a very long time, this approach really didn't pay that much attention to our second genome. It was this sort of parts to whole approach that we've seen through synthetic biology. It's not the only type of engineering that we've seen. We could have seen also something like evolutionary engineering that functions through selection. You can start somewhere with mustards, wild mustards, and you can get to kale. But even in this case, right, it's a holistic approach, at least it's not a sort of Lego approach or a machine or kind of a more mechanic approach, but it's still a top-down approach that hasn't paid attention yet to our second genome. So I think microbiome engineering has something very interesting for us. At least it introduces certain kind of variables. And it's long due, and this workshop that as a humanist I really, really welcome. I share Amber's concern that it's extremely hard for us to do work across the disciplines and even have conversations that are meaningful. The point is, yes, this is a novel way. We should be thinking about what it means to change our biochemistry. And what is the set of, again, epistemic and bioethical challenges that are largely unexplored? Whenever we think of microbiome engineering, already it was mentioned, you can start seeing some of the things that we've been considering so far, and that's the classical approach, the traditional approach, right? Find that specific taxa that you kind of know what it does. Once you know what it does, well, you know that it's part of the healthy microbiome. Put it in a yogurt and see if it works. And we kind of know that it works to a certain degree, right? We still absolutely, this is an extremely tricky conversation as to what does really consist as being a healthy microbiome relative to the tremendous diversity that we've seen in a number of studies. That's definitely one of those approaches when it comes to microbiome engineering. And we've seen more recently a slightly different approach when it comes to microbiome engineering, which is more evolutionary or holistic approach, right? Where we didn't pay as much attention to who's there or whatever they do, we were kind of selecting on a black box. And we wanted to see as to whether or not selecting on a black box will have an impact on some of those organismal features. And this is really one of those protocols that pay significantly more attention not to who's there or what are they doing, but as to whether or not there's an impact on the host function by using a community of multi-level selection. How have we done that? I mean, I'll jump over this slide. I'll tell you more about this. I'll just want to show you what a bunch of people have done in a very interesting way. Well, they did multi-selection on this, right? And Miller and Sachs have had this really interesting question as to how are we moving when we're selecting only on communities without specifically knowing who's there, but we're just selecting on different communities, and we want to measure the impact of those communities on some very specific phenotypic trait that we're interested in this case. By the way, this is not a trivial phenotypic trait that they picked here. They picked here fluorescence, and they picked here the time of fluorescence, which raises a tremendously interesting question from an evolutionary perspective. Like, how is it possible for organisms to actually not control that part of physiology that is so fundamental to their evolution? I mean, that's a very interesting question that I'll raise in a heartbeat. So what do we learn from this? We learn we learn that microbiomes are efficient because they can have an impact, directly influence a very specific phenotypic trait. We know that we can do that. We can enhance function or change function without knowing the microbiome composition or even the synergistic interactions that exist. And we learn also something very interesting, that selection of microbiome can be efficient, again, because it mediates to a certain degree microbiome assembly, and it plays on the relative abundance of some of those components. So there is something interesting of the ways in which microbiome engineering are definitely bringing different conceptions of engineering to a certain degree. And I think there's something new that we haven't seen so far, right? On the genetic engineering, on the evolutionary side, most of the time we placed a lot of emphasis on the genetic structure of the organism or the host. Here, in this case, with microbiome engineering, we're paying attention to the importance of the microbiome relative to host functioning. Two kind of interesting epistemic challenges and a series of bioethical challenges as we move forward. There is a very interesting question in the background, and the question in the background is, how is it that such a significant trait like fluorescence for a plant is actually not controlled by an organism's physiology? And this is very interesting. This is the constitution part that I want to drive home. What does it mean? Evolutionary speaking is really a bad strategy. To a certain degree, it's really a poor strategy. So one of the ways in which we think about this is that it's an invitation to think differently about what heritability will look like. And this is the whole story about sort of the importance of the second genome, that we should think actually of microbiomes as being part of this network. And definitely this network will have a significant impact. We should, if we do so, we should pay attention to the importance of vertical versus horizontal, and we should pay attention to the importance of fidelity in transmission. And those are very significant challenges that are still largely debating into the conversation. And it does something else, right? Or we should consider something also very interesting. This is more on the causal side. We should consider microbiome as the most immediate environment that actually a host would have. And if we do so, then we would have to consider it as part of the E, but we would also have to consider it as probably being sufficiently homogeneous, because we can see those functions repeatedly, those host functions repeatedly playing off. If that's the case, again, microbiome, again, we also should pay attention to the degree to which it's not subject to unilateral selection of the host. That's a very interesting sort of conversation as to how do we think about those questions. And I think the other question that we should be paying attention in this is alongside the constraints argument. To a certain degree, probably it's unavoidable. Macroorganisms just cannot avoid, and they cannot even do that energetically in a very interesting evolutionary perspective. The other point that I wanted to drive home is on this side is definitely a new invitation. Microbiomes, from an epistemic perspective, they're bringing something very interesting, and they're sort of shifting attention towards the importance of ecology and towards the importance of evolution. Towards the importance of ecology, why? Because we finally need to pay attention to community diversity, we finally need to pay attention to relative abundances, we finally need to pay attention not just to host-to-microbe relations, but also microbe-to-microbe relations within the composition of those communities. And I think to evolutionary processes precisely because, as you've heard already, through the problem of hygiene hypothesis and so on, we should pay attention to the taxa that probably got lost. And why? And what was the impact of some of those on the loss of taxa on human health? Those are important questions for us, and microbiome engineering, in many ways, it sort of drives that conversation, Now, along the same lines, whether or not Wendy's notes are nothing but her property, that's the sort of invitation. Are Wendy's notes nothing but parts of the environment? And if we were to alter those parts of the environment, we would definitely have an impact on Wendy. The question is, we would have an impact probably slightly more than the sunshine or slightly more than other kinds of environmental impacts. Often in bioethics, we function with the assumption when we constructed bioethical issues that there is a subject, and definitely we can alter the welfare of that subject. And if we were to do so, there is a series of problems that we should pay attention. And some of those problems that they already got sort of formalized in many, many ways. We should pay attention to ownership and property. We should pay attention to questions of privacy. We should pay attention to the ways in which we do research. We should pay attention to the ways in which we store some of those biological information. And there are tremendous papers that have pointed out in many, many, many ways. One of the speakers here. If you haven't had the chance to read this paper, please read it. Who Owns Your Poop? It's a fantastic paper that Kieran is one of the authors, and definitely points out to limitations and to problems of ownership and to problems of biobanking. There are a number of questions that are raised in the literature around privacy and as to whether or not we can actually use the microbiome as an individual identifier. And there are actually forensic papers out there that point out that probably to a certain degree our microbiome is an even more individual, has a more individual signature than our own human DNA would have. So we definitely need to pay attention to those questions. We need to continue to pay attention to the ways in which we do research on human subjects, and as to whether or not, what kind of information is there, who's represented, who's missing, what is the access to the data, how do we use this data, and all those questions will tend to be central to the ways in which we move forward. I want to invite you by saying that maybe Wendy's notes are part of her memory. And if Wendy's notes are part of her memory, not just part of her environment, there are a series of bioethical questions. They're different. They're not ownership. They're not about the ways in which your data is stored. They're not about the ways in which you can have access to your data, which very much assume a certain kind of subject functioning in the background. If all those environments are part of who you are, then I would say that probably playing with Wendy's notes is more of an assault than actually changing the property parts. And there is a way in which engineering the microbiome would be more than just changing an environment. We could really look at something more significant depending on how we construct that environment and depending on how we frame that dynamical unit that we've been having so far. And if we're moving in that direction, we will have a series of other questions that I think are very interesting, and you've heard Amber so far. There's going to be questions as to what does it mean to be a self or an individual. There's going to be questions about what it means to be healthy. There's going to be questions as to what our species boundary, which have functioned as a tremendously important category, moral category. Then there's going to be question as to what does it mean to be a human, and you've heard all of that so far. And some of my work went in that direction, sort of challenging some of our basic moral concepts by sort of looking towards this idea that autonomy gets porous, especially as we're thinking of those subjects as not being atomistic and individual and clear-cut, but more constructed both by their social and biological relations. I think it raises tremendous questions, not just around what it means to be healthy, but it also raises tremendous questions of this idea of spreading health. We often think of microbes that the only thing that they can spread actually is disease, and I think the microbiome of the built environment, to a certain degree, opens a very interesting conversation as to what does it mean to spread health. There's a lot of packed in there, so I wouldn't accept it too easily. There are definitely questions as to what does it mean to be a species, and I think one of the things that are coming back to us from a bioethics perspective are the ways in which, again, some of the clear concepts that we've been deploying both in biology and in our normative structures are just not as clear-cut as we hoped for. And I think last and not least, definitely there's going to be a question of what it means to be human. And as to whether or not, once we start engineering parts of the environment, or when we start playing with engineering microbes, whether, in a variety of ways, what are we doing actually? Are we changing just our most immediate environment, or are we really changing something deeply about ourselves? And if we do that, if we're changing something deeply about ourselves, we'll have a very different ways in which we really understand some of the moral harms that could follow there. Definitely, you all know it, the famous quote from Tom Insel, for at least from the standpoint of DNA, we're more microbes than humans. So I want to very briefly close, because I realized that I went way too long. So I want to tell you that I think there is something interesting that microbiome engineering opens, changing our conceptions of genetics and environment. I think it definitely renders the dichotomy nature and social way more fluid, if not to a certain degree, probably something that we should overcome. It changes our conception of heredity, and I think in many ways it underplays the importance of the human germline. And ultimately, I think it changes our conceptions, hopefully as species, and I think that will definitely will undermine the importance of some of those categories as they pertain to bioethical debates. Thank you so much for your patience. Sorry for running.
All right, now we're going to move on to Q&A. I'll ask if we have our three speakers come up here. And Kristen will be walking around with questions. Do we have any online first, Patty?
Not yet.
Not yet, okay. Let everybody in.
So I have to say, I really enjoyed the last talk. That was really thought-provoking, and that's actually saying a lot for me. However, so one of the points I want to make that you made about an organism giving up some key aspect of its reproductive opportunity to microbes, that must happen in a mutualism. Mutualisms are ancient. They occur as early as we have fossil evidence of microbes on the planet. Mutualism was there. In a mutualistic relationship, all partners of the mutualism are giving up aspects of their reproductive success to the mutualism. And I can name hundreds of examples of that, like fungal, mycorrhizae, bacteria. The human gut-brain axis is a perfect example of that as well. So it's not surprising to me at all. It's simply the way life on this planet works. It's always worked that way. Mutualism, again, is common amongst all living things, occurs in all taxa. So that's not at all shocking. That's how biology works.
All right.
I have just a very quick reply to that, because I couldn't agree more. But for me, all your points are a function of the ways in which microbiome studies have changed our more competition-driven conception of evolution, which, by the way, it was fundamentally the neo-Darwinian structure of evolution that we functioned for a very long time, where we placed way more attention to competition than mutualism. And I think
tell you that people like me people like me never thought that way. So when people start talking about the neo-Darwinian paradigm, there have been loud voices within it, and then there are folks like me who have been thinking this way since the 1980s. I mean, I literally used to date somebody in Lynn Margolis's lab. So, and that was in the 70s, okay? So there have been people who have been thinking about the importance of mutualism for decades, but you're right. There are those who've dominated the conversation with the importance of competition. Competition is only one form of interspecies interaction. Mutualism is just as common, and in fact, what we know from evolution isn't that competition is less prominent than mutualism, because selection drives against competitive interaction between species, whereas selection drives mutualism. So that view of nature is absolutely wrong. It's always been wrong.
I couldn't agree more with you.
All right, next question. Erika? Oh, sorry.
Sorry.
My bad.
I have a question for Carter. So your work and lab is so amazing, and your approach to it is just unbelievable and so rare. I guess my question to you is, have you run into roadblocks with funders, with your institution? What will happen if the community says, we don't want you to do anything? We want you to leave and never talk to us again, right? Like, so have you, like, what sort of obstacles have you hit? Because I can imagine a funding agency being like, we want to find this out. And if the community's like, no, how do you negotiate that?
Yeah, that's a great question. I've run into many challenges. I just thought of something else, but I'll address your question first. A lot of the projects that now come across my desk are directly from the descendant communities. So either they read about the New York African burial ground research, or it's through another researcher who's like, hey, I know someone who can do this in an ethical way. And so I have not run into the issue of, hey, there's this site, I want to do research, and the community is like, no, you should leave. Which that is a conversation in itself. Because helicopter research or vampire research, where it's like, hey, can you spit in this tube? I go back to the lab, do what I need to do, is very prominent in science and is wrong. I can't think of any other way to say it. And then the turnaround of the vampire research, generating the data, and then putting out a publication that the community is probably never going to read or understand is the second part of that problem. And we try to move away from all of those issues in the lab. So one, a lot of the projects come directly from the descendant communities. I have run into the issue of everybody doesn't want the same thing. Yes, we do want to exhume. We want to repatriate. We want to take the individuals back to Ghana. We want to, like, so many opinions, so little time. And what I usually do, the community engagement always comes at the beginning, throughout, and the end. In the beginning, we usually have a town hall. So You guys have to figure out what exactly we want to do. I will figure out how we can make that happen with the scientific technology that we have. So if you have to fight to the death, then so be it. Or choose the voice of the community, who's going to be the representative, then that is what I encourage for all of these projects.
Who funds it?
Yeah. So the other part of it is finding an agency that finds value in the research. And even with my doctoral dissertation research, I didn't get funding, even though I wrote a lot of grants, I didn't get funding from these big organizations like NSF, NIH. They did not see value in the project. Instead, I deferred to smaller grants that would fund parts of the research that would lead to the next step. So like Sigma Xi, National Geographic, the Cosmos Foundation, Just Julian, where we would take one of those research objectives, turn it into a project, and fund that part of the project to get to the next step.
All right, Erika, you still have a question for us?
Okay, so to be entirely transparent, I'm a posthumanist who has actually given talks about the inadequacy of bioethics for addressing social and ethical implications of things.
You're not alone.
I appreciate that, and I'm so very glad that you pointed out the challenges with the idea of autonomy. But I want to come back to Wendy, because I'm reasonably sure that Wendy doesn't live alone. I mean, not only, we don't have Rob here at this precise moment, but of course there are lots of other critters in Wendy's house, but she probably also has families or caretakers or other members of her community who help her out. And one of the challenges that I always have with the notion of autonomy is that very few people either want to be autonomous or have the privilege of imagining that they are. This is a really peculiar way of thinking about humans as being bounded creatures who make their own decisions, and where we have some sort of weird distinction between being autonomous and being constrained or being coerced, where, you know, throughout my life I make decisions on the basis of a variety of relationships, and I wouldn't necessarily call that coercion, I would call that living in a group. So I wonder, particularly when it comes to microbiomes of built environments, where we are expressly thinking about ourselves as being more than just human, where does that leave us with respect to the utility of bioethics as a framework for evaluating risk? And I'm thinking particularly in terms of the sort of the unspoken term of your nth meme, that Wendy should be able to imagine that she's autonomous.
Well, many ways of answering your question. And I'm sure that David has been thinking about the importance of autonomy for a long time. We'll have something to add to this. For me, I think there are very interesting ways in which we can reframe the question of autonomy, probably without completely throwing the whole concept away. But one of the ways in which, and by the way, the feminist tradition has been doing that for at least for the last 30 years in a very interesting way. So there are ways in which you don't need to give up on autonomy, I think, and you can sort of include a variety of elements that are somehow moving away from the notion of an atomistic self who does decisions and somehow in a very closed chamber without any sort of influences. So we can do that to a certain degree. I think what the other question that would push forward is, do we need to give up on other kinds of concepts that sort of ground the concept of autonomy? So do we need to give up on concepts of selves? Do we need to give up on concepts of individuals? Should we be thinking about ourselves as communities? Should we be thinking of ourselves as open ecosystems to a certain degree? And I think I see a variety of ways in which that conversation can go. For me, the holobiont, to give you an example, the holobiont conversation is a way of actually saving the story and not going all the way porous and open. It's actually, or from an evolutionary perspective, the holobiont would assume that actually the holobiont is a unit of selection, and that's a way of recreating individuality in a different way. So I don't have a good answer, but I see the tension in the direction that you're going, right? When you start opening the system, do you drive the system to a form of dilution and dissipation, or can you save it in a way? I think that there are interesting ways in which you can save it, but none of the ways in which I sort of put forward is immune to really good criticisms. Yeah. I'm not sure I answered the question.
No, you did. I'm just wondering why we would want to save it, but that's a conversation for another time.
So that's a very good. So I'll have a very quick question. So there is a way in which, two quick things. Even if autonomy doesn't exist, it could be that it's a very useful moral fiction for ourselves. Or if we don't want to save, autonomy, it's worth pointing out what's our positionality when we decide to give up on autonomy, right? Because more often than not, it's kind of easy for me to stand up here and say, oh, we should give up on autonomy, when actually sort of, it's a tricky part because the concept does work in many, many ways. I mean, it does some work, whether or not it's a fiction of a real thing, yeah.
You have some thoughts, David?
Let me just chime in on that too. I mean, I think it's one thing to attack autonomy at an abstract, philosophical, theoretical level, but really, when you look at the worst human abuses of human subjects, whether it's the Tuskegee study or any number, what has gone on seriously, the Nazi experiments, is a violation of autonomy. And if we have a bioethics that can't say that those things were wrong because they violated autonomy and dignity and rights, I don't know what kind of bioethics we have anymore. I really can't even think about that. So,
I mean, that's one of the risks, right? If you were to give up on autonomy, like, what are you giving up on our ability to actually name certain things as being wrong? I think that's one of the challenges. I also don't think that we need only autonomy to name the Tuskegee and the Nazi experiment as being wrong, but that's one of the ways in which we did that. Yes.
I have many other things to say, but I won't say them right now.
All right, other questions?
Thank you. This question is more for Carter. I'm actually really curious about the data ownership piece of it. It's something we struggled with our study and our engagement with communities, especially after it's been a couple years after the end of a study. How do you maintain engagement with those communities so that you can ask for permission, especially if they've moved on to other things? And then also, there's a bit of a tension between this expectation to share data within the science world, of course. So how do you balance that with, making sure that the communities still own it?
Yeah, those are all good questions. I have many thoughts. I don't know which direction to go in. The community engagement piece is going to define what data ownership looks like. So even after the scientific component of the research is over, the relationship is still there. And so even a year down the road, 5 years down the road, there should be that space that has been developed throughout the research to have that conversation about who has access, any updates, as bioethical regulations change, how do we update, stay consistent, change these things? So I think that first question comes down to the relationship with the community, and the community's relationship with the institution. The second part of it is that is also an issue that I run into, right? I want to publish in this science journal, and now we have the conversation about reproducibility, right? How do I know, how can I also do this, but how do I know you did what you said that you did, right? And so what I'm trying to write into grants now, and also have been in conversation with, which is seemingly successful so far, is that we will share the information to a certain extent, one, by removing certain information, even if it's just offering like the microbiome information or the genetic information, or only sharing it with that entity and not making it public. But you have to make a request to the community who owns the data to be able to use that information, which is why part of all of these dissertation projects and each of these projects in different locations have their own database. I mean, it would be nice to build this network, right, that is made up of these freestanding databases but are still all connected, and that's the dream. But that's the direction that we're going in.
All right, one last question. Do you have any online? No one now. Okay, you're great.
Okay, This, I know we're going to lunch, and this is just an idea thing that I was thinking of. So one of my favorite assignments to give my students is a debate activity, and that is, should microbes have rights? And I'm never going to have an opportunity to ask this of bioethicists again, so I'd love to ask this to you. As we speak of autonomy, and we're still in this very human-centric kind of way of looking at microbes, who speaks for the microbes? I'm just going to put that out to you guys to ask you that.
You have a right to replicate in my red blood cells. Just kill me. Because after all, I don't want to use quinine to protect myself because I can't prevent you from replicating because you are an organism on the planet too. I'm sorry, I probably shouldn't have gone on.
No, I mean, look, I don't even think we're there yet to recognizing animal rights, let alone microbes. I mean, and I'm not even sure talking about rights in bioethics is often the best way to deal with issues. I prefer, because rights, when you start ascribing a right to something, you introduce a conflict of rights, like the gentleman was just saying back there. So, I mean, and then the conflict of rights often is harder to deal with than just looking at things like duties and justice. So, anyway.
All right. Any final thoughts? And now we'll move for lunch.
And just return by 1:45.
All right. Be back by 1:45. Sounds good.
Thank you.
All right. Thanks, everyone.
And I sent, almost 6-year-old goes to after-school program in the States, and after school, or even anything they get in the normal school session. So she won't touch her lunch and save it all up until it's after school time. Yeah, Amber was saying that 12-year-old doesn't realize she can see what she buys on her lunch money account.
Like very specific, like sun chips, like it tells you very specifically.
But those are the health issues.
I made my daughter actually. Yeah.
a 6-year-old after-school program that's much better snack than what they get at school. Will not much impact her.
That's like great meditation.
Exactly. She's a planner. She's strategic. All right, folks, I invite you to get a last caffeine boost, something to drink, and we'll start up our panel in just a minute.
Oh, yeah, there wasn't.
Better that the cup is made out of corn rather than sugar. Okay, everyone. It is my pleasure to welcome you all back from lunch. I hope that you all are well sated and you've had a good bite to eat and something to drink. I hope the same for all of you at home. I hope that you took a nice break and were able to also treat yourself well while you learn about all of this while wearing pajamas. I'm a bit jealous. My name is Dr. Christopher Cummings. I am a senior research scholar also with the Genetic Engineering and Society Center here at NC State, and it is my privilege to be able to introduce our next panel of speakers. I also do have one housekeeping note from Patty, who asked me to let everyone know that the PDFs of speaker slides should all be completely up by the end of the day, so they might not appear perfectly right after each presenter, but they will all go up on the site by the end of the afternoon. With that, our next panel is on risk governance of the microbiome engineering of the built environment, and I introduce Dr. Megan Lott to come up for our first presentation. You have 20 minutes, and I'll give you a 5-minute warning.
Great. Hi, all. I wanted to start off and echo the thank you and appreciation for the organizers of this event. I really appreciate it. And to the participants, I've learned a lot and am thinking and rethinking about the ways that I conduct my science as a microbiologist and how I contextualize it. So I really appreciate the feedback. I'm a postdoctoral fellow at the University of North Carolina, and I'm subbing in today for Dr. Joe Brown, who's actually out collecting more samples for us today in Bolivia. But we wanted to give you all a little bit of insight and information about our work integrating quantitative microbial risk assessment into the study of the built environment.
As members of the premier ERC and a collective of engineers and public health microbiologists, our research group is interested in characterizing the microbiome through the public health lens. So we want to know, ultimately, how does the design and operation of a building translate to microbial exposures and then to public health risk? In our field, we've seen a rapid development of new measurement technologies for microbes. From field-based technology to omics and informatics, we now have an abundance of microbial measurement data from the built environment. And as for our group, we want to contextualize this data, and specifically contextualize it within public health. Typically, public health practitioners will implement an epidemiological framework to assess health impacts. So for example, we could design a cross-sectional study where we recruit a cohort of individuals and assess the associations between mold and dampness in the home and respiratory outcomes. If we design a study really well, perhaps using a randomized control trial with a control and intervention arm, then we can start to measure health impacts directly and with confidence, and we can determine some causal inferences about exposures and health outcomes. However, conducting these studies is really difficult. Conducting them well is timely and cost-consuming. So this might not be appropriate for our studies of environmental engineering interventions. They might be impossible to randomize and to blind, and for these constraints, we've turned to a different alternative framework to assess health impacts from microbes in the built environment. Cue the quantitative microbial risk assessment. This is a modeling framework that's used to estimate infection and disease risk directly from microbial measurements. We build a model where we input the quantitation of microbes and an estimate of their viability, and we receive a daily or annual risk of an infection or other relevant clinical response. So this framework parallels well that of epidemiology, where we're trying to determine the association between exposure and disease. In QMRA, what we're trying to do is use modeling to determine with empirical data from the literature those exposures to certain microbes in the environment, and then use established dose-response models for that specific pathogen to then estimate a measure of risk. So this approach has been widely used in establishing standards for drinking water safety, food security, and safety, and has been funded by NSF for the past 30 years. So the advantages are this is pretty mechanistic. We use compartmental models to follow the fate and transport of microbes from the environment to humans, and then use dose-response models to predict an estimated response. We can conduct some experiments in silico that might not be possible in real life. So I'm imagining thinking about inhalation of fungal spores. I might not be able to observe or measure that directly for an individual, but I can use parameters such as sporulation, ventilation, aerosolization, and inhalation rates. So there are a lot of disadvantages and asterisks when it comes to QMRA. We sometimes joke that it should be renamed quantitative microbial risk assumptions because we are integrating a lot of assumptions. And so it's really helpful to hear about some of those assumptions that we're integrating and those SEI implications as well. An example of an assumption is about the viability of our organisms. So typically we're relying on PCR or sequencing data, and we know that DNA can persist in the environment long after the pathogen has lost infectivity. And so we need to find methods to estimate viability and make the assumption that those are accurate estimates. So considering these constraints and boundaries, I still assert that there are a lot of things that we're doing in Premier that can be implemented into a QMRA. I'm going to bring you all through an example using some of the data and work that we've collected with our partnership and our collaborators in Bolivia. Joe has had a long-standing partnership with these collaborators where they've been working to characterize the microbiome of the built environment in Bolivian households. They're trying to evaluate the association between microbes in the built environment and those building characteristics, those physical components of the buildings. And we want to go a step further and try to translate the pathogen data that we observe into risks, exposure risks. We're in the process of analyzing the data that we collected from our 2023 pilot study, where a series of environmental samples were collected from both urban and rural homes. Sorry for the binary. And these samples are now being analyzed by multi-pathogen quantitation, real-time PCR, for 40 different pathogens. And one of the pathogens that we detected through this PCR method was enteropathogenic E. coli, EPEC. EPEC causes an inflammatory response and diarrhea by producing lesions in the small intestine. This is a concern specifically for young children, as diarrhea is a leading cause of morbidity and mortality, specifically in low-income settings. So here's a histogram of the EPEC that we detected in our study sites. While 12 homes were characterized, we really didn't find that much EPEC. But in 2 of these sites, we did find measurable and quantifiable amounts of genomic material from this enteropathogenic E. coli. In addition, we were able to culture some viable E. coli from these same house dust samples. This indicates that at least maybe a fraction of those EPEC are still viable and pathogenic. And this is where we use our QMRA framework. We want to ask, what is the health risk of that EPEC E. coli that you measured in the house dust? We've come up together with the problem formulation, and I'm going to walk you through each of the steps of QMRA to sort of demystify the modeling process. There will be math. You're welcome to take notes and check my decimals. I really appreciate it. We'll start with the exposure assessment. Maybe household dust doesn't come to the top of your mind as one of the primary or relevant exposures, but we do consume a bit of dust every day, and this consumption varies by age. So we're going to take these values from the EPA Exposure Factors Handbook for children under the age of 6 to estimate the risk of EPEC in household dust. What we're trying to estimate is exposure dose in colony-forming units. So what I've done is I've estimated from our samples from La Paz that there's approximately 3 CFU of viable EPEC E. coli per gram of dust. And this estimation should have a big asterisk. I did have to impute a lot of data because a lot of microbial data has a lot of non-detects. Big asterisk. And then we're using the EPA Exposure Factors Handbook. About approximately 60 milligrams of dust per day is consumed by children under 6. So this comes out to be a small exposure dose, less than 1 CFU per day. Is that low risk? How do we contextualize that? Well, we'll use a dose response assessment from some empirical data from the literature. Here is the dose response model that was predicted from the Tackett et al. Study from the year 2000, where 20 consenting healthy adult volunteers consumed varying levels of EPEC E. coli, and we measured the shedding in feces. We're able to use this data to then estimate the infectious dose required to infect 50% of the hosts. That's the ID50. Additionally, we're able to model this K parameter, and that's the probability of each organism surviving to reach and infect at an appropriate site. We'll pull this K parameter down into our model. We're using an exponential model with factors of both dose that we estimated together and this K parameter that's specific to EPEC E. coli. I'll very quickly walk through the math that we're implementing those factors that we measured together and from the literature to estimate the probability of infection from EPEC E. coli in house dust for children under the age of 6. And it's approximately between 30 to 40 million. If we assume that each day is an independent trial, we can calculate the annual risk of infection. For us, that's about 1 in 10 million. So to add some more context, how risky is that? Well, we typically use 1 in a million as the risk threshold for microbial risks. So in that context, this is relatively low. So congratulations to the group. You just conducted a QMRA. And in summary, we use some of the microbial measurements from the built environment to estimate the daily risk of pediatric infection and the annual risk of pediatric infection. We determined that this risk is relatively low. So great. What about other pathogens? Well, we're still analyzing the data from the 39 other pathogens. And with our collaborators at UNC Charlotte, we're also analyzing some of the metagenomic sequencing data to estimate the prevalence of other pathogens in these samples. This still opens up other lines of inquiry, like what are the sources of pathogens in the environment? Which engineering controls can we design to promote healthy buildings? And how do we ensure that those engineering controls are appropriate for the setting? La Paz in Bolivia, most households don't use an air conditioning. So what are the alternative methods that would be appropriate for this context? Once we implement these engineering controls, we can then use QMRA to ask how well did these impact a public health risk? And importantly, we can do this data typically without interacting with humans or conducting human subject research. We're collecting samples from the environment. And that reason that we're reducing the risk to our participants as well. Here are just a few ideas of the things that we think about for QMRA, but really what we want to share is that this is a tool that can be pretty nimble and cost-effective and a way to add additional context to the microbial measurement data that we all are collecting here in Premier. With that, I'll thank my collaborators and hope I did Joe a good job. Yeah, thanks all.
Okay, to continue our discussion on risk governance, I invite Dr. Chris Wozniak, please. You'll have about 20 minutes. I'll give you 5 minutes.
I appreciate that. Thank you. All right, good afternoon, and I want to thank the organizers for the invitation to talk to you today. My talk, talks about U.S. regulations. Obviously, we're not going to cover everything in 20 minutes. This is just sort of a brief overview. But it's also, I should forewarn you, since I retired a couple of years ago, this is my 25-year rant before I finish. You could be a little more honest about the work you did once you're gone. But I want to emphasize that it's criticisms of the system, of the regulations, not of my colleagues. Make that very clear. Okay, so with that said, I want to touch base at least on all of these subjects, some more in depth than others. We'll start with a little bit about the coordinated framework, which many people refer to as the uncoordinated framework, and I'll show why that is. Talk mainly about EPA regulation, just because that's where I worked for 25 years. Even though I did work for USDA, this is where my sort of emphasis is, and quite frankly, with the way the statutes and everything, regulations are set up, I think for the kinds of things you're talking about, both on the pesticidal, FIFRA side, and on the TSCA side, for bioremediation, biosensors, whatever, I think it's most germane to our discussions. And I'll give you a little bit about the history of notifications and experimental use permits for engineered microbes that came through EPA, briefly about TSCA and a couple criticisms there, and then get into what I call the bias against genetically engineered organisms that's inherent and written into the regulations at EPA and to some degree at the other regulatory agencies as well. So this little diagram here is just meant to sort of remind me to tell you that that sort of lavender area there between EPA and USDA, that's where most of my talk sits today, or within the EPA circle. Say very little about FDA, even though they do have a role. It's, again, getting more into the medical and veterinary, which I don't tend to cover. And I should also point out, you probably are aware of this, just a week ago, the three agencies came out and said they have this new proposed streamlined process that they're going to unveil. And previously, APHIS had come out 2-3 months ago and said by October, we'll have a new guidance document out after public comment is received, digested, and all that. So take what I say with a grain of salt, because if they're true to their word, there will be some changes, and I think that, quite frankly, is a good thing, because there are plenty of changes needed. So the coordinated framework was first developed in 1984, first published in whole in 1986. Remember that back then, this was really before the advent of engineered plants, the way we think about BT corn and herbicide tolerance, soybean, and all that. So this was really about microbes. And there are a couple of examples that sort of keyed the public into this, and I will touch base on those as well. But what I want to point out in this slide is one of the basic tenets of this coordinated framework at the time, and the majority of scientists, at least involved in this, tended to agree that all organisms function under the same principles and laws of physics, chemistry, and biology, and that there are no novel risks per se presented by genetically engineered organisms. And the U.S. policy, at least as proposed at the time, should focus on the characteristics of that biotech product, not the process by which it was made. As we'll see, that didn't go quite that way. I'll talk very briefly here about FIFRA. FIFRA is a complex but mostly well-written environmental statute. It is a risk-benefit statute, but my emphasis here is just to point out, and I think this is probably obvious to most, if not all of you, it's not aiming for zero risk as its sort of goal or conclusion of a risk assessment. It's no unreasonable adverse effects upon man and the environment. Okay, so that's way different than, and we have had some risk assessors that really aim for this. Oh my God, something could go wrong. Well, yeah, you're working with pesticides, you're trying to kill an organism. Yes, certainly that is a possibility and something you work with through your risk assessment, and you also put in mitigation factors into the risk management proposal. The Toxic Substances Control Act has somewhat similar language, no unreasonable risk of injury to human health and the environment. The one thing I will say about the TSCA folks, particularly since I've been retired and trying to write a paper about the history of what organisms have come through, they're very much a closed box. You just can't get the kind of information you can from the pesticidal side of things. They're two different offices, and quite frankly, it's been very frustrating. I'll just leave it at that. They have their rules on confidential business information, and I think they're a little too favorable to industry. And if you're interested, I can give you a couple citations to papers where scientists have said, we're reinventing the wheel because TSCA folks won't tell us what's been done. This may have already happened, but we'll never know. And that's really the shame of it. The other thing I want to point out, I think it was Nicolae was talking about, changes in sort of the plant behavior, whether it's flowering time, whatever, phenology. Recall that under the pesticide definition, plant regulators, which is a really poorly defined term in FIFRA and in the 40 CFR regulations, is included. So you put out a microbe, and we have had these, engineered to produce an enzyme that does something to an ethylene pathway or something else like that, change the root architecture. Those are pesticides. And quite frankly, some scientists, once they're out in the field, go, oh, you mean I'm regulated? Yeah. So it is something that I mentioned this PREA, M009. That's a category where if you want to know, is my product a pesticide, you pay your money up front, and in 3 to 4 months, they'll tell you, is your product in fact pesticidal? It's not as trivial a determination as one might think. 25B2 exemptions, these have sort of a mixed history. 25B products, if you go into Lowe's, Home Depot, some drugstores, you'll find these mosquito repellents, and you'll notice, oh, there's no EPA registration number, there's no EPA establishment number, and that means there's been no efficacy data on them, and you're putting these on your kids thinking that they're protected from mosquitoes, which, as we know, can produce untoward results in terms of vectoring pathogens. So that's kind of the history that taints 25B. But the interesting part of 25B for our discussion is that, if you read that last part of that underlying language, is not likely to cause unreasonable adverse effects to the environment, even in the absence of regulation. And I would argue that the vast majority, if not all of the biotech products we've looked at in the 20-some years I was there, meet that criterion wholly. Because unreasonable adverse effects, it's a very rare event when EPA says, yeah, so okay, some of you remember Furadan would kill almost anything. And in the risk assessments years ago, I mean, it was understood, we're gonna kill tens of thousands of songbirds every year. And even that was not unreasonable, okay? That's acceptable. Some of you know M44, still used by certain federal agencies. It's a cyanide bomb to kill coyotes. It kills some endangered species. It kills foxes, minks, skunks, people's dogs, and has injured young children. That's not an unreasonable adverse effect. So when I compare those examples to, BT corn, to a Bacillus subtilis that acts as a fungicide on a tomato leaf, they're not even in the same ballpark, same universe, really. But, again, have to convince the powers that be to think about it. Most of you, you look like a young crowd, pretty much, may not remember this Diamond versus Chakrabarti, but this was, my whole point is here, this was a big deal. When this came out first in 78 and then 80, the public became aware. Genetically engineered bacteria, they're gonna put them out there. This raised a lot of ire. There was an interesting legal point made there too, that you can, patent a living thing, so that's not to be forgotten. But the point is that, this is what got people going, got the coordinated framework sort of off and running. The other one that came actually in 83, and it took until 87 because of injunctions and people basically trying to thwart this trial for ice minus. It's a deletion of a protein in the outer membrane that causes nucleation of ice crystals and frost damage. Now, why EPA ended up with this, it's a long story, but basically it was a political hot potato. Nobody else would take it. They dumped it on us. And, they got their trials done, proved it. There was briefly a couple of products. There is one interesting product that came out of it that is not a pesticide at all. It's to produce snow. When you go to a ski resort and they're producing snow, that's Snowmax. And that's also a bacterial-induced or derived nucleation protein. But again, look at the hazard suits these guys are wearing. And there was a whole bunch more than this. There was all these Anderson air samplers, everything going on, and the field site was desecrated by vandals a few times. Again, this was a huge deal. So you kind of get that going on in the public psyche. We had a bunch of these field trials. The first one there, the killed microbials with BT inside of a Pseudomonas cell. That's the only one in that group that really made it to commercialization. There are a bunch of others that briefly did or became experimental use permit, experimental field trial kind of thing. But, and then actually going on into the 90s, we had several genetically engineered baculoviruses for forest insect control. Again, from what I can tell, not that many people got excited, but the companies got gun shy. DuPont, American Cyanamid, just Monsanto, pulled the plug, said this isn't gonna fly. And that's why when you go back to the early 80s and think, now here we're 40 some years later, we only have a few genetically engineered bacteria in commercial use that fall under the pesticide realm. And I'm wondering if anybody here can even mention one of them. Anybody ever heard of one? Yeah. As I say, it flew under the radar for the most part, which is maybe why they survived. Because, I guess if you advertise it too much, somebody's gonna question what was going on. So, these biotech notifications are meant for very small scale, less than 10 acres. It's a proof of concept kind of field trial. If you engineer a bacterium at any size release, you have to come into EPA and go through this process. Whereas if it's a non-engineered fungus, bacterium, virus, whatever, you got up to 10 acres, you can do what you want. You don't even have to talk to the agency. So that's that inherent bias that's written into the regulations. That's one example of what I'm talking about. And again, going on with these, I already mentioned that the 10-acre policy that says, you know, and this is true of chemical pesticides too. You don't have to talk to the agency. You can't put it on food and put the food into the food supply, but you can do 9.9 acres with whatever chemical you come up with, and that's legitimate because it's considered at that acreage to be a small environmental risk. Whereas inherently, if it's rDNA derived, oh my God, it must be a risk. You know, we can't let this thing out there. We have to monitor it. We have to follow it. We have to make sure it's basically destroyed or remediated, you know, at the end. And then one last thing I'll just point out here, you know, a lot of people don't think about this, but every state has its own pesticide laws. So for example, in Minnesota, if you're going to do any environmental release, you have to go through the State Department of Ag before you do that. And there have been companies that didn't know that and found out the hard way. And they're not the only state. I think California, Florida, a few others as well. So on the TSCA side, I was talking with someone here in the audience about this earlier, this idea that, well, and this actually evolved in 1984 in the original discussions, intergeneric. So we have DNA from, you know, rhizobium, DNA from agrobacterium. Oh my God, if you put those in the same cell, now we're talking hazard, we're talking risk, we're talking a problem. Really? Based on what? As somebody said earlier, horizontal gene transfer goes on all the time. These genomes are mosaics of how many organisms, I don't even know, but a lot. But this has stuck around and you just can't seem to get rid of it. You could see the obvious problem. If the taxonomy changes, which it does, you know, they split rhizobium into what, about 8 genera, a dozen years ago or more? So now those organisms can all be intergeneric and therefore under TSCA, et cetera. So if you're interested, there's a citation there to a chapter that goes into some detail I wrote with my colleagues from the TSCA side about a dozen years ago. And I point that out only because it's unfortunate. There isn't a heck of a lot out there in the literature. You can find bits and pieces on the website. But if you go to look for an article that explains, well, how does TSCA work? What is a Terra? What is an MCAN? How do these things work? Good luck. Hire a consultant. By the way, I'm retiring, so that's not a plug for my group. Anyway, I won't say much about APHIS because, as I mentioned, they're coming out with new guidance, presumably by October. I like what they've done with the plants and stuff, looking at what they call regulatory status review. Before you launch into a full environmental assessment, just step back and look. Say, have we seen this before? Is it familiar to what, you know, it's another herbicide-tolerant soybean. Yeah, okay, we've seen this. We've done the risk assessments. No need. Just put a letter on the website. This one's not going to be regulated. And I hope some of the other agencies get to that point, at least with some of their products, because again, with the BTs, you know, that's EPA's bread and butter. If we ever killed a rat with a BT injection, we'd have our names and our pictures and our article on the cover of Nature and Science in the same week. Not going to happen, yet they still kill rats every time. Got to do a tox path study, et cetera. Now, all you have to do is a simple criterion, like no beta-exotoxin allowed in that strain that you're using. You can prove that, you're good. You should be good to go. But again, this takes resources, and that's the other point that I want to make. Most agencies, I would say all agencies that I've dealt with, are always resource limited, and particularly EPA, because they're about as well liked as the IRS. And trying to get, you know, a tear shed up on Capitol Hill because EPA doesn't have enough money to do something is good luck. So I'm not real hopeful that things are going to change anytime soon. But when you say, oh, EPA needs to change that, they need to do rulemaking that, remember, that takes resources, and they're just not there. So here's sort of the meat of the equation here. I already mentioned this 10-acre exclusion. Okay, so again, on the GE side, no such exclusion. You have to look at impacts on soil microflora on the GE side, at least under TSCA. And with FIFRA, if it's a targeted, like say you have a antifungal product, well, then you're gonna have to look at mycorrhizae and things. If it's a non-engineered microbe, not so likely, no. Persistence is a big deal, as we all know, whether we're talking about horizontal gene transfer or just risk in general. Exposure is part of the equation, so persistence is involved with that. You know, you go out there, like I said, you go out there less than 10 acres with any microbe you want, pretty, as long as it's not on the select agents list. There are limitations there. But you can go out there and you don't have to retrieve it, you don't have to monitor it, you don't have to say, the assumption is population fell to background levels. But you don't have to prove that. Now, if it's engineered, you will have to prove that. And in some cases, I've had colleagues say, well, we better treat that whole plot with chloropicrin or vapam or methyl bromide, you know? And then you think about the damage that does to that plot. You're not going to have any springtails or mites or anything in there for 10 years. And that's considered an appropriate trade-off by some people. And I, again, would argue that's an inherent bias against recombinant DNA, which goes on in all cells pretty much all the time. I won't go through the rest of these. You can all read. But again, there's a definite distinction made, even though the premise is we'll treat these the same. Now, clearly you have to write down, well, how did I put that gene in and what gene did I put in? That's understandable. But to then treat it in terms of the data requirements, that's, to me, is not scientifically justifiable. Okay, I already mentioned the horizontal gene transfer. Yeah, again, we know that goes on. The one thing I'm interested, why I haven't heard any of you so far mention GERTs, or genetic use restriction technologies, orthogonals, talking about, you know, novel tRNAs, novel amino acids, novel nucleotides. I know a lot of these haven't passed full muster yet, but there are some that are in the literature and appear to work. And I think for a lot of the people that I know in the risk assessment world at the agencies, they're hanging their hat on a lot of that, because someone's going to have to do mesocosm studies and show that, yeah, this orthogonal pathway I have over in my new product, my new bacterium, precludes this bacterium from sharing DNA. The two speak two different languages. You know, an arginine over here, you know, is a glycine here. It just isn't going to work. But again, it takes proof. It takes some evidence. Somebody mentioned earlier, well, just the last speaker, Megan mentioned, you know, qualitative versus quantitative risk assessment. And EPA, at least on the biotech, biologicals, it's all qualitative. On the TSCA side, it's pretty, at least again, with the new microorganisms, it's qualitative. And the difference is that, you know, at least the primary difference in my estimation, quantitative requires lots of pre-existing data for you to really sort of make a judgment. Qualitative is relying a lot on the experience of the risk assessor. And again, you're absolutely right. There are assumptions, no doubt about it. But, you know, I would argue that at least to date, all these cases, you know, have not seen any sort of, you know, I haven't seen the body bags laying around. You know, I think there's enough understanding of what most of these bacteria do in the soil, or at least in terms of them being potentially pathogenic or toxic. They do require, you know, there's I didn't go into the data requirements, but there are tox path studies. If it's a Bacillus cereus related organism, you know, check for diarrheal toxins, et cetera. So there are, you know, things in place to catch those potential toxins, or even in some cases allergens. But overall, you know, I think there's a lot of, are you guys familiar with Z-biotics? Do you know Z-biotics? So I was at a conference at Caltech about a month and a half ago. Unbeknownst to me, the developer of Z-biotics was in the third row. And I pulled out my bottle of Z-biotics and I pointed out that, you know, this is for hangovers. I'm not sure what that is. I've heard if you drink too much alcohol, you don't feel good. I've read about it anyway. And, you know, I said, look, this Bacillus subtilis engineered with an enzyme to break down acetaldehyde. I said this very same subtilist could be used on a tomato plant as a biofungicide. And if it was, it'd be at EPA. It cost you several $100,000 and probably at least a year and a half, if not 2 years, to get it registered. Over at FDA, I think there was one test or two tests they ran. The company did some other stuff on their own, and you can buy it at 7-Eleven and shoot it down without a big concern. So, well, I don't know. I think we should have a test, a group test. What do you say we write a grant, we fund it, and we do a quantitative risk assessment this time? Okay, so on this, there's not a heck of a lot to say that I haven't already said. I'm just basically looking for, what is it going to take to make some changes? And again, not criticizing, my colleagues and things. There is a real shortage of resources, but by the same token, I think you chew up a lot of resources when you regulate things that don't really need regulation. Now, again, I'm not saying that everything genetically engineered doesn't need regulation. Clearly, there are cases where you could really mess things up if you're not paying attention to what you're doing, whether it's an allergenicity front, a toxin, whatever. But I think if you save some resources and time on some of these, another Bacillus subtilis, oh my gosh, what's going to happen there? Nothing. But yet, it's still regulated like we haven't learned anything in the last 30 years we've been doing this. And I don't know if you know this, the very first microbe registered was 1947. It's still on the market. No untoward effects. Nobody's died. The only one that died are a bunch of beetle grubs in the lawn. And it was not engineered, obviously, in 47. Nobody paid any attention. And then BT came along early 60s. A few organic gardeners paid attention. That was about it. But as soon as we hit the advent of a couple of these things coming in with recombinant DNA, then things got worked up a little bit. You see, I don't think there's a heck of a lot here, again, that I haven't already said, other than the fact that, there's a clear bias in my estimation. And by the way, I'm working on a paper with some folks. I hope it comes out in the next couple of months where we outline some of this. And, again, not to be hypercritical, but just to point out that, there is a need for some reconsideration of how we handle, engineered microbes. And I've always been a big fan of Frank's colony vegetable. And, I think he's absolutely right. There are times when it's uncomfortable, deviating from the norm, looking for something that's really a change is not always easy to accept. But I think that that's what is needed here because, I started there in 97. People were talking about a lot of these things back then. As they say, Michael Jordan had his career, retired, came back, had a second career, and has been retired for years. And that's when we started working on some of the data requirements for engineered plants and some other things. And some of it, still hasn't gotten done. Some of this stuff takes a lot of time, but there's a certain point where you say, no, got to get it done and got to move forward. So with that, I'll leave it there. And
Thank you very much. To round out our panel on risk governance, I invite Kieran O'Doherty to please come take the stage. Dr. O'Doherty.
Thank you, Chris. I also want to start by thanking the organizers. I think this is a really important meeting because I think, and I think back to HMP 1 right at the beginning, and there was an ELSI program. A lot of the focus was on research ethics, which I think is important, but really some of us started asking kind of broader questions, and there was no forum really for that. There was, and so I think this is fantastic. So I've taken notes on all of the speakers so far, and I think what I'm going to present actually builds on what many people have said. So I'm going to try and make the connections as I go. So just to situate what I'm talking about and how I got here, I started off focusing on ethical, legal, social, etc. Implications of microbiome science more from a research ethics perspective. I think that's important, but I think it was more interesting to ask questions for broader society? What are the ethical implications for broader society? And so I started off looking at this question of that we've been talking about here, like what if you genetically engineered microbes and throw those out there? And I remember doing an interview study with microbiome scientists in Canada, and I remember in particular one person, his face when I said, okay, well, what do you think about genetically engineered microbes? And his face just went like, oh, I think that's a very bad idea, right? There was a bit of that sense, and I started thinking through some of those issues. But as I was working on that, I then kind of hit on another question, which a few people have already talked about today. Rob talked about it right at the beginning, which is, but what is microbiome science telling us about what we already are doing, which is that could be very harmful? And so that kind of took my attention in that direction, and that's what all of my work at the moment is focused on. So I have sort of 5 main points that I want to try and make, and depending, you never know which audience you have, and I thought I could kind of like skip those first 2 points, but I realize now probably I couldn't have skipped. I need to talk a bit more about the second than I had intended to, but very briefly. So these are kind of premises before I get to my main point. The first is microbial ecosystems are essential to human health. So I think that's probably quite uncontroversial, especially in this audience. But if there's any questions on this, of course, I'm happy to kind of engage in those. The second one, I think maybe I thought was kind of like more taken for granted, but maybe that needs a little bit more explanation. But the second point is that there is something we're doing in industrialized, in big scare quotes, Western societies that is damaging microbial ecosystems in fundamental ways. So I'm talking here primarily about human health, and I'm talking here about in increasing numbers of illnesses that are being linked to some kind of problem with microbiome, and also increases in prevalence of those illnesses. And so this links, of course, to hygiene hypothesis that people have been talking about already as well, missing microbes hypothesis, and so on. The other kind of studies that I want to allude to here are studies on immigration, and increasing numbers of studies that are showing deterioration of microbiomes and health with movement to US and Canada from places like Haiti and other countries. Okay, so then the third point, and now I'm going to start going into more detail, is when we think about microbiome interventions, I think a lot of, and again, I'm talking about human health here, a lot of the kind of thinking is driven by biomedical science, where we identify a problem, the knowledge innovation is like, oh, this has something to do with microbiome, and then the kind of the problem solution step is we need to fix the problem here, like probiotic or prebiotic or something along those lines. But there's very little focus on what caused the problem in the first place, certainly not on a kind of a clinical level. And what we find instead is a lot of rhetoric around, you should, to have a healthy microbiome, you should eat more kimchi. To have a healthy microbiome, you should get a dog, right? So these are individual level decisions to what I'm going to argue are actually society level problems. And so I think a lot of the science is tackling the wrong level of intervention. So here, what I've got, so I'm sorry, this is very small here, so I'm not seeing it very well. So the kind of things that we find in microbiome literature is focusing on things like alcohol consumption, dietary decisions, exercise, probiotics, sanitary living conditions, and so on. So these things have been shown very clearly to influence the constitution of the microbiome, no bones about it. But what I'm going to focus on more is the second, like these other categories. So effects on microbiomes that are not subject to our individual level decisions. What am I talking about here? So I'm talking, number one, effects on the microbiome during childbirth. Now, arguably, okay, this is, of course, still an individual decision, potentially of the mother. But even more important, where in the world are you born? Where do you live? Use of antimicrobials in broader society. So even if you don't use antibiotics, where are those coming from? Environmental toxins and pollution that affect microbiomes. Industrialized food production, so what goes on in the factory, and then, of course, also production like actual agriculture, like the pesticides that are being used. And then, of course, there's also like the whole food desert thing, like in terms of where do you actually live in the built environment and the availability of fresh foods. So those are all factors that we know, like there's a lot of research on this. We know that these things affect microbiomes, but they are not subject to individual level decisions. Let's. How am I doing time-wise? I think I've got time. Okay, so let me then use this. This is to kind of like really just like beat a dead horse. If we take diet as an example, very often diet is presented in microbiome research as, oh, well, you know, you just need to change your diet. And of course, to a large degree, we can do that, right? Like, but only, I should probably also argue, we can do that because we have the privilege of doing so. But there's a lot of research, like of course outside of microbiology, that focuses on the role of culture in determining diet. Also important here is immigration as a factor. There's a lot of research in public health on food deserts, so communities that have low access to fresh foods. In contrast, they have lots of availability of processed foods high in sugar, fat, and salt. We also have lots of research on socioeconomic factors determining diet, and then also close connection between food consumption and the food industry and food industry advertising. So, for example, like studies going back to pre-YouTube time on how many ads on average children see every day for food advertising. And I think it used to be was about like on average 10 ads a day or something like that. And of those 10, 9 ads are for high salt, high fat, high sugar kind of foods. Okay, so the take home here is just to say that yes, of course, to some degree, diet is an individual level decision, but to a much larger degree, diet is determined by social, cultural, and environmental factors. Now, when it comes to just while we're talking about diet, I'm going to link this more to microbiome. A very interesting study recently by Hauptmann and colleagues on Greenlandic Inuit foods, which relies in large part on dried fish. And what the researchers did was they compared the traditionally prepared dried fish, which sort of hangs on the side of the house, to the same product sold in grocery stores. And here I would argue against the regulatory, the coordinated framework that Chris talked about, is that the mode of production does make a difference, because they found that environmental and processing conditions appear more important than fish species in determining the microbiome of the dried fish. So what are the microbiomes on the dried fish that they're eating? And they're very different depending on if they're hanging on the side of the house or if they're produced in a factory and sold in the grocery store. So, and keep in mind, this diet thing is just an example. But when studying the influence of diet on microbiomes, we need to go beyond food type to considering process of food production, and we also need to go beyond individual level decisions to looking at what is going on in the society as a whole that leads to those consumption patterns. So, as I've mentioned, diet is just an example. I'm now going to give a little bit more of a complete list of the kind of determinants that I think we should be taking into consideration. So number one, and by the way, none of these are hypothetical. These all have research behind them, right? So number one, pollution. So we find air pollution is linked to gut microbiome, dysbiosis. Many studies, by the way, my colleague Emma Ellen Verko tells me I really shouldn't use the term dysbiosis anymore. I'm working on it. Other aspects of pollution, microplastics, many different kinds of persistent organic pollutants. Availability of green spaces is another kind of macro factor. Water management is a very interesting one. So here, there's actually not a lot of research on this, but it strikes me as strange. So I'm not a microbiologist, I'm a social scientist, but if I were a microbiologist, I would want to study this, which is the fact that kind of from an evolutionary kind of perspective, we have always drunk water with microbial content, but that's changing now. In only very short past. So, like, what is the effect of that? I mean, I don't know. I have no idea. Then probably one of the most important things are agricultural practices, and I haven't read it, but I know that there was a a report from Friends of the Earth on effect of biocidals on microbiomes. So these are huge effects, and then also, and this is not my field at all. Maybe Chris can weigh in on this, but I believe that there's a lot of biocidals out there that really kind of like a grandfathered in and have never really gone through a regulatory process, I believe. But we can talk about that later. So the fact is that we just don't know. So I think what microbiome science is telling us is that we just don't know a lot about our existing practices in industrialized societies on our microbiomes. Okay, so the take-home message one then is sustaining healthy microbial environments cannot be done through individual level decisions alone. Collective decisions and actions are necessary. So we have to. It's not sufficient for microbiome scientists to say, oh, I did this study. You should eat more kimchi. Like that's fine, but there's a lot more that we need to be looking at. Okay, so the fourth point then is microbiome, and this is something that Amber and Andrew talked about, I think, really nicely. Microbiomes are shared, right? Hugging, kissing. There's studies on sports teams and similar microbes of players in the same sports team. So when we can't really think of decisions I make about my microbiome as just affecting me, right? So we have to consider the microbiome as a shared thing, shared object. We all have access to microbiome exposures on a continuous basis. And at the same time, evidence suggests that our microbial environments are degrading. So to stop this requires action at collective levels. Not just I need to protect my microbiome, but we need to somehow collectively act on this. And I'm not a regulation and policy person, so I believe that it's hard to push these things through. And I also take the point that Chris made about maybe we should be careful about pushing for regulations. But I think in my limited understanding, those are the kind of tools that are available for collective action as opposed to individual action. So the take-home message two, oh, sorry, my take-home message numbers are all wrong. That's okay. So if we start thinking about microbiomes as collective phenomena, certainly in the context of human health, and if we recognize that damage is being done to them in a way that's leading to increased rates of Crohn's and asthma and so on, then that suggests we need to think about these things more like public goods, more like natural resources that we require to survive, like air and water. And so when it comes to, like, say, air pollution, I think it's not okay. I mean, this is, caricatured in movies. Like, it's not okay to say, there's a problem with the air we're breathing. Don't worry, I have a commercializable solution, which is like air in a can. Here you go. We need to actually say, no, we need to stop damaging the air that we need to breathe and survive. And so the argument here is that microbiomes maybe are in that kind of category. And by that, I don't mean necessarily individual microbes, maybe yes, but more sort of the microbial ecologies, the microbial ecosystems at that level. Thank you. Okay, so the main point here is healthy microbial ecosystems are essential not only for human health, they're essential for all life on Earth. In spite of this, there are no protections in place for them. So if microbiomes are as important as microbiome scientists are telling us for human health, why are we not protecting them in some way? Okay, so this leads me then to a concept that a group of us introduced in 2014, which is microbiome stewardship, which is just an idea to help coordinate the protection of microbiomes for sustained health of environments, humans, and other animals. And here is a description. I very purposefully don't call this a definition, for reasons I'll explain in a moment. So what do we mean when we talk about microbiome stewardship? We mean something like this. Microbiome stewardship involves the recognition that microbiomes are an essential foundation for life on Earth. It requires recognition that microbiomes are collective phenomena and that different microbiomes are interdependent. Microbiome stewardship, then, is the conscious and strategic conservation and protection of microbiomes in the face of human and other activities that may lead to dysbiosis, I've got to change that word, and deteriorating health of humans and other animals. Effective microbiome stewardship requires collective societal action at regional, national, and global levels. So when I presented this idea of microbiome stewardship a few times, and there was one At one conference, I talked about this, and then there was, I'm pretty sure it was a grad student, who just as the chair was about to close, chair said, like, any questions? No more questions, no more questions. And just as the chair was about to close, the person managed to sneak in the question into the chat, and then it closed down. And the question was, and it was a very snarky question, but very good. It was a question like, what the heck are you even stewarding? Basically along those lines. And of course, that's a very good question. Because when we talk about like water, it's fairly clear kind of like what the substance is. When we talk about air, it's fairly clear. When we talk about like heritage houses, but what are we stewarding when we steward? And so many people already today have talked about like, we don't even know what a healthy microbiome is, right? Like we don't know what that is. And of course, whatever the answer is to that, it's going to actually vary depending on which environment you're talking about. So it's a very difficult question to answer. So microbiome stewardship, to me at least, is a good intuitive idea, but operationalizing it is very difficult. So we've got a team at the moment. We just got funding, Rob Biko, Emma Allen Verco, Sue Ishak, Diego Silva and Mallory Chudwa, who's actually right here at NC State. We're trying to now actually make this a firmer idea. So some of the challenges we're expecting are, of course, firstly on the scientific level, as I've mentioned already. How do we even define these things? And then Megan's talk about how do we then have measurements? How do we know we're successfully doing microbiome stewardship? We have to have quantitative measures. And then more sort of challenges on the kind of like implementation side is public health. So, like on a theoretical level, has been interested in the conversations that I've had, but on an actual operational level, they're just not at all interested because they're dealing with E. coli outbreaks. We're not interested in this long-term stuff. Conservation people also are not really interested because they're barely managing to have an agenda on endangered, like beautiful creatures. Don't distract us with microbes, right? So these are some of the challenges that we're facing, and we're up for the fight. So in summary, microbes are essential to human health and to all life on Earth. Evidence is accumulating that we're doing harm to microbial communities that is directly affecting the health of humans and other living species on the planet. This harm is occurring on societal levels in the form of pollution of soils, water, and air, industrialized food production, use of biocides in agriculture, widespread antibiotic use. There are currently no protections for microbial ecosystems, and we need to figure out how to develop those protections. Thank you very much.
Thank you all for a wonderful panel. I invite our other two presenters, please, to come up and take a seat, and we will open the floor for questions, both of those in the room as well as those online. Andrew will walk around a microphone.
Thank you, Brother Andrew. So I'm going to wait for the panel to get up there. So everybody good? So first and foremost, thanks again for a wonderful set of presentations. And of course, the one that I enjoyed the most was the last one. And I think that's probably simply because I'm going through senile dementia and I can't remember the first two talks. But I do have 2 points. One, the notion that humans can, quote, harm microbial communities is, I will say, nonsensical. And the reason I say that is because microbes have been here from the very beginning. They will be here forevermore. The idea that we could possibly even cause the extinction of one microbial species is so far out of my brain that it's not even worth talking about. But what we are doing, and the point I think you made really well, is we are diminishing the microbes that help us remain healthy. And that's from the point of view of us as the human species, not damage to the microbial communities. And that I certainly agree with. And one other point that you made about, what is clean water doing to our microbiome? I think the evidence is in on that, and that's really sort of clear. In public health, we recognize that the biggest intervention that has saved more human lives in our history is the development of water management and water purification by orders of magnitude. I was just reading a piece online about global deaths annually. The largest source of death in human populations are children under the age of 5 dying from diarrheal diseases because of exposure to contaminated water. So.
May I respond?
Absolutely, please. I don't know if that microphone is on.
Yeah, thank you for those comments. So on the thing of harming microbes, yes, my argument is more pertaining to on the level of human health rather than the microbes themselves. The funny thing is when I sometimes, like a couple of times when I've presented this to sort of philosophy type audiences who've never really heard much about microbiomes, and I'm telling them for the first time about this, and then a few times, like the response back was like, "How dare you be so anthropocentric in making this about human health?" So I think it really depends on where one's making these arguments and in terms of how they get taken up. I will say that there's a quote which I haven't used by I think it's Tom Curtis, who is a microbiome scientist who would disagree with you, who does think that we have the potential to eradicate species that might be relevant. So I'm not a microbiologist. I'm not going to get into that debate. I'm just looking at what's out there. On the issue of the clean water, so to me, of course, I'm not going to argue the point. Of course, clean water has saved untold lives, and I would never go and say let's stop cleaning our water. I guess what I'm getting at is trade-off considerations. So when we look at, I don't know, say Crohn's, and I know some of this is speculative still, but we do find Crohn's is increasing, and it does seem to have something to do with microbiome. So is there a trade-off situation in terms of antibiotics that have saved many children's lives, but as a result of that antibiotic use, we've changed microbiomes in a way that increases predisposition to Crohn's. By that, I'm not arguing that we should stop antibiotics, in the same way that I'm not arguing we should stop cleaning water. But are there considerations that we haven't had yet in terms of other unintended consequences, other unintended harms that maybe are not as dramatic even, but that we should probably still look at?
So only to respond to what you just said, we have considered those kinds of trade-offs a lot. And in fact, the entire discipline of evolutionary medicine, of which I am one of the founders of the International Society for Evolution Medicine and Public Health, that is the entire focus of what we discuss in that society and in our journal, Evolution Medicine and Public Health. So that conversation has been going on for a long time. Okay.
Yeah, I would agree with you. However, I think we started a little too late. I don't know if you're familiar with Alessio Fasano from Harvard. He's a gastroenterologist, studies the human microbiome, discovered a quote-unquote novel protein called zonulin years ago that closes the gap in leakage in the gut cells lining the intestines. And I happened to be on the same venue with him at NAS some years ago. I don't know why, because we had nothing in common, but thank God I was. And I've listened to a bunch of his YouTube videos, whether he's talking at the Cleveland Clinic or somewhere else. He used to be at Johns Hopkins, now he's at Harvard. And the three things he pointed out that lead to various issues, including childhood obesity and adult obesity, were if you're treated with antibiotics before the age of 4, if you had a non-vaginal birth, and if you didn't breastfeed. And guess who struck out on all three? Yeah, and it's really phenomenal. So there are studies going on just west of me, Fairfax Hospital up in Northern Virginia. They started a study a few years ago with a lot of brave women who said, yeah, I'm having a cesarean, and yeah, you can follow my kid for the next 20 years and we'll see what happens. But his work, again, if you have a rainy day, go on YouTube and listen to some of his lectures he's giving. Assuming he's accurate, it's phenomenal, the amount of impact, and not that I need to be convinced anymore. I think the human microbiome is that frontier that we missed for decades, and its impacts on all kinds of human health. But again, back in the 50s, my dad was a doctor. Oh, you don't need to breastfeed. We got this stuff in a can. It's great. You're going to love it. Oh, you got an earache here. Here's some Oreomycin. Take that. And it's just, we really, I think, not that I was alive for the start of this, but we really missed the boat back in the 50s, I think, and into the early 60s.
I
was just going to add a note on the water management piece too. Like, I do think there could be alternative treatment trains that maybe address some of the microbiome concerns that haven't been explored, because those kind of understandings of the impacts of this sort of like chlorination treatment versus, you know, UV and how that might impact. And I mean, now in the US, like a lot of concerns around premise plumbing and, you know, other types of pathogens that are kind of a concern in our plumbing systems, you know, looking upstream at what are we doing in the treatment plant, what should we do at the household level. But I really appreciate the kind of collective action piece versus this individual level decision making, because I think often, so I'm in the global wash space, and often there is a lot of emphasis on individualized behaviors and thinking about, you know, what can the individual do, when I think you make a really great, you articulated it extremely well about how this kind of collective action is, at the societal level, is what we should maybe be a little more focused on, and that focusing on the individual level decisions, we might be actually hurting the chances of more of these societal level change that we want.
Thanks. Any questions from online?
We do not have a question.
And then the gentleman over here.
Yeah, so I guess this is a question for everyone, and I have several questions. But so David Resnik this morning talked about how bioethics isn't incorporated into like risk governance or regulation necessarily, and I realize that it's often limited by statute. But it looks like the statute says no novel risks, you know, or adverse, unreasonable risk. And so would risks like autonomy or informed consent or cultural risks, would it even be legally feasible to include them? Or is there a standard? Yeah, I guess that's a question for Chris, because I'm sympathetic to your view of the no novel risk between GM versus non-GM. But I am concerned that the public concern, which we'll hear about in this next panel, is much more heightened with GM. And whether that's quote unquote natural science, scientific or not, is kind of, in a way, I don't want to say irrelevant, but it's not the focus of the public, obviously, on the natural science. So is there any way we can incorporate a broader notion of risk into our risk governance system or regulatory system, although I understand the legal constraints? And two, if so, how do we take collective action on interventions in the microbiome if the regulatory decisions are really being made between government and industry? I hope that tied all three talks together. And it's kind of a complicated question, but here we go.
Who wants to go first?
I can take a stab at it. Yeah. I'm not sure with the first question whether you're talking specifically about how they do a risk assessment versus what's unreasonable or reasonable.
No, my question is that your statute says unreasonable risk, right? Or adverse
Unreasonable
event.
adverse effects, but it also says the administrator of the agency will make that determination.
Yeah.
And that's always been a great difficulty. People say, well, give me some examples of unreasonable. It's almost impossible to do. You can't get the lawyers to do it, and for the most part, because they don't want to be pigeonholed. Oh, you said this, and I'm doing that, so mine's not unreasonable. So it really comes down to case by case. But in addition, it changes with time. You saw, with that Furadan is a good example. It took years to move that along and finally get it out the door and done. So what was, perfectly, I don't want to say perfectly okay, but acceptable back in the 80s, 90s, and into the 2000s was good. 2007, it's no longer good. What changed? I can't tell you that there was a mindset change, political pressure from who, NGOs, senators, I don't know. But, thank God somebody was paying attention. But, it's not a hard fast. And so if you want to, kind of restructure, well, how do we think about that?
But I think the more important part of my question is the definition of a risk.
Yeah. Is it always biological?
Is it always have to be a toxicity endpoint or an allergenicity endpoint? Can't it be risk to cultures, risk to autonomy, socioeconomic risk? And is there anything preventing the federal government from interpreting that word
adverse effect or risk more broadly. I think that was the main part.
Yeah, not that I'm aware of. I mean, there is language in FIFRA that talks about socioeconomic analysis. But what I can tell you, at least in the area I was involved with, it was a very rare event that they did any economic analysis, or certainly not social science. So, for example, 2001 was the last, in my opinion, the last major sort of economic risk benefit assessment, and this was focused mainly on BT crops and things like that. And since then, other than if you do a rulemaking, you have to do an analysis, how much money will the affected segment of the population save, et cetera. But to do just, like this almost, in my opinion, it should be almost a day-to-day thing. So I'm gonna register this fungus to kill that insect. What's my benefits analysis? You don't have to do it. It's only if it's like a real problem, like, I'm gonna kill, a bunch of whatever, endangered species. Okay, well, no, that's not gonna work. Benefits aren't gonna help you there. But what about, I gave you this example of Furidan. Thousands of birds dying. And I can't give you a legitimate answer as to how that was ever acceptable.
Any other responses from
And
the
then the
panel?
second part of the question, maybe for Kieran and Megan, is how do we get, I guess, public engagement or a more community approach to defining what a risk is and what we care about with respect to community action, especially risk governance and regulation or oversight?
Okay, so I guess I'll speak on this because I think my role maybe is to use microbiology as a tool to evaluate some of the engineering controls that we're implementing. And so just being squarely pigeonholed into the lab, I am really missing out on some of those other social elements. So an example in some of our other work is that we're evaluating the implementation of several new wastewater systems. And so there's a question about whether or not we can ask or require individual homes to now connect to that wastewater system. And so when you talk about personal autonomy, I think that's what comes to mind. And so it's great to be in this room because now I have a network of social scientists that I can talk to and connect with so that we can start to ask questions ahead of the intervention to figure out what autonomy means to the individuals. And then, as an evaluator, we can collect data before and after. And I think this also goes back to communicating back to the community, is that when we're working in this small community, we're also engaging throughout the process and rollout of this engineering intervention and then reporting back. So I think in that setting, we were funded to do the microbiology, and all of the social science assessments that we're doing are additional and unfunded.
Thank you. So I think that's a great question, and I think there's sort of, on the one hand, a sort of maybe like a more in principle way of addressing it, and another one sort of like a real world politics sort of aspect to it. On the kind of more on the in principle side, so. I do work in deliberative democracy where the main argument is that with a lot of areas of controversial science and technology, the range of perspectives that's considered in making judgments about what's acceptable risk or not is very limited. So even if you have bioethicists involved, collectively the bioethicists still only represent a very narrow range of societal interests. So we do deliberative democratic processes to try and kind of open up that space to get lay input, because after all, everyone is affected by these decisions, and then also to kind of specifically constitute lay publics that represent that kind of diversity. I mean, of course, this is not perfect, this is not panacea, but it is a mechanism whereby you can articulate trade-offs and also have deliberation about where those trade-off lines should be drawn. And then, of course, there's the question, but then there's still the question of how do you actually input that into decision-making processes, right? And so, folks at Australian National University in Canberra, they are working on some of these ideas of deliberative systems, so that you don't just have deliberative mini-publics, but you have them somehow coordinate into larger governance structures. And that could, of course, happen on a kind of like a national political level, but it could also be on an institutional level. So with biobanks, we're talking a lot about using deliberative processes as inputs into institutional policy for biobanks. That's easier when you have like good actors, when you have biobank leaders who actually solicit this and who want to hear about like where do public's values fall on this sort of thing. I think it's a lot more difficult in the context that Chris describes with an EPA that's unpopular and where you also have very strong vested interests going against that. So I think those in principle deliberate democratic solutions are not going to be so effective in that context. I think you need powerful counter advocacy.
With that, last comment, please. We'll have to
just, Jennifer, if you're serious about really pushing this idea of social analysis, economic analysis, the iron may be hot, maybe the time to strike. You're familiar with this American chestnut that they're talking about. Well, there's a good example. That's potentially spread on the eastern half of the U.S. There's a definite risk-benefit analysis that needs to be done. And you could point out in public comments, oh, I happen to know that in biopesticides they don't have a single economist, much less a social scientist. So this is something we need.
We'll do.
Yep. Sounds like our next article. A round of applause, please, for all of our presenters. We are going into our break a little bit, but it is our break time till 3:20. So thank you.
Good questions.
Already here, I'm ready to kick off our section on public perception and engagement with communities. I'll invite our first panelist up here. I'd like to introduce you all to Dr. Christopher Cummings and Dr. Kristen Landreville, senior research scholars at the GES Center at NC State University.
Thank you very much. I'll kick us off. So nice to see everyone. Thank you all for coming. It's an honor and privilege to see you all and be here. I normally work remotely, so it's actually nice to see people in person. There are times where I get to Thursday in a week and realize I haven't actually had like true to life social interaction on a professional level because everything ends up through Zoom or WebEx or phone calls. So today, Kristen and I will be presenting our talk, and will this populate automatically my slides? There we go. Taking the temperature of U.S. public opinion on microbiome engineering. So this is the first shot of what will become a repeated cross-sectional representative survey study sponsored by the Premier Center through our SEI core to, as we say here, take the temperature of public opinion on the topic of microbiome engineering. Taking the temperature was a great idea that Kristen came up with during one of our mini Zoom calls. Today's actually the first day I've met Kristen in person, even though we have been launching all of this work together for some time now. I'm going to share a few slides that will seem incredibly reductionist to you, and the reasoning is because we are also using a lot of the same diction within the survey studies as we actually communicate to the public about microbiome engineering. And so I have also taken these directly from the Premier website, including descriptions like this, an overview of what the center is and does, because we are using similar kinds of diction when we communicate to the public about this particular topic. So we do note that this is a multidisciplinary, multi-institution collaboration that helped to kick off where we're going with the study. And we ended up using very simple kinds of descriptors like, what is a microbiome, right? And we see here, microbiomes are the mostly unseen communities of bacteria, fungi, viruses, and other microorganisms that inhabit a specific environment. Almost everyone on Earth hosts a microbiome. Soils, oceans, airs, building, and even your own body have different and complex microbiomes, and each can be unique. This is actually taken directly from the center's website that is public-facing. You see it's written at a level that is supposed to be accessible for all kinds of folks. And so we use similar kinds of descriptions when we actually launched our larger survey study. We also talked about what is the built environment, very similarly. So the built environment refers to any space built by humans, generally what we refer to as indoors. These spaces include your home, place of work, vehicle, hospital, shopping mall. The average person can spend up to 90% of their life within built environments. And as we've heard a lot of folks talk, that is exactly where we are, especially in many of our quote-unquote Western societies. This survey study is one of the primary outputs that we've had of many other outputs from our SEI core. You see our team here. We have just about everyone here, lots of familiar, friendly faces in this room. The only one we're missing today is Joe because he's on a different continent. And so our team boasts a pretty deep bench, which is great. We have people with all kinds of experience and expertise, and we get to bring that into our group meetings to discuss how should we approach things like what questions to ask of the public. And we get to interrogate this topic from many different theoretical perspectives and disciplinary perspectives. And a lot of that tends to come from our senior leadership's experience in Dr. Kuzma and Dr. Graves, especially with deference and desire to try and cultivate this idea of responsible innovation governance within this ethical framework. And so here we do end up using concepts like bioethics as we try to inform where we're going with things like regulation and governance. And we also have quite a deep experience publishing and working in these areas across many different technology sets. I myself cut my teeth on a National Science Foundation NERT grant studying how the public comes to understand nanoparticles. $1.2 million to say they don't, right? And you'll see here in our study that we're presenting today fairly similar things. What we're going to present to you is a first baseline. Even though it's a public opinion study, What we're really measuring right now is non-attitudes, because we don't have a crystallized understanding on this fairly novel topic, especially among a generalizable kind of public. Here today, we've heard lots of talk about how scientists themselves are so uncertain about what is a microbiome, where is it, how can we change it, could we change it, right? Can you imagine what that conversation is like talking to your grandmother, right? This is all novel and new and distinct. With that, though, we do know that the public brings a ton to the table. All of us do when we come to make sense out of the new, especially out of the different, out of the complex, out of the uncertain. The way that we actually go about this process of sense-making, attitude formation, public opinion formation, stems from other experiences, other backgrounds, other kinds of things that we might call antecedent value predispositions that color or inform the way that we make sense out of the new and the complex. So you'll see in my honeybee hexagons that I really need to beef up the font size on, especially on my tiny screen here, issues like where do people actually get their information from, right? We see this obviously across all kinds of forums, that the type of information and where they get their information likely will inform the way that they feel about certain issues. And now we see a whole new era of confirmation bias in science and technology, where people are self-selecting media, which could certainly change how we can even go about doing something like media campaigning for public engagement. Issues of trust, and I put just trust up here, but there are many forms of trust. And in our study, we are investigating trust in science and scientists, so epistemic trust. We are studying institutional trust, and being able to understand that trust is a multifaceted and complex issue in and of itself that certainly has ties, especially to public will and desires about where microbiome engineering will go. And we'll be hearing some of the data from Kristen. Other things here that we have, science and technology beliefs. These would be antecedent value predispositions about how people feel about technology in general, and how do they feel about uses of technology for solving problems in society. There are many ways to address certain needs in society. Some of those may be better off without particular technological intervention. And so we want to be able to capture and understand how the public feels about that. Thank you for making my screen bigger, whoever did that. We also look at ethics and values, worldviews, so what kinds of things lean people towards particular ways in which they view the world. Are they more individualistic or collective? Are they more hierarchical or more individualistic? And what's the fourth one, Jennifer? I'm drawing a blank. Fatalist. Yeah, fatalist, thank you. And then, of course, we can get into all of the kind of census-y demographics, personal characteristics, all of the check mark boxes that many people have been saying we need to get away from during our conversations today. But we still can use those to do things like be able to say our study is representative of the public when we're talking about public opinion sampling. And so we look at a lot of those. We also used a lot of the types of questions that were informed from the soon-to-be Dr. Hardwick's work, Andrew, who's not in the room right now, which is even better. I can keep talking about how good they're doing. And Andrew led us on a systematic review of the literature of looking at LC issues regarding microbiome engineering in the built environment. That particular publication is fresh out. Speak of the devil, we're talking about you. And this particular work led to a lot of the creation of the types of questions that we were interested in asking by what was in the literature and also what wasn't in the literature when we did this systematic review. And we did apply a bioethics framework, and we're now learning from you all that there may be other better avenues since we've had some wonderful discussion in this area. But we note that from this first take systematic review of the literature in this area, that it's kind of a wash. There's not a consensus built here, right? There are people talking about individual goods and collective goods and autonomy and justice, and it is almost uniformly spread out, which leads us to understand that this is a complex issue that brings together all of these areas that we need to explore further. And so we wanted to be able to capture a lot of these kinds of issues that cropped up, both from our applying these labels on top of major themes that we saw in these papers, but also capturing a lot of the nuance and things that we saw of emerging issues that might crop up, like things like privacy, informed consent, governance, ownership, and the like. So with that, I do want to hand it over to Dr. Landreville, who is going to walk us through a very first take of our data. And this is of the entire sample of 1,000 respondents. And it is univariate statistics. Where we go from here is about 15 papers that you will get all of the hierarchical ordinary least squares regressions and experimental quasi designs and all of that stuff later. This is your first take of the temperature.
Thank you so much, Chris. Yeah, so like he said, this is just a first look, so I'll be presenting some descriptive statistics, nothing that's predictive yet. You can see here we have some demographic data, respondent characteristics on the screen. I won't list them all to you here, but it is a representative survey of 1,000 U.S. adults. We've recruited from YouGov's National Omnibus Panel during the first 2 weeks of December of last year. And these demographics generally represent U.S. Census data. So I will move on here to the next slide. Okay, so moving on to the results. When we fielded the survey, what we did was present the respondents a paragraph that defined the microbiome and defined the built environment, just like Chris said. So they had a paragraph that they read, and then they answered these questions. And in terms of knowledge, not surprisingly, we see that respondents generally did not feel like they had a lot of knowledge about this topic. So only 21% felt knowledgeable about the research and developments in microbiome engineering for improving indoor spaces. In terms of information seeking, we see even lower that only 14% of participants had actually actively sought out information or education about microbiome engineering in the built environment. Yet given that low knowledge and low information seeking, we still see that people have hope and promise in terms of what these technologies could do to improve the indoor built environment. We see that just under half of people felt that, that they had hope about these technologies. Again, like that Chris alluded to, there's a lot of non-attitudes and uncertainty. We have 38% of people just not really sure, and then only 13% didn't have hope. And then we see on the other question there about wanting to learn more. This is great news, I think, that a lot of people, so 64% reported wanting to learn more about these types of technologies, with just 21% unsure, and 15% didn't want to learn anything. Now, moving on to asking questions about how comfortable people felt with microbiome engineering in different contexts, we see that the closer we are to home on the, I think a laser pointer? Maybe I don't, okay. So you see over here that the home, you see the most uncomfortableness with seeing a comfort with, sorry, I messed that up, but introduce microbiomes to combat harmful microbes. So we asked people questions about introduced microbiomes. We didn't say if they were genetically modified yet or naturally occurring. We just said introduced microbiomes to combat harmful microbes. And you can see here, houses in your own home, they're not so sure. But if you go down to the other side, you see that research labs, they're like, yeah, okay, I feel more comfortable with these technologies being used in research labs. And then it's kind of interesting, I think the further away from the home you get, the more unfamiliar people are with those spaces, the more comfortable people feel with those technologies. And then we have two kind of case studies that we delved into with specific contexts. And so this first context I'll present on is hospitals. And so we posed questions to people about how comfortable they would be with introducing microbiomes into hospital sinks in order to combat microbial resistance. And you can see here that 46% of people thought that that technology held promise, 37% not sure, 17% don't think it's very promising. And then in terms of confidence, though, we do see a difference, and I want to point that out. So holding promise is different than being confident that these technologies are actually going to work. And so a lot fewer people are confident. So we see just over a third of people are confident that using introduced microbiomes in hospital sinks are going to contribute to a safer healthcare environment, with a lot of people not sure at 40%. Now going to the next context, we asked people questions about HVAC systems. So believing that HVAC systems with harmful microbe sensing capabilities is a promising approach to improve indoor air quality. You can see that a lot of people are hopeful that that technology can work out, 61%. Again, almost 30% of people are not sure. Moving on to confidence, just like the hospital sinks, quite similar numbers here. Being confident that these technologies are going to make the indoor environment safer, we have 37% being confident and a lot of people unsure. So moving forward here, what do people desire and want? We see that 69% of respondents agreed that government agencies should provide educational resources to raise awareness about these types of technologies going on. And communication and engagement. So 76% of respondents really would like to see public input, community input as these policies that kind of guide this technology are being created. In terms of government involvement, we see that government involvement is valued and that government regulation is wanted by most respondents. So just over half of people wanted to see government involvement to ensure the ethical and responsible practices in microbiome engineering. And in terms of regulation, 72% agreed that there should be clear government regulations regarding the use of microbiome engineering in indoor spaces, which is quite high. So up until now, again, the questions had not addressed any differences between GE and then natural microbiomes. We just posed questions as introduced microbiomes. But then once we gave them a description of genetically engineered microbiomes, we asked them how open they would be to using these, if the benefits are well documented and the risks are carefully managed, and just over half. So 54% of people are open to GE microbiomes. A lot of people not sure, at almost 30%. And then people prefer the natural. So when you ask them in head-to-head, do you prefer naturally occurring or do you prefer genetically engineered? 62% of respondents are more comfortable with naturally occurring microbiomes in indoor spaces than those GE microbiomes. And there is a psychological bias in many people that's kind of a naturalistic bias, that people prefer natural as a label as opposed to anything GE. Now, we also asked people a gamut of different concerns about risks, and so these are just the top 5, but we do have more that we asked about. You can see here that regulatory concerns are up there at 71%, so being concerned that a potential misuse or abuse of microbiome engineering if there's not adequate regulations. We have concerns about those engineered risks, so being concerned that GE microbiomes may have unintended consequences when used indoors. And then privacy at 58%, so being concerned about invasions of privacy with these types of technologies. And then we have unintentional harm, so being concerned that these technologies will cause unintentional harm somehow. And then justice issues at 47%, so worrying that the distribution of benefits and costs in microbiome engineering won't be equally distributed and fair. So moving forward, again, those are just some basic descriptive statistics. Moving forward, some kind of take-home points are engaging with the public early and often is important and crucial for responsibly shaping research agendas and fostering innovation in these technologies. Integrating societal needs and ethics, so applying a strategy of responsible research and innovation can help incorporate these societal needs and ethical considerations moving forward. And then strengthening governance. So there was, pretty good support for valuing valuing government input, government regulation, and that can build public trust and can foster that dialogue. And so, those are our major take-home points here, and thank you for listening to the Taking the Temperature. Great. Thank you so much, Chris and Kristen. I'm excited to introduce our next panelist, Dr. Erika Szymanski, Associate Professor in the Department of English and Affiliate Faculty in Cell and Molecular Biology and Microbiome Initiative Hire at Colorado State University.
So this happened to be here, which is handy, because usually my bag has a ton of whiteboard markers in it, but it doesn't right now. I know I'm supposed to stand next to the microphone, but humor me for a second. So we had this lovely moment this morning where Rob started us off with baselines and baseline healthy. Oh, hey, thanks. Much better whiteboard marker. Usually I have scads. Where Rob started us off with this idea of baselines. And we've been talking a lot about baselines all day and about drawing lines between what is and isn't healthy and such. And I am a theorist. I get to play in the land of ideas. I don't have to convince policymakers at the end of the day. But the ideas that I've been having about lines lately are that we need to draw lots of them and move them sideways and focus on connections and the edges instead of the nodes, and stop worrying about quite so much what the baseline of what healthy is and start asking what kinds of worlds are we trying to construct through the relationships that we're forming. So somehow we've ended up in this interesting place at the end of the day where we've had a lot of conversations revolving around how we decide whether the changes we want to make now are or aren't okay in light of the variety of other changes we've already made. So we're doing this. Oh, no, definitely not. Oh, hey, but there's another one.
Perennial problem in our classrooms, by the way. Here's another one. I'm sorry. And another one.
Okay. So we're sort of trying to investigate whether the line that we're drawing between what happened before premiere and what happened after premiere is a reasonable line to draw in terms of regulating, I think, in terms of sort of distinguishing the things that we have been doing to our built environment microbiomes, as has been pointed out numerous times. We've been changing things without worrying about them or regulating them. And then asking, so how do we account for all the stuff that happens now that we are aware that we're doing these changes, or at least that we're thinking about some of them, with respect to where we draw that line, with the things that happened before, the things that happen now being different. I don't think that that's a particularly productive conversation to be having. I want to ask, what do good environments look like? So that's why my talk is more about how a microbiome is, rather than what a microbiome is. I'm going to try to avoid making a bunch of obnoxious puns, because I'm going to talk about discourse as a construction tool, which poses some particular challenges when we're talking about built environments. I want to ask how we are building the environments of microbiomes, not just how we're studying the microbiomes of built environments. Because the theoretical position that I start with when I'm thinking about science communication sits under what we would call in my home discipline of STS material semiotics. This notion that we construct what things are in practice, and that our lived experience of the world and our knowledge above the world are constructed through what we do with the things in it. And also under critical discourse studies, which is a concept that I think gets some traction in STS and certainly does in Rhetoric of Science, where I also sit, where we understand texts as social spaces where relationships are played out. So practices in this case are material practices, physical practices, and discursive or language practices. I don't think about those as two separate groups, because language practices are always embodied and material practices are always relational and therefore semiotic, but also because practices that we conventionally primarily think of as being either material or semiotic are iterative. They shape one another. Language practices shape the physical things that we do and vice versa. So the particularities of how we use language to handle scientific objects among scientists and in broader conversations, which is also a line I'd like to screw up, those matter to what scientific phenomena become and to how communities are constructed in interaction with them. So my main point today is about how language matters to studying the microbiome of the built environment and, as I mentioned, to building the environments of microbiomes, considering what kinds of environments are desirable to build for multi-species getting along, and about how that work needs to involve building discursive environments that supply the visions through which physical environments come into being, and that enact particular versions or visions of community. So I realize it's been a really long day. If you remember one thing from it, please let it be that an English professor is really excited about the things that can happen in a center like this one. I am an English professor. That's also probably not a very helpful discussion or a very helpful introduction. So I'm I primarily think of myself as an STS person, and this is how I think about STS mostly, is investigating how scientific knowledge, technologies, and innovations are constructed and how they act and interact in society, and therefore the cultural and other dimensions of science and technology. But I'm also a rhetorician of science, and that means that I'm really interested in how rhetoric, how language and practice participates in constructing biotechnologies in terms of reshaping working relationships among microbes and humans. So the thing that's really unusual about our microbiome cluster at Colorado State is that we're one of the few in the country that's not primarily interested in the gut microbiome or in human health. We're studying agricultural microbiomes, industry microbiomes. To be honest, we're doing a lot less with microbiomes of the built environment, although Susan DeLong would tell me that I'm wrong. So all of those various working relationships that we're changing, I think, don't get enough attention when we're primarily focused on questions that have to do with what humans are and how we determine what humans are healthy. Humans are boring. We already have a lot of the conceptual questions figured out there. I think one of the things that I'm really excited about when we talk about agricultural, industrial, and built environment microbiomes is being able to ask what kinds of relationships are good to build without necessarily having a lot of established frameworks for what answers we should be providing to those questions. I do primarily study rhetoric of science among scientists. I'm interested in how language functions as a scientific tool for doing science. But I also think that it's illogical and unhelpful to draw sharp distinctions between science communication used by scientists to do science and science communication involving broader audiences. So I think the whole spectrum of language practices that we use to handle science are involved in what constructing science becomes and what science becomes for whom. And of course, choices exist in how scientists communicate about scientific phenomena, and choices always involve values. So in addition to functional overlaps among conversations, among scientific and stakeholder and broader discourses, scientific language is always already shaping the conditions of possibility for socio-technical institutions. It's always making choices that build politics into technology and vice versa. All of which is why I earnestly think that social scientists and humanists like me should be studying how scientists use language and not just how we use language for popular or public-oriented conversations. For what it's worth, I also came into STS by way of microbiology, and I still think that microbes are the coolest thing. So take that for what it's worth. The value-latedness of discursive choices is what I want to share about today. The project that I want to share with you today is primarily about this connection. So Microbiome-ish is an NSF career award that's centrally about words and relationships, about possibilities for feminist and multi-species approaches to scientific language that emphasize how things become together in relation to each other over what they are as discrete objects. So I'm primarily responsible for the work I'm sharing with you today, but Marie Turner, Cookie Egret, Kieran Kuzmiski, and Trevor Stewart have all been really important to the way that I've thought about microbiomes in general. And the final two there, Kieran and Trevor, are two phenomenal undergraduate researchers. So I love my undergrads. Microbiome is also more specifically about what kinds of things microbial communities are, what they're becoming through the metaphors that we apply to them. The central question of this project is what kinds of communities are microbial communities? Or in other words, what's microbiome-ish? Hence the project title. What are microbiomes like? This is a rhetoric of science question because metaphors organize physical practices. Studying microbiomes isn't just a matter of going out and discovering things. It's about asking questions about what microbiomes might be so that we can make observations about them. And those observations are always interactive. They're always relational. They're always about the meeting place between how microbes affect the world and what we humans can do to perceive them. So they're about these overlaps between our umwelts, or our shared capacities to listen and respond, which is one reason that I've started thinking about responsible research in terms of response-able research, riffing on Donna Haraway's term responsibility to describe how humans and other creatures become with each other. How do we listen and respond? And that involves how we make data about microbes insofar as we're, you could say, listening to the capacities of the things that they're doing that we have the ability to listen to. So the kinds of questions that we're asking of them, the kinds of questions we're asking and how they're asked, the language we use to ask them, matter to how microbiomes are brought into being in human worlds as things that we perceive and know about and can work with. And I think it's really easy to see how the tools that are available for studying microbial communities matter when we're talking about physical tools. We're all here because metagenomic sequencing brings microbiomes into human worlds in different ways than culture-based microbiology does. And I found that computational biologists are often really sensitive to how the conceptual frameworks that they're bringing to the models that they're building make a difference to which microbes they see and how. So I have some lovely conversations with and publications with computational biologists. I know the center's involved in really similar work. So these two papers say a little bit more about how I think discursive choices are built into the way that we're using physical tools as well. But of course, discourse tools on their own, language choices, are central to scientific toolkits for asking questions, for interpreting results, for making knowledge. So it's essential, I think, that interdisciplinary research teams be asking how the language they're using matters to how science gets done, especially since microbial communities usually have to be destroyed to be studied. So you could say that, that metaphors are kind of like the image on the top of the jigsaw puzzle box. We have to imagine what we're looking at to be able to put pieces together. They're also very much not like jigsaw puzzles because there's never one right answer. Language is never neutral. There's no one correct way to represent some kind of underlying fixed reality out here. Knowledge making is always relational. Your perspective always matters. So no one can say that their perspective doesn't matter. Language is never a view from nowhere. It's always about specific humans making choices in context. So discursive choices, knowledge-making choices, language tools are a key way that values shape scientific knowledge. I really want to emphasize that when I'm talking about studying metaphors for microbiomes, I'm not looking for the best metaphor, or I'm not trying to call some metaphors bad or wrong. I'm interested in making metaphors visible as choices that we make that matter to the results that we're coming up with and to how we understand the roles of humans with respect to microbes, to how we understand what we can do with them, so that we can recognize and use those choices deliberately, and so that we can value how a plurality of metaphors makes it easier to see and to negotiate how we're bringing different values to the work that we're doing. So I've been harping on metaphors a lot. Metaphors are especially important as far as discourse tools go because they embed analogical reasoning in language. And analogical reasoning is always partial. Two things are always alike in some ways, but not in others. Rhetoricians like to talk about chairs because there's always a chair around. When I walk into a new room, I recognize some things as being chairs, even though I've never seen these chairs before. But I can analogize them to chairs I've seen in the past, and then I can see how we're all enacting them as chairs by sitting on them and acting like they're chairs. So metaphors foreground how some things are similar. They foreground some elements of what something is, and they downplay others. We can even say that all language is metaphorical, as I just did with the chairs. So, again, I don't think it's terribly productive to draw lines between what's metaphorical and what's literal, because, well, trying to draw lines around who gets to decide what something really is always a problematic notion, and especially so when we're dealing with something that's as fuzzily known as the microbiome, as we've been talking about all day. So the example of how particular metaphors that are brought into science matters that I always hope everyone can recognize is the genetic code. Thinking about DNA and RNA in terms of, first, human language codes, and then programmable computer codes through the slippage of that source domain, has enabled building just an incredible array of biotechnologies that rely on reading and writing and editing, and now refactoring and debugging, and so on and so forth, and otherwise parsing one-dimensional strings of letters in ways that we very much treat algorithmically as human language text, which matters for things like what we don't know about the three-dimensional structure of RNA right now. And I think arguably the reason why we don't have AlphaFold for RNA has a lot to do with the strategies that we've employed for DNA that then get translated over to RNA, because RNA is a mere messenger that otherwise is just like DNA, and so we string it out into a one-dimensional string, we lose three-dimensional structure, we don't think that solving three-dimensional crystal structures is necessary, and then we don't have a training set to build, well, AlphaFold for RNA on. So I can't entirely blame the metaphor, but I think there's something there. Even more deeply and more relevant to this conversation today, molecular biology is built around analogizing molecules to human actors in ways that extend from Western white worldviews that are fundamentally hierarchical, where one molecule or other biological actor controls another. So Josh Evans, Emma Froh, and I have a short piece in this year's issue of Grow magazine on that topic that says a little bit more about alternatives to control and why we think that they're important. I've been trying to learn about ecology as a metaphor insofar as nearly all of the microbiologists I meet call microbial ecologies just ecologies. And every time I see someone reference a system that's been built for one purpose, applied to another purpose, as just the way things are. I want to know what assumptions are built into that framework. The reason why I feel like I need to better understand the work that Ecology is doing is because it's ramified through how, for example, we understand ecology is a concept that assumes boundedness, spatial boundedness, so that populations are either native or invaders or colonists, temporal boundedness, such that organisms are either persistent or transient, so that we focus attention on things like barriers to establishment and invasiveness, where we're applying value judgments to which creatures do and don't belong somewhere in ways that have, I think, finally been getting some good attention. So I'm interested in metaphors for microbiomes because these language tools perform analogical work that pattern how microbial communities are imagined and understood, what we think we can do with them. And they're value-laden analogies that inform how we think about how microbial communities hold together and how we think about humans as communities with them. So in this project I mentioned, we've started cataloging metaphors for microbiomes in the peer-reviewed literature. We started with the peer-reviewed literature, again, because I think that the way that microbiomes are being enacted in scientific discourse itself is important to what we think they are and therefore how we should relate with them. Some of these metaphors are explicit. Things like macroecologies. Scientists explicitly name and recognize them. Microeconomics is also a really good example of an explicit metaphor. There are a set of papers that recommend that we think about microecologies in terms of microeconomics. And some of them are implicit. They're characterized by choices of phrase and methodological approach, but without ever being explicitly named. So in those cases, we've had to come up with a name for them ourselves. Buckets of species is a good example of an implicit metaphor. So is the metaphor, microbiomes are like Italian city-states, which is a really good example of how I've had to come up with some titles for metaphors, for patterns that I see in the literature that aren't necessarily ideal. So if anyone has a better way to describe the pattern of analogizing species and microbial communities to groups that live in proximity to each other in an uneasy coexistence that's principally characterized by land grabbing and trying to monopolize resources, where actions that look like collaboration are always invariably ultimately about trying to land grab and monopolize resources. If you have a better way to describe that, please let me know. I've already said a little bit about macroecology, so I'll briefly say a little bit about just two more of the metaphors that I've mentioned as contrast cases to help describe why we think that the differences among them are significant. Microbiomes, or buckets of species, is probably the least differentiated metaphor in our catalog. We never see this precise phrase, though bucketing is sometimes used to describe a step in sequence analysis. But the idea that the microbiome is a bucket of species or of phyla or other taxa is implicitly characterizing a lot of the who's there studies. And it's sometimes just a holding pattern for more detailed studies to come. But buckets are also a set of functional assumptions about microbial communities' lack of internal structure. And they usually relate to equating small sections of DNA with microbial presences. So that equation matters because bits of DNA employed to identify species don't necessarily tell you anything about what microbes might be doing because of genomic fluidity and environmental interaction. We've already worked through that today. In another register, that equation of sequence and identity perpetuates the idea that communities are characterized by their genetic complement, which becomes really problematic if we're trying to use microbes and humans as models for each other. So we also see a lot of human microeconomics in public goods games, which focus attention on individual microbial cells. Here, environment is resolved not as a bucket or a set of niches, but as neighborhoods wherein cells have opportunities to trade locally produced resources, so spatial and temporal relations matter on a cell-to-cell level. Microbial cells are either producers or consumers, they're fair dealers or they're cheats, they're dependent on their neighbors or they're independent from them, so cells are defined according to their market position, and microeconomics kind of assumes that other creatures do economics the way that a small subset of humans does economics, which is interesting. There are a bunch of things I'm not going to take time for because I took time to draw on the board instead. So ultimately, we have a lot of metaphors, and I could talk more about them, but I'm not going to. Ultimately, I want to ask the same question of science communication with broader audiences that we've been asking of science communication with scientists, which is, what kinds of choices are we making about the way we're imagining what communities look like, and why? And how do we think not only about the way that our choices matter to microbial communities, but to how multi-species communities live together, which I think is ultimately the question at the center of this center. I've talked about discourse as a construction tool. I've talked a little bit about turning the line sideways so that instead of thinking about the lines we draw around creatures that do and don't matter, we ask how do we attend to the lines we draw between creatures and to sustaining conversations that allow us then to ask what responsible research looks like when we're asking about listening and responding to other creatures. There are a lot of interesting questions there with respect to how we do sensing in built environments that I want to ask y'all. But I like the way that that question refocuses our attention not on how I am responsible to some imagined person out there, but how we generate recursive systems that embed capacities to feed information back to each other, where I think about what I'm learning from the microbes, because microbes have something to say. So from where I sit, those are the interesting questions. The last thing I want to mention is that response-able research is a key principle in some collaborative research that I'm doing with these fine people, Jane Calvert, Rob Smith, and Koichi Mikami, primarily around whole organism synthetic biology, but really the questions we're asking are how do we do multi-species science policy? And there, the main questions I'm interested in are how do we come up with alternatives to that sense of control, that assumption that one thing controls another in a very hierarchical way that's really built into the center of biology. DNA makes RNA makes protein. No, it doesn't, and yet we keep saying that. How weird is that? So one idea we've come up with so far for thinking about a better way or a different way to understand what kinds of relationships we might be trying to build with other creatures is through living rooms. I have no time to say anything about that, but please ask me about it later. Thanks.
Thank you so much, Erika. I'm pleased to oh my gosh, it's you back there doing the changing. I'm pleased to announce our final panelist for this session, Dr. Pajau Vangay, Senior Director of the Scientific Network Hypothesis Fund. Let's introduce Dr. Pajau. Thank you so much.
Hi, everybody. Last speaker, so bear with me. Really appreciate it. Thanks to the organizers for having me here. I'm really excited. And you might be wondering why I'm presenting on this work, being at a philanthropy organization, but this is actually my PhD research, and I love talking about it. So, all right. So I'm going to just probably skip through these pretty quickly because I don't need to tell everyone here that the human gut microbiome is obviously shaped by the environment that we grow up in. Several of you here already mentioned that, living in Western society actually is very correlated with higher incidences of chronic diseases. Among immigrants, we know that health deteriorates after moving to the US. And we also know that the gut microbiome and the immune system have a very intimate relationship. So first couple of years of life, your microbes are talking to your immune system, really getting primed, and it's really critical. So given all of this information, our hypothesis was really formed on wanting to understand if changes in the gut during migration was responsible for some of the chronic diseases that we were seeing in the US. So I like showing this epi graph that I took from this paper on the bottom here, but this healthy immigrant effect really is, again, just explaining the point I made earlier about folks moving here and essentially getting sicker, getting heavier, and developing the chronic diseases. One might think that it's associated with eating more burgers and fries, but the community is actually saying that that's not what's happening. Most people are moving here and still sticking with their cultural foods, not switching over to American diet right away. So that's really what we wanted to understand. I look so young in this photo. So I wanted to highlight this partnership because it was very serendipitous, but also really special. So Dan Knights is my advisor. Kathy is a local clinician in the area. She also co-founded a nonprofit that works with communities, partnering with local communities on social determinants of health. For the reason, I happened to meet Kathy at a small conference, and the rest is history, really. So we partnered in that way. Dan's a computational biologist, so doesn't have a lot of experience in this arm of research, but then he was very, very open to letting me run with this project. And really, Kathy's team has this great experience in community-based participatory research. So for those of you who haven't heard of this method, it is more used in, sorry, some of the formatting didn't turn out quite right, but it is much more commonly used in some of the social sciences, and essentially it means that you partner with the communities that you're working with from the very beginning. So from defining the problem to actually implementing it, and then of course disseminating what you find. We had actually early discussions with a variety of communities in Minnesota. If you don't know, Minnesota actually has the highest number of refugees per capita in the U.S. And so we spoke with, Somali refugees, Hmong, Karen, and in the end, based on a number of different feasibility constraints, we ended up working with the Karen and Hmong. So these are two minority ethnic groups from Southeast Asia. They're from slightly different places. So the Hmong are from Laos, ended up in Thailand in refugee camps there, and then migrated over to the U.S. around the 1970s, 1980s. They actually helped serve, partnered with the Americans during the war. And then the Karen also had a lot of persecution in Myanmar, ended up on the border of Thailand, also in refugee camps, and the refugee camps are actually still open today. And so we worked very closely with these two communities to define a problem. Obviously, we came from the perspective of caring about the gut microbiome and wanting to understand chronic diseases. Obviously, these two communities didn't know much about the gut microbiome, but I think they were very open to learning about it and exploring this question of whether it might be explaining. I think both communities noticed that they were getting heavier after moving here, and they were developing diseases, and they didn't understand why. Again, pointing to the fact that it wasn't just diet. We also wrote the grant proposals together, and then after receiving funding, we developed an MOU so that we had a process for making decisions. So it was a very egalitarian process. And we put together a multidisciplinary team. So our CBPR experts are Kathy, Shannon, and Rodolfo. We also recruited community researchers from the Hmong and Karen community as well. These are leaders within their community who are champions of health, but then they are also really well connected. And so that contributed immensely to the success of the project. We also had community advisory boards, so these are kind of at-large members of these two communities who can voice the concerns of the community. And then, of course, myself, Dan, and a med student. I put myself over here as a community researcher as well because I'm actually Hmong American. That really informed the decision behind going after this project. So now lessons learned from this phase of the project. Definitely engage with the community before starting any project. This was a problem they cared about. So as a result, we were able to successfully find people to partner with us. And you'll see as I carry on, it'll lead to the success of the project. Also really important to develop a collaborative agreement so that you have a process for decision making. And then the implementation stage of the project, which is really focused on data collection and recruitment. So I just wanted to quickly share the study design. We ended up having over 500 people in the study. I will have to say that when we started, we had a really low expectation of only 40. It was meant to be a pilot study. But it eventually snowballed. There was a lot of interest from the community, and we were just lucky to get additional funding. Generally, we collected stool samples from folks that were still in Thailand, those who were born in Asia but then moved to the U.S., and then second generation, so those whose parents were born over there and they were born in the U.S. You'll see that we don't have any Karen who were born in the U.S. because their time frames are slightly different. So the Hmong started arriving about 1970s, Karen started arriving only about 5 years prior. And, this picture is really meant to convey that this was a very grassroots approach. This is not a fancy multi-site clinical study. Essentially, it was me cold calling a bunch of local organizations to see if I can table at their site, working with my community researchers to really go out there and put ourselves out there. And I put a star here by word of mouth because that was actually by far our most successful recruitment tool. It's really developing trust with the community and that snowballing and them referring friends and family because of that. Now, I also went to Thailand. Obviously, the refugee camps that the Hmong were in have since closed, and so we couldn't go back there. But we went to a very rural village just an hour outside of Chiang Mai and worked with community researchers there and collected stool samples there. And then we also were able to gain access to the refugee camp that were the Karen refugee camp, and were able to work with community researchers there and collected stool samples there as well. So for this stage, definitely important to respect that refugees have historical experiences and concerns about research. the thing I want to point out here is that the Hmong actually had some negative experiences in the past, which made them a lot more hesitant to want to work with us. Despite being Hmong, that didn't matter. It definitely allowed me to communicate from a language perspective, but you still had to spend the time building the relationship and really ensure that, you know, we were trying to actually do something good. For the Karen, it was actually quite different. The refugee camp I mentioned actually had a really large Malaya research unit within it. So they were exposed to research early on, had really positive experiences, so we had no problem recruiting there. And then actually I told someone earlier that our IRB committee probably hated us because we were constantly learning from our community as we were recruiting. So we came up with methods for recruitment, but then realized, like, no, actually everybody wanted to do group consent instead because they wanted to discuss things with each other. Or maybe they wanted to bring their partner and have them sit in the room. And so, or maybe they wanted us to go to their home. And so we were constantly amending things and changing all of that. But at the end, definitely developed cultural linguistically informed consent processes with them. As you can imagine, for nearly every single person who participated, we were communicating in their language. And a couple of modifications that came about from actually co-learning alongside our community is that we ended up focusing our study only on females. This was because our advisory board had informed us that they believed that the majority of Hmong who were coming over were women. And so we ended up just at the beginning limiting it to that. We also had thought about a dietary intervention, but we had major pushback, and so we ended up taking that out and just doing a longitudinal sub-study where we followed folks who arrived for 6 months. And what this actually looked like were, you know, basically in-depth one-on-one conversations with our community researchers. They would get a sense of their, you know, migration history, understanding what they ate by using these food models, and then eventually we would have to manually enter all of that data into, you know, a software process. It's quite different if any of you have done dietary recalls. Usually you just send your participant off with like a website and they do it themselves, but this was the additional steps that we had to put in. You could imagine that with groups from Southeast Asia, the foods may not necessarily be found in the USDA databases as well. So what was kind of a good thing was that the USDA database is actually hierarchically coded. I put an example here so you could see that like milk products are all lumped together. For that reason, it's not perfect, but we were able to construct a tree, kind of similar to a phylogeny, I suppose, but it's not perfect, and representing the database of foods. And we were able to account for culture foods that we couldn't quite identify or we didn't know what it was exactly, but we had to manually do extensive research to figure out what it was. So maybe we don't know what the genus and species of this fern is, but we at least know it's a vegetable, probably has some fiber, and just at least put it in the right spot in the tree. This matters because we had over 200 foods that were not found in the USDA database and over 70 foods that were not found anywhere. So we had just to do lit review to try to do some research on what they were. And of course that's important because if you're looking for dietary associations with the microbiome and half your foods are missing, you're not going to find much. So for this phase, definitely important to have shared decision-making. This is really related to that collaborative agreement, encouraging co-learning between the two, the community and the academicians, as I mentioned about our study design changes, and acknowledging that, you know, being very sensitive to the fact that, you know, just because somebody changed what they ate doesn't mean that they're a bad person or that they made the wrong decision. So we're very sensitive to that. And then lastly, I guess the most fun part is really dissemination, sharing the results. So we went kind of over and beyond in terms of doing that. So the science findings, I'm not going to talk too much about it. You could read the paper if you're interested, but just a really quick summary. We did find that there were drastic changes in the gut microbiome really quickly upon coming to the US. For the folks that we followed for the first 6 months of being here, they essentially started losing strains of Prevotella, which is commonly found in underdeveloped countries, immediately. And these are also really important for degrading fibers. We saw that there was a huge loss of diversity over time in the US, which is also associated with obesity. And then one would think that diet matters, but it didn't explain the total variation that we're seeing. This really alludes to the importance of why we're here today, because that means that something else was happening in the environment. This also corroborated what the community told us at the beginning, that essentially, they weren't changing their diets. I'm going to skip over that because I'm running out of time. And so then in terms of communicating out what we learned, so if you remember, the Kieran had only been in the US for a couple of years. It made no sense for us to go and bombard them about gut microbiome information, so we really needed to meet them where they were. They had actually difficulty navigating things like the grocery store, how to use an American kitchen, how to read a nutrition label. So we partnered with the University of Minnesota Extension to actually offer those classes and workshops, and then we snuck in a bunch of gut microbiome kind of training in the middle. For the Hmong community, it was a little different. They've been here for like 30 years plus, and so it was even harder to get them to come because they're like, I don't care. I have other things to do. And so this is really where we were trying to remind them that the main thing that we found was that just because you move here doesn't mean you should rush to acclimate and become American. The reality is that you're healthier back there, and try to maintain some of the cultural things you were doing. So I show a picture here of a fermented mustard grain that is a traditional food that we make in our community. And we had a workshop focused on learning how to make that again, especially for younger folks that don't really know how to do it. We partnered with the local Hmong chef as well as the Hmong Farmers Association for that. And it got some coverage in the local newspaper. We also put together an animated video narrated on all 3 languages, which really simplified what we found. Essentially, it's not going to play here, but just wanted to mention that, and you could visit the URLs if you're interested. And I just wanted to say that we published the science results. We were forced to do the community methods in a different journal because the science journal didn't want it. And then we also did a whole bunch of outreach, and I wish that all of that could have been done in one place, but so be it. And I think the main thing that I just wanted to end this with is that our community realized that we weren't going to necessarily have actions for them right after. I think the important part is that we established a relationship with them. And I'm proud to say that Dan is still working with the Hmong community on a gout project now, because there's a high incidence of gout there. And they're following up with some animal experiments to understand things like water and antibiotics and other things that's been happening with these samples. Massive collaborations. And I just wanted to leave this here, and it'll be in the PDF for anyone. Shannon is one of the co-founders of Solamo that we worked with. If any of you are interested in CBPAR, they have free resources as well as partnerships that they do with external collaborators. So thank you so much.
Great. We'll kick it off with any questions from the live audience.
Yeah, I think it was Kristen mentioned privacy concerns with the microbiomes, and you kind of lost me there. Could you expound on that a little bit, please?
Yeah, sure. So on the survey, we asked different questions about their concerns about privacy in terms of invasion of privacy, like if their data in the microbe sensing HVAC system, for example, like if someone's entering a building and there's an HVAC system that's sensing microbiomes, does that individual's microbiome, is that private information, or is that something that is public now that you've entered a public building, for example, where the HVAC system might be? So those were some privacy issues. Do you have anything else to add to that?
Not much further, just that there are pretty widespread concerns just about biosurveillance in general as an invasion of privacy.
Yeah, kind of like a HIPAA thing, or no?
I don't think we went that far in the survey, and we probably wouldn't get to that in the quantitative scale measures that we have, but I imagine that you would if we follow on from this with other forms of interviewing. But that would be my personal thought, is that yes, there would probably be people who are concerned about that kind of level of, violation of their medical data or personal data.
Thanks.
My first question is really a comment for Kristen, and I was hoping that slide 13 of your presentation, if you would be so kind as to give that to the NSF, so that they will fund us to do that kind of work. That would be great.
Which one was that?
That was the one to do with educating the public about microbiomes.
Sure, yeah.
So I was like, absolutely, talk to program officers, make them give us money. The second thing was for Pajau about the diet. So when you say that they weren't changing their diet, this is one of the things that I found intriguing when I came to the States. So I eat chicken in Ireland, right? And I ate chicken in America. And chicken in America is corn-fed and very different. And that's all I wanted to say about that. But the diet might be the same, but the actual diet is different. And I wondered if you could talk about that.
Yeah, absolutely. I mean, I didn't have enough photos in there, but essentially, you could imagine both in the refugee camps and in their home villages, they're raising their own chickens, growing their own food, foraging in the forests, and so it's quite different in terms of the source of their foods. So yeah, they may not be changing their diets and trying to replicate cultural foods, but it's not quite the same. And I think that's really where that massive environment part that's missing, that's really coming in.
So I spent about 10 years working on a biotechnology product that went into the built environment. And the single biggest question that I got asked by people who weren't educated in microbiology who would look at our product is, will this make me sick? Because that's kind of the perception about bacteria that we have trained the public to have. And the best answer I could give is, no, this is non-pathogenic, which the backing for that is, really the answer is, no, this does not fall on the list of things that we confirm will make you sick. It's not an answer of, no, this won't make you sick. So in terms of working with the public, and as we start to talk about microbiome and the built environment, how do we engage the public into really understanding or having that conversation in a way that bridges what public wants and what science knows?
Thank you for the comment. I mean, this is right in my area of desire for future work. I've spent a lot of time on issues related to highly complex, highly new, highly sensational issues. When we look at issues of risk, right, and we look at technical risk, it's risk as a function of magnitude and probability, right? And that's how we can actually come to a quantitative assessment of, hazard identification. Peter Sandman, Dr. Peter Sandman, who is now departed, best doctor name there is. He said that when it comes to the public and public sensing and public understanding of risk, you need to add another variable. It's not just risk as a function of magnitude and probability, but you also need to add in outrage. And he talks about outrage factors. And these outrage factors are all of the sense and sensibilities tied to risk that have nothing to do with magnitude and probability, right? These are all of the things that we bring to the fore when we're making sense out of an issue. So he puts all these outrage factors into a book that's free to download. It's a fun one. It's a good read. And these outrage factors includes things like, is it exotic? Is it man-made? Is it voluntary? Or am I coerced into accepting this risk, right? And so I think that that is certainly a part of this. The other notion that I go to is from a communication theory called frame semantics. So this is, as we confront a word, a term, we make linguistic sense of that based on the other words that might seem similar to it. And so when we're talking about things that feel science-y, that can feel uncertain and scary by default. And that's, I think, where we get the interplay of things like the natural versus GE, where we're activating a frame semantic version of this when we're communicating with the public. Different topic entirely, but I ran an experimental study years ago where I used one term was influenza vaccine, and the other term that I used in a different experimental design was flu shot. An influenza vaccine activated that frame semantic, and we saw greater likelihood for people to intend to get their influenza vaccine than their flu shot. Changed nothing else in any other material, but that scientific term was enough to activate some threat level of severity, right? Where influenza needs a doctor's attention. The flu needs chicken soup, right? And when we're communicating to the public, we do need to consider that the terminology that we use, especially in issues like synthetic biology, genetic engineering, we are triggering these frame semantic associations, and we need to be able to do that upfront definition in order to set that first baseline in a way that will allow for communication that is potentially less biasing. And so that's where like the kinds of definitions that I started with my talk try to build that level of openness around these things. Like microorganisms are everywhere, and there's lots of things that are a microbe, not just bacteria, right? Because gosh dang it, that needs Lysol, right? Every bacteria needs Lysol. That's the jump that we make in the heuristics there.
Sorry, I'm going to say something really, really quickly, which is just I always get super uncomfortable when you use the term the public a lot, because from you know, Psychom 101, there's no the public. There are always lots of different groups of people, and you have a really fantastic opportunity when you have a person in front of you, because they're not the public, they're this guy or this person. And there we have the difference between population statistics and being able to have a conversation.
I will definitely tag on. This is absolutely right in my, yes. When we talk about the public, when we're talking about the univariate statistics across a representative survey sample, this is not a perfect set of who can be the public. But when we are talking to policymakers, we do not get to talk about publics. There is the public and there are my constituents. And that's about it. And I work in the federal government in a different capacity, and we have a lot of hardship trying to translate what we're talking about when we're talking about publics when we're going up the chain for decision making.
I get that. Thanks for great presentation. Is it on? Oh, thanks for great presentations. I wanted to, a comment and a question. The comment is just on the risk communication stuff. I mean, I completely get it that we have to be very careful and mindful of the effects of particular framings and so on. But I just want to quickly draw attention to key studies like mainly from STS, like Brian Wynn's kind of classic sheep farmers, and then also a colleague of mine, Mike Pettit, has interviewed, did a history of psychology book on kind of like the risk communication literature and interviewed Paul Slovic and those guys. And they did a lot of work on a few decades ago on kind of like public perceptions of nuclear reactors and all that sort of stuff. And one of the interesting things that he came away with more recently interviewing Slovic is like this is my reading into it. So but there was a little bit of sheepishness, kind of like at the time we were so like, oh, the stupid public, they don't like they got like catastrophizing and all that sort of stuff, and a little bit of sheepishness, like, well, Chernobyl did happen, like Fukushima did happen, So I think there's also the flip side of it is like yes, we need to be precise in this, but also I think also like the humility of listening to the public, and I think this is where the CBPR work gives the counterpoint of like, but there's also stuff like that we don't know, like we don't know like. Brian Wynn, like how does the soil fall and where does the water go when we make our measurements and things like that, and the locals, So anyway, that's just a comment. My question was to Pajau on the CBPR. So I think that what a beautiful kind of like CBPR example. And when you sort of talk, like it's not the diet, and you look to these other things. So I mean, this is what we're trying to get to with the microbiome stewardship thing, is that like there's large environmental things going on, but I'd be very interested. Were you able to kind of like identify like kind of clear things that we can sort of point to and say, look, these are larger environmental things that we know, because it's so difficult to get at that.
Yeah, that's, my disclaimer is that this was the first study of its kind to really kind of do a bird's eye look at what was happening when people were moving here. So for that reason, we didn't actually look at like everything. I mean, we know especially with refugees, mental health is massive. Physical activity is massively less than when they were back in Asia. And so all of those factors were just harder for us to track. It's also like a bare bones study. And so I think that those are important to look into in addition to several of these environmental changes. I think anecdotally from just what I saw, I think there's a lot of depression. There's a lot of just you losing your home, getting displaced. And so I would imagine that a lot of that is severely impacting just their livelihood and eventually their metabolic health as well. I think the biggest association we found was just time. The more time you spend here, the lower your diversity gets, you lose your bugs, and then you gain weight. And so, which we already knew that part. But I think a lot of those other factors we definitely need to look into. We did look at things like breastfed or not, but I think it's too muddled for us to see any signal there.
Oh, it's on. Thank you. So this question is for Chris and Kristen. So I know you're just beginning the survey work, but one of the things that always irks me is when I see a survey demographic and it says 65% white, and then you show all the results, but you don't show the results in a disaggregated way. Because we know full well the experience of structural racism in the United States has people who have been racialized responding to these questions differently. I'll give you a really good example. For a long time in the literature of evolution acceptance, people would always show a correlation between evolution knowledge and evolution acceptance. So in disaggregated data, in data mainly European Americans, you would always find the more the person understood evolution, the more they were acceptance of it. However, we did the first large survey of Black students and evolution acceptance, and we found the exact opposite correlation. In our students, the more they understood evolution, the more they rejected it. That's why, even if you have a short period of time, you still have to disaggregate the data, both by gender and socially defined race.
We're getting there. And we have the data, which is good. And we have you also, which is also good. One of the other directions that I will want to go with this is to be able to do that disaggregation and then look at those differences in concert with some of the other things that I was discussing, like antecedent value predispositions, like trust, like ethics and values and norms that you bring, as well as all of these personal characteristics, because most of those personal characteristics fall out in the light of these other more lived experiences than the color of our skin. And so we will be able to look at that and also be able to look at if things like how people feel about science and technology in society for problem solving trumps those other kinds of personal characteristics in things like where they actually want to go with this new technology. So we have literally mapped out about 15 papers of where we want to go with this large data set. And we're also going to hopefully be replicating this study with the same panel in another year or two. So we will get longitudinal assessment to understand how does public opinion change with not only a next cross-section, but with the exact people. So we will be able to look at individual change as well, which could be really fascinating. No, December 2023, just a few months ago. Hot off the presses.
So this question is for Kristen and Chris, and maybe the other Chris too. This is just a personal question. Why do Americans accept chemicals with no questions, but the idea of bacteria scares the shit out of everyone? Like, we are talking about introducing bacteria to HVACs, but Febreze is totally cool. Like air
it was called a
fresheners,
probiotic, not a chemical. If we called it a chemical, you're going to see very similar activation of that semantic framework, I would say. Everything is a chemical, and chemicals also freak everybody out.
Do they? I mean, I feel like we're pretty chemicals.
I could probably find you some public opinion survey
the
studies.
ingredients in anything you put on your body, right? Like people are like, that's okay. But maybe, and this maybe is for the three of you too. Has public health done too good a job in really indoctrinating us to the idea of pathogens where, I don't know, like bad news for Premier? I don't know. Is it are people ever going to be okay with it? Because there's just such an idea of disease-causing organisms. That's kind of a bummer of a question.
I would go to the generational hypothesis, We have seen across the board of emerging science and technologies over the last 20 years, in the last 15 that I've been doing the public opinion studies, a shift in demographic acceptance on things like synthetic biology, nanotechnology, geoengineering, and the like. Younger audiences are much more accepting of these technologies. I have studies on gene-edited foods that are published that you can read, and we do see a generational divide. I'm sorry, boomers, when you go, they will get accepted more. And that is the simple truth. We are seeing a generational build here. And that comes with experience, that comes with education and familiarity. But that is happening, and we do see that shift. And it's not changing. There's not a plateau where you hit 40 and suddenly you turn into an anti-technologist. We're not seeing that. We are seeing the upward trajectory of acceptance of these technologies as they mature. And that coincides with the new cohorts and generations.
Okay, thank you. Just really,
Thank you. So also, I think that people assume that if it is sold in the free market, that it must be, highly researched, or that the government, puts a stamp of approval on it, when we know that's not always the case, and products go out on the market and they haven't been tested. Like, look at the supplement industry and the vitamin, right? So I think that there's some misperception of trust among some parts of the public too.
Capitalism seems to be winning, yes.
Are there any questions from online?
I do have one online.
Okay, this might be our very last question and then last comments from the panel.
So I do have a question from Kirsty Wissing, who's down in Australia, I believe. And she says, thanks to all the presenters. My question is to Erika. If I heard correctly, happy to be corrected, you mentioned that some ways of studying microbiomes also mean to kill these microbiomes. Could you expand on the ideas of more than human care and efforts to understand microbiomes? And how does violence come into understanding what is microbiome-ish?
Hi, Kirsty. So two parts to that question. The first, most methods of studying microbiomes are destructive of the community. So we have to reimagine what we think the relationships among the microbes that we are inferring from bits of DNA. Like, we have to reimagine what a community is. So that's the reason why I think that the metaphors that we apply to making sense out of what kinds of communities, microbial communities are really important. The point about care, I think, extrapolates a notion, to some extent at least, of care from humans to other creatures that I really would guard against doing. We know well that caring for other creatures does not necessarily mean the same thing as caring for humans. I love baking bread. I have zero problem with killing scads of Saccharomyces cerevisiae every day, even though they are one of my favorite microbes. And that's fine. They live at very short time scales and they live at very long time scales, and the cultures of Saccharomyces that I'm working with don't care if I bake bread. This is unimportant. So I do think that we can ask, what does care look like in a multi-species context, where we're asking, how do we take good care of environments that many different creatures have to share, where we don't necessarily begin with the assumption that killing is bad, or that violence looks the same for every creature, and we instead ask, how do we build environments that fundamentally allow the creatures who, and here we get really fuzzy, have a stake in that environment? I mean, there are all kinds of questions, and who belongs and who doesn't, and those are the fuzzy bits. Those are the really hard bits. But we can ask, what does living well together look like, and then have a conversation about what relationships we're trying to sustain when we talk about sustainability, so that we're not just sustaining capitalism, we're sustaining communities.
The C word. Somebody finally said it.
Oh, I didn't get there first, but yeah.
All right, thank y'all, and a round of applause for our panelists. Thanks so much. Joe, but I'd love to introduce you if I could do the honor. Okay, I'd like to introduce Dr. Joseph Graves, Mackenzie Scott Endowed Professor of Biology at NC-A&T, and the Associate Director of Premier. Dr. Graves.
All right, so how do I make the screen show this presentation? Oh, it just did. So I'm going to try to make these comments quick. They came forward after our conversation about the concept of race in this morning's panel. I began thinking about this problem in the early 1990s. My first book on this was entitled The Emperor's New Clothes, Biological Theories of Race and Millennium, at the Millennium. And in this book has not really lost its importance over the last couple of decades because most of the people doing work about race don't understand the history of the concept. And so these mistakes are made all the time in scholarly literature and conversations that I've been engaged in for the last 30 years. Shortly after The Emperor came out, the race myth, which assumed you knew the history of racial thinking in the Western world, examined the ongoing pathology. In both of those first two books, I talked about race and health disparity from an evolutionary perspective, which I'm going to continue to discuss here. But after 20 years, Dr. Alan Goodman, former president of the American Anthropological Association, and myself recognized that there really hadn't been much progress, that people had the same sets of misconceptions. And so we wrote Racism, Not Race, Answers to Frequently Asked Questions in a question-answer format. And we've literally took every question we heard over the last 30 years, every misconception, and we dealt with it as a question in a chapter. Now, there may be some we missed, but I think if we did, there are very few that we missed. Now, another thing I've been involved in the last 3 years is directly taking on racial misconceptions in medical research and clinical practice. So I was called on as a consultant by Elsevier for the revision of the new edition of Robbins and Kumar's Basic Pathology, now in an 11th edition. Now, we were able to get 75% of the racial misconceptions out of that book. We couldn't get it all because most of the time we were arguing with the other editors who, believe in race like it's gravity. And so, but we still made some very important improvements in that book. And the reason why these improvements are important because Robbins and Kumar is the most widely used textbook of pathology in the world. So we were able to get in a section on health disparities from social determinants of health. That had never occurred in that book before. We were able to put in inclusive images showing diseases that manifest in the skin or diseases of the skin in people who were not fair complected. Virtually every pathology textbook in the world shows them in fair complected people. So these were important accomplishments. Now with my good colleague, Dr. Andrea Darup, who teaches the pathology course at Duke University, we were able to take on many of these misconceptions in medicine directly. So we had a piece in the New England Journal of Medicine, my piece in genealogy called The Myth of the Genetically Sick African, the piece we did in race, in the reading, a study of problematic uses of race and ethnicity in a prominent pediatrics textbook. We literally found hundreds of examples in the textbook used by the American Academy of Pediatricians. And this paper came out in March. 2 months later, that same academy put out a statement decrying racial medicine. Now, they didn't cite our paper, but they felt compelled to make changes. Now, I just told you what the book is about, but there's a couple of other papers that came out last year that I think are really important for people who don't know of them. The first is the paper in Evolution Medicine and Public Health entitled Embedded Racism, a Critical but Neglected Health Determinant in Evolutionary Medicine. Now, this paper is nominated for the Omen Prize this year and was the most cited paper in the journal for several months. Also, my paper in the Cold Spring Harbor Perspectives of Biology, Favored Races in the Struggle for Life, Racism and the Speciation Concept, and this paper in Nature, Behavior Genetics, Population Descriptors, and Behavioral Genetic Research. This is based upon the report of the National Academy of Sciences, Population Descriptors in Genetic and Genomic Research. So for this group, the chapters in R&R that are most important are Chapter 1, what is race, racism, and human biological variation? Chapter 3, everything you wanted to know about genetics and race. Chapter 5, why do races differ in disease incidence? And Chapter 6, life history, aging, and mortality. Now, I'm going to say this is said all the time. Race is not a scientific concept. It's become a platitude amongst scholars in the humanities and the social sciences. But I feel that this is wrong because it conflates two very different concepts with two very different intellectual histories. First, the biological race concept, which begins in the Western world in the 18th century, and it travels a journey from morphology to geographical location to population base in the 20th century after the neo-Darwinian synthesis. And I do want to point out, for most people who are unfamiliar with this journey of the biological concept of race, for the most of the time that this concept existed in the Western world, it existed on the foundations of special creationism. It has only recently became part of the evolutionary lexicon, and that was with the neo-Darwinian synthesis in the 20th century. And then later in the 20th century, whether a group could be considered a phylogenetically or evolutionarily unique lineage. Now, the other thing about this biological race concept is that it applies to any species that is biparental. So when we think about socially constructed or socially defined race, we're thinking about an idea that only ever had its utility in the human species for the purpose of oppressing different groups of humans within the structures of colonialism, racism, slavery, and genocide. This concept came into existence for one purpose only. It was not an objective examination of human biological variation. Now, these two concepts had a dialogue across the time in which they came into existence, but they do really deal with entirely different things. So, now just to quickly give you some reasons why humans don't have biological races, which is what I say. I never say race is not a scientific concept. I say our species, anatomically modern humans, does not have biological races. There are some species where this category has meaning and is useful, but not in ours. So first and foremost, physical traits can't be used to give the word I'm looking for is repeatable or meaningful differentiation of different groups of humans. Because if you use physical traits, you will get groups that are in complete disagreement with our evolutionary history. So a good example would be, nobody here would doubt that Swedish and French people look more like each other, and that's at the top, than they do people who are called Eskimo in this study. But that Eskimos look more like Swedish and French people than they do North American Indians, or that North American Indians look more like Swedish and French people than they do South American Indians, makes absolutely no sense in terms of the migratory history of our species. And similarly, in the bottom portion of the cladogram, the idea that sub-Saharan Bantu language-speaking people look more like Australian and Papua New Guineans doesn't make any sense evolutionarily either. And I give this example of a person, I show this picture, a Solomon Islander, and I ask my students, where's this person from? And they all say Africa. And when I tell them, actually, no, this person is from the Solomon Islands, they're surprised. So, at the end of the day, biological criteria for races fail in humans. And even though we have apparent physical differences, they actually reflect a very small proportion of our genome. And I'm going to demonstrate that quickly. So one of the ways that we can address this is to ask questions about, well, how similar are populations genetically? And there are a number of population genetic statistics we can use to examine that. And when we do that, we find that we have continuous genetic variation across the range of which our species exists. And there's at really no point can we see any clear, distinct breaks between one population and another. So this is using the population genetic substatistic, population subdivision, and we see continuous gradual change in genetic similarity across the entire range of our species on this planet. Furthermore, most of the genetic variation that exists in our species is not due to natural selection at all. It's due to genetic drift. And this is due to the fact that people migrated around the globe in small groups as opposed to whole groups of people moving all at once. And so by chance alone, genetic variation was lost. And so there's a linear relationship between genetic variability in Africa, where our species first evolved, and the last places that we began to occupy, which was the Americas, sometime around 30,000 years ago. Now, on that journey, there were some adaptations, but now I want you to count. These are traits for which we have strong evidence of adaptation. Now, in our genome, there are 21,000 or so protein-coding genes. Now, on this map, there's maybe 30 genes or 35. So we don't have a lot of adaptation in our species. So things that are obvious, lactase persistence, not so obvious. Now that might have had some impact on microbial gut community, or skin pigmentation. That is one of the obvious examples, certainly impacts skin microbiome, but also we know impacts gut microbiome. High altitude impacts skin microbiome. Things like high-fat diet, pretty sure that impacts the gut microbiome as well. Things like thick hair probably do, and so on, starchy food. But we're not seeing 21,000 proteins, we're seeing about 35. Now, if we look at polygenic selection, that has been relatively recent and is not very strong. So in Europeans, the strongest selective signature is in skin color. And by the way, for those of you who do not know this, the fair skin phenotype is only about 10,000 years old. So our species is 300,000 years old. When it first entered Europe, it had tropical skin colors and tropical body dimensions. It took tens of thousands of years before adaptation to less solar intensity led to lighter skin phenotypes. Blue eye phenotype is only about 6,000 years old. So now, looking at the diversity of our species, okay, if you look at the width, do we have a pointer here? No pointer. But if you look at the width of genetic diversity illustrated on this diagram, you see Africa represents clearly the vast majority of the genetic variability in our species, followed by the Middle East, followed by East Asia, followed by the Americas, followed by Australia, Melanesia. And that's why I say that we are all Africans. The only difference is when your ancestors left Africa and under what conditions. So now, we don't have biological races, but we do have health disparities. And so what I argue is health as a complex phenotype is determined by the effect of genes, environment, gene by environment, the covariance of genes and environments, and the error in which the traits are measured. Now, in some of these traits, the error can be quite large. Things like, for example, cognitive function. I would argue the error term is not insignificant. But things like height, for example, I think most of you could go around the campus and measure the height of individuals and not make too many mistakes. Now, what we find is that there's actually a very small effect of genes, a much larger effect of environment, a much larger effect of gene by environment, and a much larger effect of the covariance of genes and environment. And what that means is that different genotypes will have negative associations with specific environments. So I have argued that in the United States, the health environment and persons of African descent are generally negatively correlated. And that accounts for the health disparities we see. Now, why do I say that genomics doesn't do it? This was a study done in 2008 in which the researchers looked across European, American, and African American genomes. And they were able to characterize genetic variants into two categories: those that were possibly deleterious and those that were probably deleterious. So for possibly deleterious variants, there's no difference. But we find for probably deleterious genetic variants, European Americans have more of them compared to African Americans. Now, flip forward 20 years. The All of Us Genomics Consortium looking at 245,000 whole genome sequences. And here plotting genetic variants that are, again, likely to be clinically deleterious. And we find, once again, in a much larger sample of individuals, that European Americans have more possibly deleterious clinical variants than African Americans do. So once again, we cannot make the argument that the health disparities we see are the results of genes. Because if that were true, we would see the exact opposite pattern. We would see European Americans displaying more problematic health phenotypes and African Americans being healthier. So this brings us to the report of the National Academy of Sciences, which I strongly recommend that you all read if you have not read it yet. Using population descriptors in genetics and genomics research, a new framework for an evolving field. And as you might have guessed, I was literally the first person that the committee cited in the report, where I say our social conceptions of race and ethnicity do not match the underlying biological and genetic variation within our species. And we should never confuse the things that were created for the purpose of oppressing people with the nature of that biological and genetic variation. Now today, we had the opportunity to discuss the microbiome writ large, ethical issues with regard to how we study it, the impact of what could possibly happen with regard to engineering it, and how those changes might impact different sectors of our society. I thought we had a really well-constructed agenda, really excellent discussion in all of the sessions. And so I want to give the opportunity for anyone on the panels or anyone in the audience who has a comment, statement, or question to the group to let it fly. How much more time do we have, Jennifer?
5 minutes? 1 minute.
1 minute. I'm sorry, I didn't mean to take up all the time, sorry. But I'm gonna go sit down because I don't need to be up here.
We can do that tomorrow. So
Sure.
thank you, Joe. So we really took a trip around all the, well, not all, but many and diverse societal and ethical implications of the microbiome of the built environment. And I want to thank you all for your presentations. I agree with Joe that they serve the purpose of this meeting well. And tomorrow, in person only, unfortunately for those online, but you're welcome to to come register to be in person tomorrow if you're in the local area. We will have more time as a community to deliberate about all the bioethical issues, the social equity issues, the risk governance issues, and the public engagement and participation issues that we've discussed today. And so this is just the beginning. I realize it was a smattering of all types of issues, and tomorrow we're going to bring it into a comprehensive framework of all the things that we can do for research, action, and integration of SEI into scientific research. So thank you all, and I'll just stop there.